Testing. Nicholas, can you say something?
Yes, hello.
Hello, we can hear you very clearly.
Oh, wow. Okay. Good morning to Boston from Berlin.
Yep. Good morning, everybody. I think we'll kick us off. Yeah, so yesterday I think we had really productive discussions, a lot of exciting things that got shared yesterday. So let's, I think, in the same spirit, uh, continue these discussions and uh, continue, discussing ways that we can improve, um, single cell technologies. So our first speaker today is going to be Professor Nicholas Rajewski. Professor Nicholas Rajewski is the founding director of the Berlin Institute for Medical Systems Biology at the Max Delbruck Center and professor at Charity University. He's made enormous contributions to the fields of RNA biology and gene regulation, notably the elucidation or the identification of computational target prediction of microRNAs and also identification of circular RNAs in animals. More recently, his lab has developed strategies such as OpenST, which enables high-resolution single-cell spatial transcriptomic mapping in complex tissues. So let's please welcome Professor Rajewski.
Okay. If you want me, I can start my presentation. Okay.
Yep.
You hear me? You see my slide?
Yep. You're all good.
Okay. Thank you for inviting me, and I thank you, Nikolai, and colleagues. I'm sorry for not being able to be in Boston. I wish it would be different. I love my colleagues and friends in Boston and the US in general. I spent some time in Rome. Just came back to Berlin, but you can see in the background the Peter's Dome, or where the new American, actually Pope, is residing. Okay, so this doesn't work at all. The figure is not showing. Yeah, I don't know what's happening here. I had to use a different computer because of technical problems with the Zoom account. I just hope it will not continue like this. But my lab for the past 9 years has been interested in disease trajectories to understand the onset of diseases, to understand the molecular mechanisms to predict the disease trajectories, and of course trying to prevent them. And we just had some publications about tumors and tumorigenesis and spatial transcriptomics. Okay, so this will not be working like this. Can't. I have to use the non-presentation mode for some reason. Otherwise, I just can't show you the slides. Do you see now the? No, this is completely crazy. I don't know what this has in my entire life not happened to me. I don't know what's happening here. There's a major malfunctioning of the um, of I don't know what to say.
If it shows in non-presenter mode, I think that's okay too.
But it doesn't really. I'm just unable to explain this.
Do you need to swap, maybe we can swap speakers until you figure it out? Oh, did he leave? Do you want to present then? And then I can just message him separately? Because I don't know.
Yeah, I can go if that's,
Okay.
Sure.
if
Yeah.
that's helpful.
Okay.
Okay. Yeah. Let's do that.
Yeah.
Okay, yeah, technical difficulties. So we're going to move on to Scott Coyle. Professor Coyle is an assistant professor at the University of Wisconsin-Madison at the biochemistry department. Professor Coyle completed his PhD work in the lab of Wendell Lim, where he studied signaling proteins and how they evolved new allosteric interactions, altering their activity and leading to potential functional divergence. More recently, he's developed, I think, really truly creative ways of cellular biosensing using frequency modulation of streaming single cell data. So I believe that's what we're going to hear about today. Please welcome Scott Coyle.
All right. Okay, well, hopefully we'll get to hear that exciting talk on spatial RNA and protein omics shortly. First, I just want to say thanks for the opportunity to speak at this meeting. I am not by any means a single cell proteomics person. I am a single cell, I have a lot of interest in single cell biology. Whoops, sorry, just trying to get this working. And so I've really enjoyed listening to all the talks and learning about kind of the scope of the data that all of you are now able to generate and collect at a single cell level using these methods. It's mind-boggling to me. And what I'm going to tell you a little bit about today is imaging-based tools that can't really necessarily capture the kinds of scope of data that you guys are able to capture using proteomics, but it does give us an advantage to look at individual cells over time as they perform various tasks. And I'll tell you about some of the imaging technologies that my group has been developing that we think can enable and improve our ability to make internal state measurements inside cells using these sort of psychedelic waves that you're seeing here. And so our motivation really is that cell biology, so the activity of cells, is animated by dynamics, proteins' activities being organized in space and time. So when a cell is crawling around like this, the activities of proteins are being organized, and internal states inside these systems are changing over time. And that's what's powering the ability of these cells to perform tasks like interact with their environment and process information. And the activity of individual cells is, of course, integrated over much larger length scales in organisms where all of the individual cells working together, powered by those internal protein dynamics, organize complex processes like development. This is a video taken by my former colleague, Jan Huisken, of zebrafish vasculature development. And so within my group, our kind of overarching goal is we really want to be able to understand how the gears of cells, so sort of the internal protein machines and inner workings of cells, turn in real time as biological processes unfold. And really there's kind of two themes to this approach that we're interested in terms of single cell biology. One is an aspect of measurement. So what are the internal sort of hidden states or activities that are driving cell behaviors we see, and how are they changing over time? And secondly, we're also interested in perturbation, which is how does the spatiotemporal organization of molecules inside the cell impact behavior, and what kinds of behaviors can cells realize by simply reorganizing molecules and components in different ways. And what I'll tell you about today are some approaches that my group has been taking in which we've been developing genetically encodable tools and technologies that generate synthetic protein dynamics inside cells, particularly oscillatory dynamics, and how those kinds of synthetic protein dynamics can actually enable a lot of progress on both of these fronts. And I'm going to kind of try to prioritize maybe the measurement aspect of this, but I saw this morning that I had like half an hour to talk, so maybe I'll spend a little bit more time on perturbation as well, depending on how things go. So to give you some context, why or how might the ability to create synthetic protein dynamics assist us in an ability to make single cell measurements? So in my field, where we use a lot of imaging to query what's going on in cells, usually the way in which we follow dynamic cell states is through fluorescence microscopy, taking images like this. And the way we do that is we use cameras to collect the intensity of fluorophores at some moment in time. And that intensity provides sort of the direct readout of what we're interested in. Now, some issues with this approach, even though it's very quantitative in many respects, is that these units are arbitrary and they're susceptible to a lot of artifacts and unwanted effects like photobleaching, illumination artifacts, or instrument-to-instrument variation. And furthermore, these signals lack metadata, which connects the source of the photons that we're measuring, that is individual cells, to their contribution to the fluorescence measurement. So there's not really necessarily an effective way to separate out or multiplex, sort of the way I observe many of you guys doing with TMT or other kinds of tags. You can't sort of stack data from the same cells in a single fluorescence channel. And this method of acquiring data, having objects transmit information to us, kind of contrasts with most other forms of information transmission or measurement that we make either in our daily lives, or I've really enjoyed the discussion about Orbitraps and things like that, where these you can acquire a lot of powerful information through studying sort of oscillatory dynamics. And so usually, like when wireless signals are transmitting information to all the observers on the Zoom call, what's happening is that the signals of interest are being used to modulate the temporal structure of an oscillating carrier signal. And this carrier provides a very powerful information-rich handle that actually allows many, many kinds of data streams to be sent or transmitted on the same medium along different unique channels based on the underlying frequencies that we assign to different senders in the population. And so a general interest of my group has been, how can we create biochemical analogs of these kinds of carrier signals that we can potentially use to improve our ability to make single cell imaging measurements or perturbations? And so the approach that my group has been taking has been to identify simple systems that can generate a rich space of spatiotemporal dynamics that can be implemented at the protein level. And we drew inspiration from a class of self-organizing systems called reaction diffusion systems. So these are models for how interacting chemical species can self-organize themselves through dissipative processes into either spatial or temporal patterns. And the ideas here is that a certain combination of reactivity and nonlinear reaction terms in the presence of diffusion can sort of spontaneously give rise to self-organizing phenomena. And this is something that was conceptualized by Alan Turing a long time ago to explain patterning in animals, and has since been sort of an object of study for physicists and for some time. So to find protein-based reaction diffusion systems that we can deploy for synthetic applications inside cells, we turned actually to a class of bacterial patterning systems that are found in nature that are unique to bacteria and are not found in eukaryotes. And what these bacterial patterning systems do is that they use the energy of NTP hydrolysis to organize a reaction diffusion process that can position molecular cargoes in the cell. So basically, there's a wide family of these proteins, and they all work by basically undergoing a nucleotide-dependent interaction with the surface that can be displaced through the activity of an activator protein that will stimulate hydrolysis. So something assembles in a nucleotide-dependent manner, it recruits something that can trigger ATP hydrolysis, and then those molecules fall off. And this process is used by bacteria to actually generate very stunning oscillations inside themselves that they use to perform various kinds of tasks. So if you were to look at an E. coli, which many of us just use for making plasmid DNAs or mini preps, to prepare for cell division, they instantiate these waves like you're seeing here inside themselves. And what they're actually doing is they're using this oscillation that they've created to perform a computation which is to locate their midpoint, because this wave will actually have a nodal structure where the node will appear in the middle of the cell, and that will be the location where the cell divides. So they actually just use this wave phenomena to compute a geometric property. And there's many examples of these systems all throughout biology. Some of them are used to segregate plasmids in these bugs or space complex organelles like carboxysomes. So there's an enormous space of these systems just sitting out there being used by bugs to create interesting dynamics. And when I was a graduate student, an interesting paper was published by Petrischwil's group reconstituting, showing that basically these two proteins plus membranes were sufficient to generate self-organizing phenomena. So if you just take an ATPase and activator from this MINDI-E family of patterning systems and you throw them on a supported lipid bilayer, they'll spontaneously begin to organize into these wave structures you see. So these two proteins are sufficient to generate these kinds of phenomena. And so given the minimal requirements to produce in vitro self-organization, my student Rohit wondered whether we could potentially exploit that simplicity and compactness of this system to generate synthetic protein dynamics in eukaryotes where these proteins don't exist. And so what he did was clone and deliver these components into eukaryotic cells. This was actually a rotation project that he started. And by the end of his rotation, he had shown that we could, when these proteins go into cells, you could generate traveling waves inside these human cells like you see here. You could generate very fast standing oscillations, and you could even generate stationary patterns, the kinds of things that Alan Turing was imagining. Here's this, my mom really likes this because it's sort of a leopard printed organization of these components here. And he saw all of these things actually from a single transfection of cells. It was pretty cool. And I'll explain what gives rise to the different phenomena we see in a little bit. But beyond the beauty of this, I'll explain many applications. But before I do, I just want to go on to say that since his initial discovery, we've been able to generate these kinds of synthetic protein dynamics in all kinds of eukaryotic systems. In fact, we haven't yet had one that hasn't worked. So, I had a student that wanted to do high throughput screening in yeast. It's very straightforward. You put them in the yeast, off they go. We are working with a collaborator where we can put these into primary neurons where now the waves propagate all through the neurites up and down the dendrites. It's very fun to look at. But we've also started to put this into more multicellular context. So we can put these components into tumor organoids where now each individual cell within that structure is beginning to produce these kinds of oscillations. And recently with a collaborator in Claire Richardson's lab, our student Nikita has put these into worms. So there's a C. elegans now where we have epithelial cells that have been labeled with these proteins. And to the best of our ability, it seems like cells are very tolerant of these waves running through them because they have no endogenous interaction partners in the cells we put them in. So that's all very visually interesting. We can put them in the nucleus too. I forgot to put that slide up there. But that's all very visually interesting. But the real power that we believe that these systems have isn't for making my mom feel excited about my research for the first time in my career, but it actually has to do with the temporal structure of the fluorescence signals that we're able to observe from these proteins. And so Rohit actually had an NMR background, and so he noticed when we were looking at these videos that if you look at the time trajectory of any individual pixel over time, you'll get this really, really nice oscillation. So this isn't sort of like a bursting pulsatile pseudo oscillation. This is a very consistent one. And as an NMR person, he wanted nothing more than to apply mathematical techniques for analyzing oscillating signals like FFTs, Fourier transforms to these. So you can take pixel level data and map it from the time domain into the frequency domain. And what happens when we do that is that you get basically a very crisp, solid peak that arises from these oscillations within that cell. And if you apply that technique and cast it over all the pixels in the image, individual cells, like this little pair of recently divided cells up here, will light up at a very specific frequency slice within the transform image time series. And what this means actually is that if we have different cells in the population oscillating at different frequencies, then they will appear on different slices in this when we Fourier transform these images. And Rohit developed a technique based on color coding these cells by frequency to help you visualize the output of that, where now many different cells in this population are being imaged in one channel, but the signals are being mapped and colored by the frequencies of the cells that are there. And this has a variety of interesting applications, which is that we can actually sort of see through cells that are overlapping with one another. So fluorescent signals from overlapping cells contain multiple frequencies, which we can deconvolute in the frequency space using Fourier methods. And this gives us a very interesting sort of approach in which instead of the photons that we collect being agnostic to the source of where they came from, the cell's own biology is sort of coupled into the fluorescent signals that we measure, giving us this unique cell-specific fluorescent signal that we can track and analyze. And this is something where now if we think about some of those neurites, what we've been able to do is do things that are sort of like Brainbow's type of imaging, where individual neurites that are highly overlapping or complex can be mapped into this frequency space to enable things like neurite tracing in very complex overlapping environments. So with some amount of time that I have, and I really don't know how long I should go, I'll tell you a little bit about the goals we have of sort of turning cells into streamers using these sort of synthetic protein wave approaches. So I think I'll briefly talk a little bit about the platform that we developed to be able to control the kinds of oscillations that we generate, because in order to use that sort of technology, we really have to understand how to control the frequency and amplitudes of the waves that we can generate. And then I'll tell you how we've used that knowledge to design circuits. So these are biochemical engineered versions of these components that enable us to stream interesting kinds of internal state data about the cell using either AM or FM modulation approaches. And then if I have any time left, I'll also tell you about how we've been sort of using this approach in reverse, sort of inspired by my son's recent interest in RC cars, where we can perturb or target cellular processes with this inside the cell with these oscillations to actually explore different kinds of dynamic constraints or make new kinds of dynamic measurements inside cells. So and if you're interested, we had a paper about this last year, as well as a few new preprints that you can read to learn more about this technology. Okay, so to control the oscillations that we observe, there's really two kinds of levels that are at play here. There's a genetic level in terms of how we deliver these components. What the expression levels of those components is going to have an impact on what kinds of phenomena we see. And then there's a protein level effect. These are kind of more biochemical effects in which, depending on what components we use or mutants or what species we derive from them, various reaction constants or rate constants within the reaction cycle powering this change, and that changes how expression levels map to frequencies. So Rohit developed this pipeline in which he collected thousands and thousands of single cell oscillating data, used that FFT approach to measure the frequency and amplitude of a wave as a function of the concentration of the ATPase or activator in the cell. And he identified some really nice trends. I guess they came. Does this appear? No, okay, sort of. He identified some really nice trends, the main thing being that the ratio of the ATPase and the activator was what was setting the frequency that we observed. And that sort of makes sense. The activator controls the rate of ATP hydrolysis, so more and more of that activator will cause these waves to go faster and faster, whereas the ATPase itself controlled the amplitude of the wave. And this also sort of makes sense because in order to produce a wave, the ATPase assembling in the first place is rate limiting in this process. And so he has sort of this little programming language that tells us how these concentrations map to the waveform that we're measuring. And this structure actually revealed some interesting things that's kind of nice. So that tells you that a wide range of frequencies and amplitudes are available to a cell. But if we generate a single copy clonal population, the frequency within those cells will actually tend to be extremely robust, because even though cells may vary in their overall total bulk expression levels, the fact that the frequency is a ratiometric property tends to lead to the system only being kind of buffered against all that extrinsic noise. And that means that when you follow clonal population, like over here on the right, for more than 24 hours of continuous imaging, these cells maintain this frequency over time. And that's very helpful because it means that we can potentially use frequency modulation to encode information in these systems effectively. And yeah, we can make different clones and stuff. Now I will add, and I won't spend too much time on this at this point, I don't think, but I'll add too that although this suggests that we have a wide range of frequencies and amplitudes available to us, any specific capability for using these components is constrained by this phase portrait as well. What I mean is, if you wanted to have, if you wanted to have a red cell here, you can't do it because the protein's biochemistry doesn't support giving you that behavior. So working with an undergraduate in our group, we actually had Rohith and this undergraduate Tommy cloned a variety of different evolutionarily related components that varied in their sequence composition. And they tested these and built phase portraits for those components and found that they could actually find a number of species where the expression levels could be the same, but the oscillation dynamics could be very different. So here's kind of a panel of slow, medium, fast, and extremely fast, all being realized at the same expression levels. And they went on to show that you could kind of mix and match these different components together to create a very large bandwidth of frequencies available at a very modest frequency range. And an interesting thing that will come up later before I just move on past kind of the core part here is that they also found that you could compose multiple activating components together into one system where the ratio of those components will control the frequency that we observed. And this actually suggested to Rohith a strategy for building frequency modulation circuits that I'll describe a little later. So this main point I just want you to take away at this point is that this combination of expression level control with a lot of protein components at our disposal gives us an excellent ability to control the kinds of oscillations that we can generate. Okay, so now how do we go from the ability to kind of give a cell a unique frequency to encoding data in that structure that we can recover? And again, I want you to be kind of thinking that we want to emulate or sort of try to replicate this paradigm of something inside the cell is changing that we want to measure, and we're going to use that to manipulate the structure of these waves over time. So I'll describe, I guess before I do, I just want to mention that we've developed a variety of tools to analyze these data based on digital signal processing techniques. So when you have a time varying, when you have a non-stationary signal over time, a Fourier transform is actually not the correct way to analyze these signals. So we've developed a lot of ways to apply either kind of FIR filters, Hilbert transforms, or wavelet tools to the signals that are generated by here. I won't go too much into it, but thanks to Zuckerberg's team of PyTorch nerds, we can accelerate these processes because these are essentially large tensor type structures and do this kind of image processing very efficiently now. So armed with that toolbox of computational approaches, I'll describe kind of two ways in which we can encode data. One of these is an amplitude modulation strategy in which the idea is to simply have a second fluorescent protein inside the cell that can conditionally interact with the waves that are going on inside the cell. And the idea here is that in the absence of any interaction or any signal promoting the interaction of these components, this blue protein is just going to be sitting in the cytoplasm doing nothing. However, if a signal promotes or favors the formation of an interaction between two proteins inside the cell, as many signaling processes do, this blue protein will now bind or co-assemble with the wave components, and now that blue protein will begin to co-oscillate with the wave that we've generated. And what this means is that the blue protein now is a second signal that we've generated in the cell, whose amplitude is going to be modulated by the level of protein-protein interaction that is occurring between those two components. And we kind of like this because it's a very biochemical, we're going to read out the actual biochemical interaction between these two components directly. And so I'm going to present an example that we've done this now for many different systems where we took a sort of model kinase signaling pathway protein kinase A, which is activated by cyclic AMP through G protein coupled receptors. And so there's a pair of proteins that interact in a phosphospecific way. And so one of those, the FHA domain, will be fused to our wave generating components. And then the substrate for pKa will be fused to a second protein. So if kinase signaling is occurring, these proteins will interact and we should see co-oscillation. So I'll show you an example here of what this data can look like in practice. So the top is a carrier signal. So that's an oscillation that's always running in these cells at all time. And the bottom is our what we call the data line, which is the blue protein whose co-oscillation we're going to be looking for. At a certain point in time, indicated by this graph, we add a GPCR agonist called isoprenaline. And when we do, you'll see the blue protein goes from being non-oscillating now to co-oscillating very strongly with these components. Okay, so we're seeing that interaction between those proteins visualized for us by these wave structures. Now, what's really nice is there's a lot of tools from electrical engineering we can just borrow from to do quantify this co-oscillatory behavior. So every pixel in that image that we collect provides a readout for the interaction that we're interested. So we have a carrier signal on top, and below we have this data line that upon the addition of isoprenaline begins to immediately co-oscillate. I realize for this audience, maybe some time scales would be helpful. We're talking about that whole trace there is about 3 minutes of video. So you see it almost instantly upon the addition of this compound. And then there's typical techniques of using power ratioing between these signals to quantify at a pixel level everything that's going on. And so this turns every cell in the field of view here into a display where when we add isoprenaline, cells are going to light up in this blue channel as their signaling activity goes up. And then we can analyze using a variety of cell segmentation things, the different kinds of response profiles we see. What's also nice about this is there's a lot of ways to calibrate these signals using, for example, soluble or cell permeable cyclic nucleotide analogs so that you can go from the amplitudes that we measure now to the true internal concentrations of the CMP that we think are going on inside these cells. The system works also, can be multiplexed at the level of different signaling pathways by simply putting more reader and substrate interactions onto the same protein. So we've done this for PKA and PKC simultaneously, which has been pretty cool. But I do want to mention, so maybe some of you are thinking like AM radio, isn't that why our country's in the toilet? Can we move it to maybe the FM world? And for those of you who aren't familiar, one reason why FM is sort of gives us a much nicer, higher quality signal is that it's very, it shows what's called excellent noise rejection. That is, intensity fluctuations in an AM modulated signal can creep into the data and corrupt it. But if you encode information in a dynamic property alone, this frequency itself, then it doesn't matter if the signal goes up or down in amplitude, all the information is encoded in the dynamics directly. So Rohit decided to develop what he calls these GeoFM systems for frequency modulated data encoding, where the idea is to exploit the fact that we can have more than one activator in this system and still produce functional oscillations. And so his strategy is very simple. He'll always have the expression of one ATPase activator pair, but he'll couple the activity of a second activator molecule in that system to a biological process of interest. So this could be transcription, proteasomal degradation, any number of different things. And to figure out whether this would work or how to best make this work, he did a lot of simulations. I won't really go into it here, but he basically identified kind of the composition that you would like to have in order to get a very strong amplification of small changes in protein levels delivered into a change in frequency. He also developed a way to calibrate these circuits based on measuring the frequency of all possible combinations of these components all at the same time, and built a machine learning model to invert this mapping. So I'll show you just one proof of principle that we've implemented on this. So he just wanted to show, for example, that if you take an existing transcriptional reporter circuit, in this case, a DOX-inducible component here, you can basically have DOX drive expression of the faster activator component and then see the frequency modulation over time. So in the absence of any DOX, you can follow cells over time and you'll see that they just kind of crawl around and they stay at the same frequency even when they divide. So you'll see them kind of looks pretty fun, but they stay blue basically in this video that I'm showing you over here. And then in contrast, if we add DOX to these cells, this clonal population over here will begin to get faster and faster and faster over time as the frequency increases. And he was able to quantify a variety of nice FM responses for cells with different sort of backgrounds and then use his model to infer the transcriptional states at play. And so these kind of AM and FM strategies for data encoding that we have enable us to combine the power of the sort of frequency barcoding method I showed you with other strategies for introducing to map data onto those structures to recover. And I just want to think some of the pros and cons of these different strategies. The AM strategy tells you about two proteins that are in direct interaction. So we're really measuring that interaction as it's occurring, and we're just seeing it sort of displayed for us on the system. So these are very straightforward to design. These FM strategies that I presented as well seem very actually well suited for things like transcriptional state measurement, or things where the levels are changing perhaps more slowly over time. And an advantage of these FM encoding strategies is that these measurements we make really are true and independent of any sort of intensity effect. So if you measure at different magnifications, if you measure in different instruments, that frequency is a true value that is not arbitrary, and that gives us a lot of ability to collect wide field or zoomed in data that can be integrated very nicely. So, and we have a lot of collaborations with people on campus. If there's something interesting you would like to measure with these, you can always reach out, where we've been looking at effects of chemotherapeutics on PKA signaling in pain sensing neurons. We have our own funding to look at oncogenic signaling in tumor organoids. And we have another migration-oriented project with Anna Hudenlacher's lab. So I think rather than talk about the last thing, I'm just gonna park it here and jump. If anyone's interested, I can show you some cool things that we can do. And I will leave Mark Zuckerberg alone for a moment. And I hope at this point you've seen, while we certainly can't make the same measurement scopes that you have, the idea is perhaps by integrating the kinds of large data sets that can be made from single cell proteomics, we can find sort of like Nikolai was talking about with Katie, a subset of particularly interesting measurements that we might want to make dynamically over time. And so I think there's actually a lot of opportunity for synergy between these different approaches. And I'd love to talk more with all of you about these. So thank you for your time, and thanks for the people that gave us the money to do this kinds of research.
Questions for Professor Coyle?
Nice talk, and good to see this MinCD proteins after a long time. So my PhD work was on the bacterial cell division. I worked a bit on this stuff. My first question is that, so this MinCD, though it has ATP, ADP binding site and it works, but mostly in the bacteria, it works along with the FTHZ or cell division proteins. That's what it functions. The question is that, but when you express this protein in the eukaryote or in other cell lines, does it fold exactly the same the way it is in the bacteria?
So are you asking if I were to include FTSZ, what happens? Or are you just asking what?
Even because you express the MinCD proteins in the eukaryote.
We only do DE, no C.
Yeah.
But
yeah. So how is the 3D structure? Because bacteria is a prokaryotic and eukaryote, and there is no cell organelle in the bacteria, whereas it's fully organelles in the eukaryote. How it get localized to its specific thing in the eukaryotes, how different it is from the prokaryote to eukaryotic.
Gotcha. So I think I think the question was about kind of the differences in the 3D structure of eukaryotic cells versus bacteria and the different kinds of lipids. So how does it choose which membranes to run on and things like that? Yeah, so when we started this in bacteria, there's no endoplasmic reticulum, and so these waves propagate on the exterior of the cell. And so I actually wasn't sure whether this approach would work when we tried it in the eukaryotes, because oftentimes to make, we thought maybe these waves would run around the edge of the cell and be hard to see. And instead, we kind of saw this beautiful space-filling wave occupying the cell. And so it turns out, and I didn't show any data for this on this, but they're using the endoplasmic reticulum to help support the wave propagation. And what that, it seems like the membrane composition of endoplasmic reticulum is very compatible with most of these Min family proteins. And it actually works very conveniently for us because it causes the entire cell to light up in this way.
And what is the rationale or what is the specialty of this main protein to select? Because there are a lot of proteins which have same kind of ATP, ADP kind of mechanism. So what is the unique about this protein to select?
Oh, why did we choose the MINS rather than like the PAR proteins or? I think mostly because the MINS were probably maybe the most well characterized in vitro. So I feel like the in vitro reconstitution of two proteins on a membrane producing this gave us a lot of confidence that that would work. Whereas, and we've started to look into some of these, but some of the other bacterial ones use DNA as the scaffold for the supporting structure to oscillate on, and they'll have additional sort of factors that they require, like specific DNA sequences that they need to support the wave propagation in those systems. So we thought this was kind of like the most streamlined set of components. And I guess I didn't mention this, but the combined system is only like 30 kilodaltons, so like 22 kilodaltons for the D and 8 kilodaltons for the E. So it's very compact, easy to deliver into cells. So that's another component, very small.
So can we use this property to isolate single cells?
So the one thing that we can't do is make this measurement on the cell sorter. I actually feel like there's a lot of really interesting things that could be done if we were able to like hold a cell long enough to make this measurement and then sort it. I had a colleague, Phil Romero, that made droplet microfluidic devices. We had this plan actually to kind of like measure it several times as it went down the channel, but then he left the university, so gotta find a new guy that knows how to do microfluidics for that. But that would be extremely cool if we could sort based on that property. Yes.
Great talk. Love the waves, especially the tiger waves. There are a lot of natural waves in biology. Have you tried to modulate them and sort of they try to cancel out and?
Yeah. Okay. Really cool idea. So I'm glad you appreciate that because I've also had like colleagues that are like, "There's no what's a wave?" Like, it's what happens when you're in a very biochemistry, RNA-oriented department. I actually have a colleague, Bill Bement, who studies natural rho-actin waves in Xenopus. So a Xenopus, when it starts as like a big cell, before it gets going, it creates these cortical actin waves on its surface. They look actually almost identical to the ones I'm showing you here, but they're made up of propagating gradients of rho and actin. And we've actually kind of been working with him to disrupt basically the endogenous rho actin wave biology with the waves that we create. And these are things that are actually quite easy to do because the proteins are so amenable to engineering that we can put things on them like a actin nanobody or like to interact with them and manipulate those structures. So it's ongoing, but it's like definitely something we're thinking of. Nikolai. Oh, there's someone in the back.
Quick question. Thank you for the talk. Really cool that you can measure protein-protein interactions in real time directly. Is there a way to couple this system with protein RNA interactions or with ribonucleoproteins like CRISPR systems?
Yeah. Okay. Fantastic thing. So that's something my student Casey actually has worked on. So we can use, I believe it's called Cas13b. There's a few of these Cas13 proteins that basically the guide RNA will base pair with an RNA. And she's shown actually the hardest part of that system is visualizing the RNA component to convince yourself that it's working. But yes, you can basically have Cas13b interact with an RNA. So like an idea we have, or that she had that I thought was neat was like maybe you can measure endogenous RNA levels inside the cell in some way. But the major challenge remains like having a way to actually confirm that make the RNA itself fluorescent. And there's all these techniques people use, like putting MS2 hairpins into the RNA. But a lot of them don't work very well, so it's kind of hard. But we have shown that if you transfect a fluorescent RNA in just to cheat to make sure that you can work, then you can definitely drive an interaction that way, which is pretty cool. And that creates some interesting opportunities also for localizing RNA in the cell as well.
Biological processes happen in time.
Yeah.
So I'm deeply appreciative when people invent technologies to take time into account and they think we don't do enough. That generally biomedical research. It's really fantastic work. I have a couple of different questions.
Yeah.
The first one is if you can give us a rough sense of the order of magnitude abundance of these proteins in terms of copy numbers per
Sure.
cell that you're expressing, and what fraction of the ATP flux is being consumed by that system? Is that a significant fraction of the cell's energy or totally negligible?
Yeah, great questions. And some quite important for when you're thinking about making measurements and whether your tool is perturbing the system. So we've done a little bit of trying to think what the name of that instrument is. It's called a seahorse. It measures metabolic activity in cells. As far as we can tell, the ATP budget associated even with very high expression levels of these components, so like those of you that like the leopard print, you need to express them quite highly to produce those kinds of phenotypes. There's like almost no detectable change in the energy budget to produce these waves. And I think actually this is because sometimes the hidden cost of life is mostly in terms of making nucleotides for DNA replication process or making your transcription. Towards the other point in terms of the copy number, again, it will depend a little bit on what we're trying to achieve with that. One thing that we quite like, and I have a lot of dudes in my lab that always want to like push the expression levels to the highest, but my student Casey has actually chosen a more different tactic of being like, how low can I go to make these measurements? And you do not require very high expression levels. So many of these, when we've done I guess maybe proteomics might be a nice way to do this. Expression levels are like well below housekeeping genes will produce nice waves. So it's actually not really about like you need a lot. It's like you need a ratio that's balanced, and then they will organize pretty well. So these can be well below gap DH levels and things like that. On the other hand, the higher the amplitude of the wave, the more straightforward it is to encode more data or to give yourself more dynamic range. So sometimes there's kind of a trade-off between like how low do you go versus like how much bandwidth you get for kind of encoding that data.
So my second question, I have more, but I'll allow myself only one last. My second question is, you articulated very clearly one potential benefit and application of increasing the plex within the spectral domain, being able to image many more cells. What other applications can this have? It seems such a cool phenomenon that they haven't thought much about, but we have considered for a much longer time. So share with us some additional capabilities.
Right. Totally. So I kind of presented most of this from the aspect of this measurement idea, like basically replacing better instrumentation with wetware sort of things to take existing fluorescent tools and make them oscillating. But like another direction, and this was actually kind of the original motivator for this, is to use the tunability of the system. So the fact that we can genetically encode a space, or rather a particular genetic setup produces a specific waveform, and we can generate a wide range of different dynamics and amplitudes. One thing that we've been experimenting with is connecting those different kinds of waveforms to endogenous proteins in the cell to see what types of dynamic or organizing activities have phenotypic consequences for cells. And actually some of this was motivated, I don't know if Galit is here, but she had I don't know if he was a student, but he was a postdoc in our lab, Jared, who developed this approach of using optogenetics to use light to reveal these kinds of like specific dynamic constraints on signaling pathways, where it's like to turn on RAS, it needs to be on for at least some amount of time. Otherwise, you don't get any signal out of it, right? And I remember being blown away, actually, as he's the one that taught me all some of these Fourier transform stuff, at sort of these hidden timescales or dynamics at play that are important for creating kind of switch-like behaviors in these signaling pathways. But for him, he had to do all of that light stuff, like basically like one experiment at a time, right? So it'd be like, I'm going to flash these cells for 10 seconds on, 10 seconds off, and then he would do like 20 on, 20 on, and try all these different patterns sort of manually using light. So one idea that we're quite actually interested in is, can we sort of take that concept and package it into a library to screen cells all at once for those kinds of dynamic phenotypes. So that's one space that we're looking at, and we have some funding for it, or maybe we did. I don't know. I haven't checked my email this morning. But and I think that'll be really interesting because there's all these like time scales or sort of like even steady states that are like maintained at the same level. The lifetime of that state is often invisible to us, and that lifetime is sometimes important for whether or not signals pass on. Third area that we've also been experimenting with is on a more engineering side, where we have a lot of collaborators where it's like, can we use these waves to localize things like in a T cell during, or an engineered T cell during killing or things like that, or to impose new spatiotemporal organization on cells for therapeutic applications. I think that's kind of like cool, although it's a little bit maybe like too futuristic, maybe at this point. But mostly we're kind of just letting the phenomena and what observables and measurements we make sort of help us see how something that's cool has utility, I think.
Thank you.
Great. Let's thank Professor Coyle one more time.
So quiet. We could have started.
All right. So the next session is going to be a panel discussion. We've started having the panel as part of the conference a few years ago, and it has always been a very lively discussion session. As you know, we value discussions throughout all of the presentations, but having the more dedicated time for a panel discussion has worked wonderfully in the past. So that's why we've made it a permanent part of our meeting. And we are very, very fortunate today to have Khalid Lahav joining us, who can provide a lot of perspective on biomedical applications, protein dynamics, imaging, and all of the other things related to her presentation yesterday. And of course, we have Jonathan Swidler, who has long been a leader in analytical chemistry and a master of many different measurement modalities. And myself joining to represent different areas of technologies and biologies to enjoy and conversation with all of you. So the goal is to enjoy the next half an hour or so as part of this discussion with questions coming from the audience and from moderators here. It is Sarah, but you're going to fulfill her role. Okay. So Sad is going to serve as our moderator to begin with.
Mic jockey.
See anything.
There you go. Well, here, can I start with a question to the audience?
Please.
So when I started in the field of single cell analysis, I was using capoelectrophoresis, and the center of capoelectrophoresis was at Northeastern by Barry Carter. And it was when we were doing sequencing, that was the biggest area. I was doing single cells in a little corner. But the reason I'm bringing that up is every year Barry Carter made capoelectrophoresis better with higher precision, higher accuracy, higher read lengths. And this resulted in the ABI sequencing. And even though their figures of merit were great, they got completely supplanted by other technologies developed not too far from here that were poorer, lousy accuracy, lousy precision, lousy read lengths, but they were fast, and cheap. So now I'm going to ask, in the area of single cell analysis, everybody talks about more proteins, more IDs, higher precision, and that's great. And I really love the 30-second, runs and things, but is there a need for poorer quality data, but hundreds of thousands of cells? When do we do the trade-off? Because I sort of look at this field a little bit like the ABI and other sequencing, and there's an interesting dynamic. Analytical chemists want better data, and yet sometimes cheaper and faster data is good. Go ahead.
Generate bad data.
I want terabytes of bad data.
So not to tout my field as opposed to the chemists, but that problem was solved by the bioinformatics people. That problem was only realized when the bioinformatics algorithms came through and said, this will work. If you had just dumped truckloads of crappy data on people with no algorithms, it wouldn't have done anything.
Well, actually, crappy data probably wouldn't work. It still had to be valid.
But the error values and the read lengths and all of the Burroughs-Wheeler's transforms that make all this possible, all that comes from really integrated multidisciplinary teams. And so please, as you develop new technologies, reach out to your local nerds and like fold us in, because we would love to be part of new technologies that have caveats.
Yep.
And we can profile whether that'll work or not work.
So go ahead. I wanted to make two very quick remarks regarding this very astute observation from Jonathan. I agree with it. One is that in the field of mass spec proteomics, the rise of very fast detectors such as Astro and TOF detectors that have outperformed Orbitraps is another example of fast data that are noisier, outperforming in many applications the high quality Orbitrap spectrum. So I think that's another example of the same trend that you're observing. And the other aspect to chime in is that when we speak of noise, there are very different kinds of noise. There's the random noise that we can deal with and algorithms can largely overcome. And there are systematic biases and artifacts that we don't know about and are hard to characterize. And this is a very different kind of noise. It is much less tractable and potentially much more dangerous. Galit?
Yeah, I would bring another point. I would bring another point, which is sensitivity. My worry is that if the technology is not accurate, sensitive enough, then you won't detect things that are lowly expressed, and then any fancy analysis won't be able to fix that. And in some cases, I'm obsessed with those lowly expressed proteins that matter a ton.
This is just related to the statement that you said. Do you think that at the moment we're improving technologies in mass spec, but something like nanopore sequencing of proteins could really take over, or like start doing things that are not currently feasible by mass spec? I mean, I know the technology is not there yet, but over time, what do you think is the promise of that?
I mean, so a nanopore sequencing would be a good example where you could imagine it doing sequencing, but not doing, even though there's a few, it won't do most of the post-translational modifications. And so if you actually were able to sequence proteins without PTM, sort of the way transcriptomics started, and even today, transcriptomics can do a few of the 200 modifications of RNA, but we think it's fairly complete. So then there would be a space for that. Will that answer all your questions? No. Sometimes you'll need to know the phosphoproteome or maybe glycosylation. And so I could imagine both. I will say, as an editor of analytical chemistry, one of the hard parts is people say, why don't you have generic standards for every type of data like you do for X-ray crystallography? And the problem is, if you come up with standards and say, I will not publish a protein sequence unless you meet them, nanopore sequencing will probably have a completely different set of requirements. And so I don't ever want to set up a set of standards that prevents a new technology from being published. And so if you're using a standard technology, yes, if you come up with something crazy, then please publish it with us and we'll deal with that later. And I don't know where it will fit, but I imagine that protein sequencing through nanopores will become an established technology. Whether it's 3 years or 10 years, we'll see.
So I have two comments regarding the feasibility of the various single molecule approaches. The first one is that biology is incredibly complex and difficult to study at any layer. The genome is large and complex. The transcriptome is large and complex. The proteome is many orders of magnitude larger and has far more interactions and complexity than the genome or the transcriptome. And this has real implications about the feasibility of single molecule analysis. And we feel so strongly about it that a couple of years ago, Mike McCoss and I and colleagues got together with some single molecule experts, single molecule proteomics experts, and wrote a perspective on that topic in Nature Methods. And we essentially think that the single molecule approaches are much more suitable for transcriptomes with their limited dynamic range, where they're order 10 to the 5 RNA molecules in a typical human cell. They're order 10 to the 9, 10 to the 10 protein molecules. And if you want to count them one by one, those 10,000-fold of difference really matter. If we can make Illumina 10,000-fold more scalable and cheaper, and then make it also work with proteins, that can work well. And I don't think it's likely prospect, to be fair. I think that we would still need to invent additional capabilities, take advantage of separations and other approaches. But can nanopores and other single molecule approaches contribute to proteomics? Absolutely. I personally collaborate with colleagues to make that happen. And so I'll give you one example of an area where I think there is great potential, and this is for analyzing intact proteins. I deeply admire my colleagues in top-down mass spectrometry for doing the amazing things that they're doing, and it's incredibly hard. Mass spectrometry at this point is very powerful method for analyzing peptides. Analyzing intact proteins, as much as we've made progress, remains far harder problem. And this is much more natural to solve by having a technology that is actually reading long molecules. So are nanopores going to sequence proteins anytime soon? I think it's highly unlikely. Can they provide useful constraints in combination with other technologies to provide some information for intact proteins? I think that's an exciting prospect. And going back to my very first point about the challenge of the proteome, I think that's a really, really exciting big frontier in science that has not been illuminated by technologies because it's been too hard. So any technology that can help us is my friend. I want to support it. We need them all. And with all of their capabilities, there will be a lot to study for the proteome for a long time to come. It's just immensely large and complex, much more so than what we normally appreciate when we think of this duality, the transcriptome, proteome, number of genes quantified at the protein and RNA level. We're not quantifying genes, they're gene products, and they're really very different in terms of complexity and scale.
I agree.
So following up on that thread, what needs to happen to make single cell proteomics faster and cheaper, as Professor Swidler mentioned?
Give him $100 million.
Thank you, Jonathan. Deeply appreciated. To make it 200.
Well, then I guess.
But I think that is happening. So we are seeing, we had this conversation at dinner last night with many colleagues who have followed the conferences, and there are two perspectives. On a day-to-day basis, it seems that things are not moving fast enough. We wanted to acquire this data set today, and the instrument's not performing well, and it's somewhat frustrating. But if I look at a different time scale of where we were 5 years ago, where we are now, it's remarkable progress that is accelerating. And even today, after this session, we are going to hear about approaches that increase throughput by a factor of 10 relative to what we could do last year. And that's substantial. And what is important in this particular case is that there are opportunities to have those gains in scalability and throughput without sacrificing the depth of coverage and the quantitative accuracy. And what we are pursuing at the moment seems to be far from hitting an asymptote. Those methods are promising to continue scaling for a while. And I'll give you more of an insider perspective from our team at PTI that has been developing those multiplexing technologies. As excited as we are for the new barcoding reagents that we just released on bioRxiv, we are even more excited about what's coming in the pipeline. This is a rich space of possibilities of inventing chemistry, inventing small molecules that can improve peptide fragmentation, that can support higher levels of multiplexing. So proteomics is the field to be right now. I think it's a wonderful area with lots of big, tough questions. It's an area for people who like difficulties. I'm not saying it's easy. I'm saying there is a lot of space to grow.
So I've seen a lot of applications of single cell proteomics so far, but so I'm surprised that so nobody is actually doing a single cell proteomics in the PROTACs research. So my question is that because though we are getting a good coverage at the proteome level, but the sequence coverage for individual proteins is still lacking. The reason is that, so I mean, in terms of PROTACs, so a lot of people are working on the kinases and ligases. Those express very less in the cell. But if we just want to look those ligases or kinases in the single cell level, and if we're hardly identifying some of these proteins with a single peptide sequence, how reliable is the data and compared to the bulk proteome? And what are the, like, I mean, I'm very new to the single cell field. I'm just curious, is there anyone in the field actually working in this direction to improve the like the single cell method to apply for this PROTACs research?
The short answer is absolutely. We are making a lot of progress on all of these fronts. We are increasing the fraction of sequence for which we detect peptides. We are counting millions of peptide copies from a single cell, which is far more than what you can count with RNA sequencing, just because there are many more proteins, really. But that actually has real consequences in terms of quantification, because if we are counting hundreds and thousands of copies per peptide rather than 0, 1, 2, 3, as it's oftentimes with sequencing, that means we can have much better quantification. And one of the sections that Andrew left out from his presentation yesterday that we are all very excited about is if you look at the reliability of computing and interpreting pairwise correlations between proteins among cells from the same cell type, we see this as being feasible and giving good quantitative data that is interpretable. We try to do the same for the RNA templates for those same proteins, and we see a lot of zeros and correlations that we don't even want to consider any further because the data are not as reliable as what we are able to obtain from single cell proteomes.
I have a question back to
Patrick was raising his hand. Maybe if we can bring a microphone to him.
so my apologies first. And my question goes back to throughput of single cell. I wonder, as we heard yesterday from Professor Kelly, that now we can potentially operate with 1-minute gradients. Is there a scenario where we could completely remove the LC and then with improvement in computational methods that we could basically just run every cell within, I don't want to say seconds, but pretty fast, and that's increasing throughput, may not be as deep coverage, but still providing useful information. I wonder what you think about that. Thank you.
Good question. I think the issue, though, is if you remove the separation, then the dynamic range, in essence, per cell will go down, and you might lose lower levels. Obviously, you'll see later on today, I do neuropeptides in individual cells where the individual peptide, not the others, are important, and I can do those at 1 second a cell with MALDI or faster, depending on how you want to focus the laser and how fast you want to go. And so you can do that, but the coverage goes down. That is a good example, just like a few other peptides. There are high concentration in individual cells, and so you don't need to do a separation to get pretty much a complete neuropeptidome of individual cells. And you can do tens to hundreds of thousands of cells that way. Those are gene products, I guess you could call them proteins that are cleaved. So there are cases that you can do that, in some cases it wouldn't work.
There's a gentleman in the audience sitting in the back by the name of Jesse Myers who has actually done this with bulk samples.
Yep.
without using liquid phase separation. And this has its advantages in terms of speed. It also has substantial trade-offs in terms of sensitivity, dynamic range coverage, and all of that. And there is another approach that one might consider to increasing throughput in addition to decreasing separation time, which is what if you separate multiple samples in the same time with overlapping active gradients? You're going to hear about this today. What if you barcode a large number of samples together as part of the same run and analyze them so that you can use very high performance separation? You can have a run that is taking 20, 30 minutes, but if you're analyzing hundreds of single cells at the same time, that actually gets you to a higher throughput than no separation at all.
Of course, the nanodesi approaches and others don't really, at some level, there's not a separation, and yet you get a lot of it. So it depends on the information you're after. You know, if you think of the Nature Biotechnology paper that the Kelleher group just did at single cells, that certainly did not have a separation and could, when you looked at enough cells, since the detection was stochastic, actually started to get a pretty good proteoform coverage of enough cells. And so it depends on how you frame the question and what information you're after.
Yeah, so this is Patrick from TGen. So we were a pretty new player to the single cell proteomics field, but we really realized that there's so much work that needs to be done at the computational side. Downstream analysis, we're dealing with ultra sparse data. A little shout out to Andrew and Luke and others. They all developed their own pipelines for doing data analysis, and I think this is wonderful. Seurat doesn't really work too well, right? So we're trying to do Liger, we're trying to do a number of different techniques, and every group is trying to build or rebuild or reinvent the wheel to a certain degree. Single cell sequencing has already built excellent tools. But again, I feel like as a field, we should try to come up with better methods to do everything from imputation to normalization. Sam is going to present some work here today about that, but I think we need to think about that. That's where a lot of the downstream analysis is just not figured out yet. And I hope that you can comment on that.
I can comment on that. And I agree that we can benefit from having more robust workflows. Part of what prevents this from happening is the organization of funding and incentives for doing it. I'm not saying anything new. In some ways that applies to other fields, except that in the case of single cell proteomics, resources have been more limited as compared to, let's say, single cell RNA sequencing. We certainly would like to see more standardization and improvement, and there are some initiatives within PTI that are aiming to contribute to that. I think what you're doing at the moment, Patrick, of raising awareness and making the case for it is helpful. It doesn't solve the problem, but I think pointing out that we need to invest in that direction and then thinking of ways of how we can do it and potentially having industry partners in the long term contribute in some ways in developing those workflows and then find a way to commercialize them and recoup that investment from for-profit companies that are using them. I think what really needs to happen for a sustainable development in that direction is to figure out what is the business model or what is the resource support framework that we can use to develop this in nonprofit environment. And I agree with you. It is something that needs more time and resources.
I'm curious for a second, just for listening to those questions. How many in the audience right now view themselves as focused on technology development, whether the experimental or computational? Like, raise a hand. Okay. And how many view themselves as technology users, like asking biological question with existing technology? Okay. Interesting. And some people didn't raise their hand at all. So who are you? Like, what do you do?
Those are the ones that need coffee.
The reason I'm asking that is because I think those conversations are really critical. I view myself more on the other side. We do not develop the technology, right? We use it. We say, this is what we want to ask biologically. We're missing a technology. And then we look for collaborations or industry or academia who can help us get there. And I feel like there's not enough these kind of conversations. Often I see technology being developed and then that company come and say, what can you use it for? What is it good for? Or we are saying, yeah, but we actually in order to use it, we need other things. So how do we get to talk more? I think this conference is obviously one of the solutions, but I'm just wondering, because even just listening to this discussion here, I learned a ton.
You've identified a very big bottleneck in, I think, the community of not having very efficient interface between technology developers and people who would love to use the technology for biological questions. And a lot of it is really intellectual barrier. It's not necessarily resources, it is know-how. What are the capabilities? What are the limitations of existing technology? What is really needed to be developed rather than the thing that a technologist would find themselves most exciting to develop next? And I certainly don't mean to say that this is not an appreciated bottleneck, but I think it remains a bottleneck. And the more we can overcome it, the better off we all are.
So we're having great discussions. I wanted to check in. It is 11:15. Should we continue for maybe 5 more minutes and just delay by 5 and move on, or should we?
I would like to turn just for a couple of minutes at the end to talk more about really promising biological applications. We talked quite a lot about technology, not as much about the biology. Exactly. And in fact, I would first like to ask Galit this question. Given the technologies that you have been hearing about yesterday and today, what do you think are some particularly important, promising, low-hanging fruit that are approachable with this technology? If you had the resources to implement them at any scale you want, what would you do with those technologies?
I don't know if they're hanged low because nothing feels like it's low, right? And everything is somewhat challenging. But the opportunity that I see that we have at this point in time is merging two worlds. I feel like there is the world of dynamical systems, which looking at protein dynamics at very high resolution in live cells, high temporal resolution, single, like the talk we heard also this morning. And then there is the omics, right? Transcriptomics, proteomics, high throughput, a lot. Bridging these two together in the same experiment is where I would love to see the field going. And I think we have the ability to do that. It's just these two worlds don't talk much. The challenge right now is the connecting these two platforms, following a single cell, and then at an endpoint, taking and doing the sequencing or the mass spec or at the single cell level. To me, like in our system and also in many other systems, I think it will open tons of possibilities. Now, let's connect it to biomedical. So I'm thinking about diagnostic. And I'll give you the most simple example. Senescence, there is currently no good marker for senescence. There probably you read papers that they are, but I'm telling you, there's none. Senescence is not a binary state, so you really cannot measure something fixed and say 100% is a senescent cells. You can do it if you look at markers dynamically. If you see, there are different dynamical behaviors of target genes like P21, P53 that looks different in senescent cells and in non-senescent cells. But when you do diagnostic, you're not going to put reporters in every patient cells or tissue, right? You need to connect the two and say, this dynamical regime behavior translates into this snapshot of fluorescence through proteins, and this we can do in the patient, right? So that's why, again, I think connecting the two is a huge need, and I think we're very close to getting there.
I love that example, and I can attest, having been part of the senescence consortium at NIH, that it's very difficult to define senescence in vivo, and the standards that people use are oftentimes inadequate. So if live cell imaging and dynamics can help with that, that's tremendous. Andrew has been raising his hand for a while.
Okay, so then
Last just really quickly, I was curious what you think, Galit, for those dynamics that you're measuring in P53. Do you think that these kind of temporal dynamics are, if we could measure these like proteome-wide in a number of different situations, do you think it's kind of a narrow subset of transcription factors and signaling molecules that these kind of oscillatory behaviors are important for the signaling state? Or do you think there might be thousands of proteins doing these kind of things that are playing into cellular biology?
Because these oscillations are so robust and so conserved, it's hard for me to imagine that they don't have any purpose, right? If we look downstream at the pathways responding, most of them do not oscillate, right? They more integrate the oscillation. So that might be something that is better than just a sustained response. If we do phosphoproteomics on the DNA damage and repair proteins, tons of oscillations. Okay. And there are some studies suggesting that they are actually important to physically bind to double-strand breaks in order to prevent the Oswald effect that droplets around the breaks actually grow and grow and grow. And so when you remove them, you get an opportunity for the repair protein to distribute evenly again with breaks that were not being repaired. I love that hypothesis. The theory is beautiful. We still need to prove it experimentally. So it's possible that P53 oscillations are like an epiphenomena of the other upstream regulators that are oscillating. So it's a very challenging question, but super interesting.
Wonderful. I think we can continue having this discussion for a long time and enjoying it, but it's also getting time for our next session. So unless we have a burning question or something else to say, I suggest we transition to the next presentation.
That working? Cool.
Okay, so while we're getting set up for the next speaker, I'm going to introduce him. Our next speaker is Kevin McDonnell. He has a PhD in computer science from the University of Galway in Ireland, and he is now working at PTI as a computational scientist, and he's going to tell you what he has been doing the last 2 years.
Cool. Thank you, Corinna. So yeah, I'm going to talk about some of the technologies we've been developing at Parallel Square Technology Institute, and just a brief overview of what that is. I was asked a lot yesterday, and you're going to see some of the people floating around. So we're a non-profit focused research organization. And one of the goals of PTI is to develop technologies that can increase throughput for proteomics. And so I'm going to talk about a couple of those today. And just off the bat, I want to acknowledge one of those is a project led by Harrison Specht in terms of new mass tags, and then also another project led by Jason Dirks in alternative domains of multiplexing. So to start off, we'll describe throughput. We want to increase it. Well, what is it? So we define it as the number of parallel peptides times the number of parallel samples in which we can identify those peptides. And if we want to maximize, say, the number of samples which we can analyze in parallel, typically we would do something like maybe a data-dependent acquisition approach. And in this approach, we take an MS1 survey scan. In that scan, we will send particular precursors for fragmentation, and we can use those fragmentation spectra to identify them. We can combine this with something like TMT, which can get 35 plex. So you can, if you identify a peptide, you can quantify it in 35 samples simultaneously. However, if we want to parallelize the number of peptides or maximize the number of parallel peptides we acquire, we might do something like data-independent acquisition. In this, we again take a survey scan, but instead of selecting particular peptides, we actually send these set windows. And so multiple peptides are sent for fragmentation. And so this is great because we can actually, for a single scan, we can identify multiple peptides. However, that creates more complex spectra. But current tools are well able to deal with this kind of complexity, but that can change as we actually multiplex even more. So as described by Dirks et al. In 2022, with 3-plex is kind of some of the state of the art. I know Proteome Sciences have some more tags out, and there are other technologies like diethylation, but we're working towards developing N-plex DA. Can we maintain the level of parallel peptides and yet scale to the number of parallel samples we can get with maybe a DIA, or excuse me, a DDA approach? And so just to talk a bit more about what is plex DIA, so this is where we multiplex in the mass domain. So for a particular peptide, we attach a molecule, so something like this ImTrack molecule. And so when we identify a peptide, we will have a channel that has the molecule attached, but in the next peptide, we'll have a different isotopes incorporated into the molecule, so we get this mass shift. And so we can identify 3 samples here together with just mass offsets between them. And what has been historically done is separating or inducing these 4 Dalton mass shifts, and this is to account for the isotopic envelope of the peptide, so to minimize the overlap. And so for this particular example, there are 2 limitations. One, it's currently only a 3-plex if you're using ImTrack. And so we need maybe bigger molecules that we can incorporate more isotopes, get greater plexing. But if we could reduce this 4 Dalton offset, then we could actually also increase the multiplexing for the same molecule size. And so that's where we're going to go. So in this talk, like I said, we're going to talk a bit about some new tags we've been developing, a project led by Harrison Specht. I'm going to talk about a new dimension. So I've just mentioned multiplexing the mass domain. What if we multiplex in the time domain? And I'm going to talk about JMOD, which is a piece of software we've been developing at PTI, which enables searching and analyzing the data that we acquire with these, because the way this data is generated is not currently searchable by the current algorithms, so we created our own. So to talk a bit about the story of developing new tags, so there are a couple of criteria we're looking for. So we set up the split pool synthesis approach where we can screen lots of different molecules, and with the goal of maintaining the number of parallel peptides. So we don't want to attach a molecule to our peptide and make it harder to identify. We want to maintain the quality of the spectra we produce, make it still easy to identify, or perhaps improve that. And we also want to allow the molecule to parallelize the number of samples, like I said. And so step 1 was screen all these different tags. So each point here is a different tag attached to both this peptide and this peptide. And these look at the scores, the Andromeda score in this case, for all of the different molecules when they're of the 2 peptides when the different molecules are attached. And PSM tag here is the molecule that we settled on, as it showed high scoring for both of these peptides. But to give, I guess, a more comprehensive evaluation, well, you can compare it here to label-free. So just to step through this graph really quickly, each point here is a different peptide, and it's on the x-axis, we're showing the ratio of precursor intensity. So how abundant was the peptide when it was labeled with the tag versus label-free. So if a point is here on the right-hand side of this graph, it means it was more abundant when the tag was attached, or it was observed a greater intensity. Then we have the same ratio, but with score. So on the y-axis, so if a point is above this line, it means when the tag was attached, it scored better than the label-free counterpart. And you can see that most points are scoring better, and most points are actually more intense. So this tag is exhibiting these positive spectral qualities when attached to a molecule compared to a label-free scenario. And just to explain maybe what that would mean. So here we have a label-free peptide. And so when we want to identify it, We send it for fragmentation. It generates this series of ions. So the C-terminus ions are the Y ions, and you can see for this particular case, we have the full series of Y ions were observed, and we also have some B ions were observed. And this results in a hyperscore of 34. And without going into detail of a hyperscore, it just encapsulates the intensity that was observed and the number of fragments. More intensity, more fragments, better score. Here's the same peptide, but with PSM tag attached. And the first thing to note here is we're getting a higher score. And we can look into why that is. We can see again the full series of Y ions is observed. But now we're observing more B ions. And so you can see how the Y ions are observed in the same positions, but the B ions are shifted as this is an N-terminal tag in this case. And so for this particular case, it might be super important because we already have the full series of Y ions, but this is particularly powerful for technologies such as de novo sequencing or maybe PTM localization, where you want as many fragments from as many of the fragmentation sites as possible. And you can read the preprint if you want to see more on that analysis. But just to explain this more completely, here we have B ions on top and Y ions at the bottom. Our blue bars indicate the frequency with which we observe these particular B ions in label-free spectra, and in orange with PSM tag attached. And you can see how PSM tag is helping the observation of these B ions systematically in our spectra. And this is the particular molecule itself. And so, like we said, this is great. We're exhibiting positive peptide properties. We're not going to sacrifice the number of peptides we see by attaching this molecule. We may, in fact, enhance the number through these positive properties it's exhibiting. But each star here indicates where we can introduce a different isotope. And this allows us to multiplex in the mass domain with this particular tag. If we maintain this limitation of 4 Dalton spacing, we can get to a 5-plex with this particular molecule. But if we were to relax that constraint and just require 2 Dalton spacing between the different channels, we can get to a 9-plex. But currently there aren't software available that can search this type of data. And just to talk about the effect of this. So we're talked about this in a DIA. We're parallelizing the number of peptides in a particular spectrum. We may have different peptides in our library. All of these are present. And so this is a cartoon, but it illustrates the case. Some peaks will be distinct, will be only attributable to one particular peptide. Some peaks will actually be a superposition of the contributions from multiple peptides if they share that fragment or very similar M/Z properties. And so just to look at how much this happens in a label-free case, if we look at, say, here for a 16 Thomson window size, which is not that big, you can see that most peptides have fewer than 70% of their peptides are distinct. And so even in the label-free case, we're seeing this overlap in terms of fragments. But we want to push towards greater and greater multiplex, and so we're putting more and more peptides into these spectra. And so this is only considering monoisotopic peaks, it's not considering isotopic overlap. And you can see how we're driving the number of distinct fragments per peptide down towards 0. So this is including the 5-plex and the 9-plex. And so as we move towards greater and greater multiplexing, we need to think about how can we handle the type of data where this overlap is increasing further. And so you can think of different ways. Some current technologies throw out overlapping signal. I think it was talked about yesterday. There are others that assign it to a single precursor through competition. And I guess both of these methods, as we drive more and more towards this greater multiplexing, you're maybe getting rid of some of the signal you want to use. And so there's an alternative maybe where we can model this overlapping signal. So just, okay, make the assumption it comes from multiple sources. Can we try and deconvolve those sources into the individual contributions? And so this is the approach we took. And so in doing so, we developed a piece of software called JMod. It is open source, it's available on GitHub, and there's a preprint as of yesterday online. So I encourage you all to check that out, as there is for all the different technologies I'm talking about today. And with the goal of supporting multiplexing, thereby enabling this increased throughput. And it's, as I said, built on this assumption that we can model this spectrum as a superposition of the different library precursors. This is not a new approach. This was done by Spectre. It's done by Chimeras. But it's something that needs to serve as the baseline for a model as we move towards greater multiplexing. So that was a cartoon example. Here's a real example of JMOD. So these are just monoisotopic peaks, so they're just filtered for clarity, and they contain two very similar peptides. And you can see again in a real example where distinct peaks can be attributed to the individual peptides, but it can account for this overlapping signal and attribute that signal to the different peptides it came from. But what current algorithms can't do is model this overlap induced by plex DAA. If we were to when we start having overlapping fragments that are induced by these shorter offsets in the mass domain, we don't currently have technologies that can deal with that. And so I mentioned this 4-Dalton spacing, and it's like, why do we care about this? So just to briefly labor on the point, the reason we have these is because we don't want, or historically we wanted to minimize the isotopic overlap between the channels. And just to give a brief explanation, it's very clear probably to most people, carbon-13 occurs naturally. And so for a typical maybe peptide, you get a distribution like this. So the monoisotopic peak has no carbon-13. These are where we have one carbon-13 in the molecule, two, et cetera, et cetera. This follows a binomial distribution dependent on the number of carbons. And here you can see there's 4 distinct peaks, hence we have 4 Dalton offsets. For larger peptides, which are maybe observed less frequently, but you can see how 4 Daltons is not actually enough for these kind of peptides. But like I mentioned before, PSM tag allows this 5-plex if we maintain this 4 Dalton spacing. But what if we could halve the number of spacing we require, so down to 2 Dalton, we could effectively double the throughput with this same tag. And so that's what we did. We evaluated JMOD with its ability to model this overlap induced by these overlapping isotopes. And we also just compared it to label-free and to a 1-plex to give you an indication of how this the performance is doing. So to visualize this, here is a real observed MS1 signal from this very nice long peptide. And you can see the different isotopes are distinct here, and they're stacked like a train back to back. And so when we use JMOD, we can deconvolve this signal into the individual contributions. And you can see how here, because it's a long peptide, we have signal coming from one channel into the next. But this can be, with the joint modeling approach, or this, we can deconvolve this signal into the individual contributions. If we relax the constraint to 2 Dalton offsets, now we get this very non-distinct isotopic overlap. It looks something like you might see a protein isotopic overlap. But applying this joint modeling approach, you can see it's made up of all the different channels. And this is real data fitted MS1 coefficients that come from JMOD for this peptide. And so we can look at the identification. So this is 200 picograms per channel, so approximating single cells, labeled with PSM tag or label-free and acquired on Orbitrap Astral. And just to step through this graph, you can see the number of proteins here identified in label-free context with just a single PSM tag channel. And then as we increase the multiplex, we can see we're scaling the number of precursors. Our protein data points we are acquiring in a single run. And so maintaining this depth, or almost maintaining this depth of coverage as we increase the plex size. And we can compare this to Diane. So Diane is the leading search algorithm in the plex DIA space. And you can see we're performing comparably well for the label-free delta 0 on the 5-plex. But currently, JMOD is the only algorithm that can deal with these overlapping isotopic envelopes. And so it uniquely enables this 9-plex setup. And as Alexei said yesterday, IDs are not everything. It's not about getting the most identifications. We want to care about the quality of the data, how good is quantitation. And so we set up this mixed species experiment, as other people have talked about, where we have constant inputs in different mixtures of human, and then we have variable amounts of yeast spiked in. And we evaluated this in the 5-plex and the 9-plex conditions. And we just label different mixtures for every different configuration of PSM tag, combine them, and then ideally the algorithm is able to give us back the quantities, particularly of yeast, the different relative abundances for the different mixtures. And so to visualize this first, I showed you previously this human peptide. So all of the input amounts are the same intensity. But if we have a yeast precursor, you can see how this changes. And so you can see here now there's some significant contributions coming from previous channels into lowly abundant channels if they're followed by lowly abundant channels. And so to start, we'll just evaluate in this 5-plex example. This is still separated by 4 Daltons. You can see JMOD has given us very accurate quantitation here. So this is a 4-fold change delta 0. It's 4 times higher than delta 12. And we're able to retrieve this 4-fold change back pretty well. And then to maybe give a more systematic evaluation of the quantitation accuracy, we're going to collapse all the different comparisons into their mixture ratios. And so look here, A over B instead of channel over channel. And we can look at all the different all the different mixture ratios. And so we can see we can get accurate quantitation for 2-fold, 3-fold, 4-fold change. And this is as high as we've evaluated currently with this particular setup. Again, we can compare to Diane, which is the state-of-the-art in Plex DIA. And you can see here we're actually providing more accurate quantitation for these mixture ratios labeled with PSM tags. So this is 5-plex data, so it can be searched by current methods. So next, obviously, we want to evaluate the quantitative accuracy of 9-plex data. Again, I showed you this human precursor previously. And it's somewhat easy or easier because we have a significant signal in each of the channels. But what happens here is it gets a lot more complex when we have these 4-fold or large fold changes between the different channels. And you can see here back to delta 12, again, if you look at the monoisotopic peak, it contributes less than half the signal to the monoisotopic peak that it's attributed to here. And so the question is, can we accurately quantify peptides with this level of overlap? And so these are the results from the 9-plex, 2 Dalton offset spaced for the different mixture ratios. And you can see we're still getting accurate quantitation for this data despite the overlap induced. And we can compare this to the 5-plex methods for the two, for both JMOD and Diane. And you can see it's pretty comparable, pretty on par with the other 2 methods. So we're not really sacrificing quantitative accuracy. We're still maintaining those accurate predictions of the relative abundances of these yeast peptides in the 9-plex configuration. And we can focus in a bit more on, say, these 4-folds. So in this case, the largest fold change considered. If we look at those in the 5-plex and the 9-plex configurations, you can see for both here, we're getting, this is the delta 0 and delta 12, delta 0 and delta 12. We're getting a really accurate quantitation for both. It is to note, we are a slightly wider spread for the 9-plex data, but this is still very accurate quantitation with a tight distribution around the expected amount. And this is despite, as I said, this is quantitation coming from this delta 12 channel here, which for the monoisotopic peak is very little signal actually being attributed versus the delta 0 in this case. And so we're still able to maintain that quantitative accuracy. And now I'm going to talk a bit about alternative multiplexing domains. So I've described a lot of multiplexing in the mass domain and how PSN tag enables us to increase multiplexing in the mass domain. But a project led by Jason Dirks at PTI was exploring alternative multiplexing dimensions. And so analogous to multiplexing in the mass domain, we also acquire data in the time domain. And so we can multiplex in the time domain by inducing offsets in retention time to our different samples. And so how that works is, as our peptides typically elute off the column, we have a very high expectation of when they will elute, and the order particularly in when they will elute. And this can be improved with transfer learning, machine learning. And so we can get very accurate predictions of when our peptides will elute off the column. And so taking advantage of this, Jason stacked a second column spraying at the mass spectrometer, but with an induced retention time offset. So this one now is the same mixture or same peptides, but they elute consistently 4 minutes later with the exact same pattern. And this can be expanded to M columns. And so you're effectively multiplexing in the time domain by however many columns you spray at your mass spectrometer. And what is really, I think, the particular strength of this approach is that while it's similar to Plex DAA, it's completely orthogonal. So what you can do is you can combine the two approaches and you can get multiplicative increases in your throughput. So here you have a 3-plex, say, in your mass domain, and a 3-plex in your time domain gives you a 9-plex overall. And so you can get these multiplicative increases. And of course, it begs the question, well, I've just talked about 9-plex. I've talked about a time in the mass domain. I've talked about time-plex. Can we combine those approaches? And that would effectively give us a 27-plex. And so this is data acquired 1 nanogram per channel on Explorers 480. And you can see how we have label-free, the number of protein data points, 9-plex DA, and then you can see this 27-fold increase in the throughput for a single run. Here is just showing some of the chromatograms acquired by time-plex. So you have the human traces where you have this consistent quantity because they're spiked in at the same amount, and you have the variable quantities of the yeast. And so just to conclude, we've been developing technologies. So time-plex, it gives us parallelize the sample so to this PSM tag, but by combining these approaches, we can effectively, get this multiplicative scaling of throughput, which is very powerful. And so to finish, just to acknowledge all the people that made this happen: Smith Futures, Convergent Research, Griffith Catalyst, partners in Creagen and the Pentalutia Group, and of course, everyone at PTI, and particularly Harrison Spector, as I mentioned, and Jason Dirks, who have led those 2 projects that I talked about, and the whole team, it's been a complete team effort. So thank you all. And just to acknowledge, so as of yesterday, as I said, the 3 of these projects are now available on bioRxiv. They're available on GitHub. They're open source. So please check them out. These landing pages will give you all the information to the data, GitHub, and the preprints. And thank you very much for listening.
We have time for a few questions. Oh, okay.
Thank you, Kevin. Great talk and really impressive research. My question would be about if you've used MS1 and MS2 quantification, if you've also looked into MS2-based modeling, and if you have any expectations about how the more complex signal can actually help identification of these very lowly abundant peptides. Once we know that the signal doesn't come from 1 peptide copy, but actually from 27 peptide copies.
How that helps identification?
For example, yes. Kind of like stacking in TMT.
Yeah, I guess, and that's something we've observed. Now, there were other confounding factors, but arginine, typically the Y ions stack on top of each other. So for low inputs, identification becomes better for those kind of data points. So it's really powerful in that side of things. In terms of quantitation, we focus mainly on MS1 quantitation because we've been dealing with single cells. So we are currently working on improving our MS2 quantitation, but it's been mainly MS1 focused. But yeah, I guess that's one of the advantages of multiplexing is you can kind of almost, boost your signal. But then alternatively, you need to have strong channel FDR to account for that. So you're not giving false identifications either.
Thank you.
So you mentioned that this PSM tags will help to boost the B ions. So, but I mean, though you are quantifying the protein at the MS1 level, but considering even after identification, so if you see the spectrum knot or the dions, so they never take the whole series of ions to identify the proteins. It takes maybe top 6 or top 5. So what is the advantage having a more of B ions with these tags?
The advantage in the context of DIA, what I showed you is a really nice peptide that had a lot of Y ions. That is typically not the case, particularly as we get to maybe longer ones. And Alexi talked just about requiring at least 5 fragments. Well, maybe that's currently throwing out some signal, but if you can boost the number of fragments you're actually observable of your peptide, you are increasing the number of peptides you can now match confidently. What's really powerful about this kind of approach, though, is like I mentioned in terms of de novo sequencing, where again, that peptide we saw is maybe atypical in that it had the full series. De novo sequencing is very strongly reliant on having, the full series of ions, which is of fragmentation sites having a particular fragment present. And that is shown in the preprint that this tag is helping boost the full fragmentation site coverage in DDA peptides for de novo sequencing.
So this is timescale multi-column approach is also possible on the timsTOF?
On the timsTOF, yeah. So this is kind of, I guess, the next step. Some of the third-party software we're using to work with timsTOF data, it just didn't work out in time for this particular preprint, but it's not currently available for timsTOF data, but will be shortly. That's our main priority now, once this is.
How does the joint modeling take into account decoy peptides? Are those also modeled within the partial contributions?
Yeah, so the spectrum fitting fits both the targets and the decoys simultaneously. Yeah, I guess this is there's a Nathan who works with us has talked about, and I think it was done maybe in the Spectre paper too, how you could do an identification run where you just fit both targets and decoys together, and then for MS2 quantitation, remove the decoys and just fit those high-scoring targets to get better quantitation. But for the identification, yeah, we just fit them all simultaneously. And so, yeah, if decoys are attributed signal, they should be at the same rate as your false positives, ideally.
Just curious if the limit ahead is chemical or computational. How many maximum multiplex tags you can?
Computationally, I'm not sure if the limit is. Maybe in terms of how many ions you can get into the mass spectrometer might be more of an issue, but we haven't run into that. Another limit is we want to explore more is, I guess, the fold changes we can look at with this particular model. I've showed a 4-fold change. We can still accurately model 4-fold change. What happens if you get to a 100-fold change? Now the level of error maybe in your fitting, if you don't have the correct model of your peptide, does that now error proportional to actually the fold change you're observing? So these are things we still want to explore, but yeah.
Hi, great talk. Just quickly on the, like, a little bit more about how the joint modeling works. Are you fitting more of like a deterministic linear model on the isotopes that come out, or is there any like probabilistic kind of noise you might be assuming, or is it kind of, would that be kind of under constraint, or maybe you don't?
Not currently, no. It's, yeah, I guess, a deterministic in that sense. And I guess, like I was saying to Brianna, having the correct model is super important, because one of the things we noticed was if you don't take into account the actual isotope distribution of the tag, that's a pretty significant contribution. So you have the incorrect model, you're going to get incorrect quantitation. So it's super dependent on having, the correct predictions of, say, your fragments of your MS1 data. And so with machine learning and approaches like Georg's alpha DAA or alpha PEPT deep, you can get accurate predictions. And so that kind of, I guess, enables this kind of approach.
Yeah, and I wonder, like, if you were to take examples where, say, the fit didn't match your expectations, if you think there's anything to learn there from other things that might be happening, or essentially, I don't know, something that maybe you thought or is playing a factor that you didn't consider that you could learn from kind of cases where you have a lot of ions, but for some reason the model doesn't quite fit the spectra or something like that.
I guess we've looked into it somewhat. I can give you one example is if there's a peptide there or some species there that's not in your library, that's going to mess up your fit, because that's going to be signal that you're attributing to somewhere else. So it's super important having the correct library for these approaches. But yeah, maybe there are some other interesting things we can see for sure.
Very nice talk, maybe not related to the software specifically. It's nice that it's open source though, I like that. But how easy is it to implement PlexDia? I mean, if I would like to do it in my mass spec lab.
Oh, I'm purely computational. You have to refer your questions to the mass tags team. They're scattered around here. They have worked a lot, I guess, on optimizing and making it simple. I think that's one of the things we want to do, is to make this accessible to everyone. So I guess I can't give you more detailed answer than this is something we're strongly looking at and trying to optimize and release protocols so that it makes it easier to do.
Great, though.
Also, not a computational question, but if you run 2 or 3 or 5 columns, how significant is the hit on the sensitivity and the quantitation accuracy that you're observing in your data analysis? So you're using one mass spectrometer, you're using a trap, they're competing ions and so on. What do you see?
The effect of the heat, sorry, or
the effect of multiple columns.
Oh, okay,
You're
excuse
making
me.
artificially increasing the complexity of the sample.
Yeah, yeah.
So-
One of the things I guess Jason has done great work on is if we are, I guess, acquiring multiple samples from the same column over time, you have to do this normalization, I guess, for all the different samples. But what happens when you have 3 columns spraying together? You have spray issues, variable spray. You need to normalize, I guess, the intensity that's been sprayed at the column. So that is just fitting like a lowest fit to actually, you can see the intensity will kind of fluctuate somewhat across the run, but having a fit, you can actually counteract that and normalize that. And in terms of then, I guess, globally quantitative accuracy, it is a bit lower currently for this data, but I was also comparing Astral versus Explorers, so maybe there was some of that involved, but it's things we're ironing out because I think computationally we can also make some improvements there.
And it's a nice concept, but how practical is that? Can you run hundreds and thousands of samples on 3 columns and for days and days?
Yeah.
I just want to make a quick comment here. There are 2 different things. One is the concept of multiplexing in time, and the other is particular implementation. Jason has done 2 different implementations for that concept. We think none of those is optimal. I think each one of them is sufficient to demonstrate the potential and to make us very excited about it. But we certainly envision possibilities of having multiple separations on a single column using isocratic separation, other approaches that we've discussed with you and Jared and other colleagues. I think there's a lot of space for innovation to make this a really robust product. At the moment, I would not recommend everybody go run TimePlex. I think that's a good thing for experts to do and get their hands on and innovators in the technology to push it forward. But we foresee that the ultimate implementation of TimePlex is going to be different from the specific proof of principle demonstration that we've done. And we can also reduce flow rates to minimize the impact on sensitivity. And generally, our modeling approach is very much focused on dealing with the increased density of data points, modeling that so that we can minimize the impact of quantitative accuracy. And thanks to our friends who develop more sensitive, higher resolving instruments with higher capacity, we see a lot of space to keep pushing in that direction.
Nice concept. Thank you.
Okay, I think there were a few questions online. Can we see them here?
There were a lot of questions about commercial availability of the tags. So there were at least 3 or 4 asking that question. So I can chime in on this. We certainly would like to make these tags available to the community as soon as possible. And at the moment, you can immediately take our open source synthesis protocol, go to a CRO and synthesize it. We are also discussing with partners interested in licensing those tags and making them commercially available. And we would love to speed track this as much as possible. So that's the plan. Stay tuned.
Okay, yeah.
So thank you. I have a technical question. Because there's a difference due to mass defect between the isotopes of carbon and nitrogen, as we know from TMT. Is it possible with high enough resolution to split the MS1 peaks into doublets and then doubling the multiplexing this way?
This is something we're looking into, exactly that. So actually taking advantage of that mass defect. And so, yeah, having them, I guess, offset by millidaltons as well as overlapping. And so you can, theoretically, if you introduce, say, enough nitrogens into your molecule, you can do this.
Okay, any more? Yeah.
One of the biggest trade-offs that you have when you are labeling with TMT is a loss of dynamic range that is getting the larger, the more plexes you're using. So how big is this loss of dynamic range when you're doing plex DIA?
As I said, yeah, this is the next step to explore. So we looked at a fourfold change. We didn't see a lot, but I guess, do you mean dynamic range in terms of different molecules or in terms of the same peptide analyzed? Same peptide. Yeah, I guess this is, yeah, like I said, if we have 100-fold change, is the error if you don't have the perfect model of your data or there's some nice like noise in your data, that fold change now if they're overlapping is relative to the same size as maybe the error in your measurement. And so this is something we need to explore, but yeah, we don't have an answer for that, I guess, yet.
The one related thing that we can certainly share at this point is that there is no ratio compression as in the case of TMT. In the case of TMT, for any fold change, even if that is 50%, twofold, there is ratio compression, while in this case, we are quantifying ions that are specific to the sample and to the peptide and to the protein. So there is no systematic shift in the ratio. Quantitative accuracy is as good as for label-free analysis.
Any last questions before we close the session? No? Okay, then let's thank Kevin again.
Thank you.
All right, it's time to enjoy lunch and posters and to have some more conversations and discussions. introducing Jonathan?
Yeah.
Okay. I want to say a few words before we start the session.
Yeah, that's good.
You have the magic touch.
Don't say that.
Analytical chemistry running is way easier to understand.
All right. We are about to start our first afternoon session of the second day. And before we start it, I want to share with you a private conversation that I had over lunch with Ryan Kelly. I think he'll forgive me. He's a good friend, but I want to share it because I think it's very illustrative of this conference and why we're having it. So it starts from yesterday when Andrew Leduc talked about his data set of analyzing 4,000 single primary cells from a mammalian tissue. It became clear that this required 2 months of instrument time, which is a substantial resource and investment to do. But what we discussed with Ryan is that we can run some of our 9-plex labeled samples with PSM tags on the 5-minute gradients that he talked about. And that will give us a throughput of 2,592 single cells per day. So with this kind of approach, in 2 days, we can acquire data for 5,000 single cells. And we'll see how well that works. We can try it and we can find it on the short gradients. Our quantification accuracy is not as spectacular as what Kevin showed. But this illustrates 2 things that I wanted to share with you. The first thing is the power of our community in those meetings where we can combine methods developed by different groups and use them together to reinforce and do things better than what any one method can do on its own. And the other one is our general approach, at least my approach, that I like to push the limits. And sometimes we hit a hard wall, sometimes things break, and sometimes they just look very good. And I think at the moment, we have a lot of headspace, a lot of room to grow. We haven't yet hit that hard wall. We haven't yet seen a lot of things break. So I'm totally intending to send Ryan some of our 9-plex samples to be run on 5-minute gradients, and thoroughly enjoying this meeting, as I'm sure I'm going to enjoy the first talk of our afternoon session. So Malin, please.
Good afternoon, everybody. Our next speaker will be Jonathan Sweedler. And in giving an introduction, I had intended to give a very traditional, straightforward introduction, highlighting Jonathan's contributions to single-cell analysis, the important discoveries that he's made in neuropeptide signaling. But I actually instead want as part of my introduction, to talk about just the conversation I had when I went up and introduced myself to Jonathan. We talked about his work across a variety of different model systems: honeybees, algae, mice. And we talked about his diverse range of scientific interests and how the thread of single cell analysis, even at a time when people didn't think single cell analysis was feasible, was a thread that wove through all of that. And throughout the conversation, Jonathan's excitement, his enthusiasm, it was infectious. And those interactions are exactly the sort of thing this type of conference supports. So we're very fortunate to have people like Jonathan here who bring both a wealth of expertise in single cell analysis, but also the enthusiasm and excitement that we would like to have in our day-to-day, with the science that we do. So I'm very much looking forward to Jonathan's talk, and please give him a warm welcome.
Right. Thank you very much. It's a lot of fun to be here. I always feel a little bit of out of place because we do single cell measurements, chemical measurements. We do lipids, small molecules, neuropeptides, and we don't do as many proteins. But once in a while, we're allowed to slip into this conference. And so that's been a lot of fun. I learn a lot. One thing to point out, single cell chemical measurements with mass spec is not a new area. You can argue SIMS is older than this. But when over a couple decades ago, when I visited Franz Hillenkamp, he handed me a whole bunch of glass slides of some of his work from the mid-1970s before MALDI came along with LAMA. And this is interesting because you can actually see where laser beam 5-micron spot was hitting the cell nucleus or the cytoplasm, and the spectra are not very high quality. This is the days of taking a picture of a photo of a oscilloscope trace, and that's how you digitized it and things and so it was very different. But the idea of pushing mass spec to the single cell analysis has been around for many decades. The worst thing of all, I don't know why, is Franz Hillenkamp loved to publish this in weird microscopy journals, and so they're really hard to find these images in the literature because most of the journals are since out of business. Okay, so I thought I would start a little bit, just one, maybe two background slides. The approach that we did with single cell analysis started out actually in the mid-1990s, where we would take large cells like this, for example, remove a cell, and then we would do mass spec on it. And so what we would do is pull out the cell. If you could isolate the cell, get it on a target, grow it, we'd get a spectra of peptides, lipids, some proteins. And if you knew this cell, for example, had to do with swimming in this particular sea creature, then even a chemist can say, wow, we discovered a peptide. A biochemist would say formurfamide, phenylalanine, methionine, arginine, phenylalanine, amide, would drive this creature into swimming. And so knowing the cell and knowing what it did, you could actually figure out function. And I mean function in terms of the physiology, what it did. And so in the beginning, we did this for many animals, and we discovered literally hundreds and hundreds of neuropeptides, not peptides in the brain, but actually functional. You might recognize the graduate student who helped start this was Lingjun Li when she was in my group, and she's gone on to a wonderful career. So we did this across the metazoan. Some people say, oh, it works in a sea slug, it won't work in a mammal. I don't see how the mass spectra can know the difference. Right, so obviously this works in any animal, and we really showed that. I never understood the comments that said that it was easier to isolate the cells in some cases, but that's it. So this slide says improving spatial resolution. We started out with bigger cells because why not? That's a good way to start out. That were very important. I just wanted to put a few up here. I'm going to go through these and then talk about how we would go from single cells and subcellular domains. But the ultimate limit in, say, MALDI mass spectrometry is not the size of the probe, it's detection limit. And I point that out because if you take the probe from 100 microns to 1 micron, that's 2 orders of magnitude, but it's 10 to the 4th less area. If your detection limit is 100 zeptomoles, as soon as your probe gets small enough and you no longer have 100 zeptomoles of material there, you won't detect it. And that's one of the reasons why, in some ways, the minimum size, if your detection limit was 100 zeptomoles, for a GPCR would be very different than a lipid. I put this slide up because this is using a Shimadzu Chem printer, putting 60 femtomoles down to 3, attomoles down to 600 down to 30, down to zeptomoles. And even with an ultraflex stream, and I put this slide up, it's an old slide from the year 2006, we could, if we had a clean slide on a good day, get detection limits where there would be 300 zeptomoles of angiotensin anti-pain peptide put on the target. By the way, if you used radioactive angiotensin anti-pain peptide, when you finished this measurement, there was 300 zeptomoles left. Actually, it used 10%. It went from 330 to 300. And I know that because we would image it with a phospho imager before and after. And so our detection limits and went into the instrument was about 20 to 30 zeptomoles, but the amount I needed to do this experiment was 300. The reason this is important is that means even 20 years ago, the detection sensitivity of mass spectrometry for neuropeptides was well beyond what you needed to do a peptidome. What was the problem? Was sampling. Right now, I like new instruments. We have a timsTOF Fleck, MALDI-2, and I'll show when we need that. But most of the issues really were sampling, and that's what we spent a lot of time trying to solve. You could use a better probe. I don't know if it's a better probe, but if you want to do higher resolution, you can do that. I'm not going to go into it. We did complete ablation 20 years ago. And that, a lot of people use that for mass spec imaging also. But a probe, this is just some work again a while ago, and we're actually looking at a nerve terminal with about 400 nanometer resolution, and you can see vitamin E is in the terminal, and we can actually see that if you damage the nerve, that vitamin E accumulates. And that was some new biology as well as kind of a fun demonstration of SIMS. So you can get resolution, but you don't get much molecular coverage. So I wanted to talk about mostly the bottom one, but I just dug up one slide worth of information just to say I see a lot of people in mass spec imaging now doing tissue expansion. This was our first foray. This is something that we did actually before optical microscopy came up with the idea of tissue expansion. We talked about this in 2004 at an ASMS meeting, and the idea is you take parafilm M. By the way, it's the weirdest comment I've ever gotten from a company, is we published that we were using parafilm, and parafilm M complained to me greatly that there's no such thing as parafilm, and if you look at the side of the box, it says parafilm M. So if you ever want to use this approach, make sure to include the M, or you'll get a rude email from the company. I don't know who at Parafilm or Parafilm M actually reads this type of literature. But anyway, now you see I'm very good. We use Parafilm M. Okay, we spread it. We can pull it apart 5-fold in each dimension, 25-fold, and now you can actually hit individual beads and see individual peptide spectra. Just for the fun of it, we actually did this with colored beads, and we actually anchored trypsin, chymotrypsin on. The neat thing is they're separated by a hydrophobic parafilm, and so you can put them in a humid environment in Illinois in the summer, and there's enough water that you would digest the proteins, and then the red beads or the green beads would be different. We could actually image as we stretched, and so we could put together the image directly, and so this worked. And then lastly, you can even do single processes, and so it was a way of sort of breaking the resolution barrier when you didn't want to. As we were making this work, of course, mass spectrometers became better, and now we can get the 5-micron resolution we want, so we don't have to do that. So this is the approach we use, and I've shown this before, and I just keep it to one slide. You take a microscope slide, you put a bunch of X's on it to fiducial marks. This actually is about 10,000 individual cells. They're GFP labeled, and this is a mouse xenograft model, and so it has a human tumor in it. And these are some of the cells, slightly different conditions, and so we put our controls on it. You figure out the locations of all the cells. You basically target the laser to individual cells, and we do a random walk because unfortunately it doesn't matter what mass spec company you use, the stage movement is actually slower than the laser acquisition. So we spend more time moving the stage, and so we actually in advance figure out how to minimize the number of stage steps because that takes more time than the acquisition, which I find sort of ludicrous when you buy a 1.4 million or whatever it is $1 million timsTOF that you're spending more time waiting for a $100 stage to move, but we won't go there. It's a great instrument besides that. So when we started publishing this, everybody said, prove it works. And one of the problems is if you're going to an unknown cell and getting data, how do you prove you don't know what the cell has? How do you know what you did? So one of the things that I started to do, and I'll come back to this, but I like to show this, is I went to an islet in a rat because I knew in advance that alpha cells had glucagon, beta cells have insulin, gamma cells have pancreatic hormone. We spread them out, and lo and behold, we would get really nice quality spectra of the right profiles. And the reason I put this up is twofold, is the cell-to-cell heterogeneity is infinite in the sense that from an alpha to a beta to a gamma to a delta cell or a Gelron cell, there's just no overlap of peptides. And that's not strictly true. About 5% of the cells had overlap, and I am absolutely convinced that's the 5% we ripped apart wrong when we separated. It's not biology there. And so that also tells me that there is a little bit, when you rip cells apart, there's a little bit of carryover from one cell to the other. Mass spectrometry doesn't know that you got a little bit of alpha cell on your beta cell. One of the reasons for doing that is, now I'm going to jump to humans, is after we did that, American Diabetes Association actually started supporting this research. We started getting human islets. This is just 4 islets from a healthy human donor. And each individual dot is a cell, and islet 4 has a lot of somatostatin, islet 1 does not. But over here you can see different levels of insulin, glucagon. There's a lot of information supposedly about knowing about differences, and there's a lot of cases that are really important. For example, if you have type 1 diabetes, the beta cells are attacked, and what happens is you regenerate and you keep on regenerating beta cells until eventually you lose that capability. And then some of the alpha cells supposedly become beta cells, at least by transcriptomics. And looking at that transition is of great interest. And so even though I just told you alpha cells or beta cells are different, there is this ability to look at these differences. And it's actually, they're stark. One of the fun things or surprising things is when we started getting cells from various donors. Now, I can tell you if you're an islet transplant specialist and a healthy human donor dies, and you're going to transplant islets into people that have type 1 diabetes, as soon as you find out that a donor has diabetes, you're not going to isolate their islets because, of course, you can't use those for transplant. But on slow days, if you really, really beg the transplant surgeon, they will isolate and send us some type 2 diabetic islets. But they don't do it too often because they're not going to use them. And so we tend at Illinois to get the dregs. And this is from both the University of Pennsylvania Transplant Center, it's from Alberta and a few others in Indy that we get these. But when we get type 2 diabetic islets, we get differences that look like this. Now, glucagon and oxyntomodulin are from the same gene, same prohormone, proglucagon, but different enzymes make oxyntomodulin, which is supposedly made in the gut. But once in a while, if you have at least we see only in type 2 human diabetics, we see cells that look like this, where all of this particular cell now makes oxyntomodulin. And I like this example because people like to use statistics and talk about changes. This is a rare cell. It's about 1 in 10 to 1 in 20,000 cells, but the change is at least 1,000 to 1. It's completely making a different set of peptides from the same gene products. And so statistics are sort of easy. Can you find those cells? Don't need to have high signal-noise ratio. It's a dramatic change that has profound health implications, but it's rare. So doing 10 cells or 100 cells or 1,000 cells is not enough. I need high throughput to find things that are 1 in 1,000 to 1 in 10,000. But I don't necessarily need the highest quality data. This is the question I asked before, because the change is huge. And so these are sort of the realm that I like to work in. And yet, even when I say that, I guess they're low-hanging fruit. They're very important for health. Let me show you some data I haven't shown before. You may know this, but everybody's heard of Zepbound and GLP-1, and they work on the enteroendocrine cells, and enteroendocrine cells are labeled just like alpha, beta, and gamma, except when they came up with the name, they thought Greek letters were, it was too complicated. And so they went to things like, K, L, M, and N, and these have different names. Turns out, now we're using a mouse where the enteroendocrine cells are labeled. By the way, in the gut, if you were to image, they're very hard to find. They're 1 in 10,000 cells, but these are fluorescent. We can isolate them by hand. That's a lie. I can't. But a few people in my lab don't drink coffee. They have this to hand. They can isolate these cells. And I put this slide up because what I'm showing you is that when we isolated about 20 cells, this cell, for example, 4 cells had just GLP-1, but 9 cells had GLP-1 and 2. A few cells also added PYI. And so they're adding, this cell over here is a K, L, and N cell. I don't list all of them. And so they're combined. Once we got this to work, we actually went a little bit further. And FACS sorting of peptide cells are hard because these cells are alive when we're doing this. And the readily releasable pool of vesicles are all released. And so when you FACS source living cells, all the peptides are gone. You have to keep them alive for a few days so they regenerate the peptides. So we partially cross-link these. And then we discovered that these cells are more complicated than anybody thinks, in the sense that we find groups of cells that are just combinatorial mixtures of these canonical cell types in the enteroendocrine cells for mice that are obese that we've metabolically challenged. And so these cell types are descriptors that don't seem to be very accurate. Now, I don't know in this case. I do know in the islet case if this change is based on particular day, or they're fixed for life, or they're actually dynamic. And so lots of questions, huge changes, and you need to do large numbers of cells. So I'm going to switch away from peptides and go to the most boring part of the chemical complexity, and that's lipids. Now, the one reason I'm interested in lipids, I used to say to people that your brain is like a bar of butter, and they thought I was insulting them. So now I'll change that around. My brain is like a bar of butter. And you can take that in any way you want. But what I mean by that is my brain mostly consists of water and lipid, right? And then a few percent are proteins, and a few percent are transcripts or DNA. Remember, 95% of the molecules in the brain are either water or one of 10 to 20,000 distinct lipids. Yes, there's 20,000 chemical entities known as lipids in my brain. And it's well known that if you lose a few of those, you have lots of problems or you're dead. Worse than that, the cells in the brain can't make those lipids. So transcripts and proteins are a problem. Why? Because they make those lipids based on what you ate, the fatty acids you ate, based on your gut microbiome changing some of them around based on your liver. And so if you're missing something in your diet, what does the cells do? Do they up or down regulate a transcript or change proteins, or they say it's hopeless, you're not supplying me with the right things, I'll make do with another one? And so the link between lipid enzymes in the brain and transcripts and actually the lipid profiles is very poorly understood. And so there's lots of reasons we want to do this. The other thing, of course, is that lipids are the world's easiest class of molecules to do with MALDI, but we'll ignore that. So this is just spreading 10,000 cells. This is with Elizabeth Newman. She's now a professor, assistant professor at Davis. On a slide, we used an FTICR, and this is color coordinated by the most intense lipid. Now, we don't see 10,000 lipids. We see about 50 to about 300 in an individual cell. But the cell heterogeneity is high, so even the most intense 50 lipids are very different cell to cell. After we did this, Elizabeth was talking to somebody who was very interested in this and brain development, and she did a quick bit of math who said, I want to do 100,000 cells, and said, I can't afford to do 100,000 cells on the ICR because I'm doing something else. So we just used our ultraflex stream. This is 150,000 cells. It's a few years ago, and it's unfair to say that it only took a couple days of time. Actually, most of the time it was actually just loading the cells. It was a few hours, but that's not the fair part of, because our collaborators spent weeks getting these samples together. And I'm going to ignore, whenever we talk about throughput, we tend to ignore that part of the experiment. And then also it took weeks to do the analysis. And so I always laugh when somebody says, oh, this took 3 hours. No, it took a month or two of all that part of it. But the mass spec is very quick. It's a second per cell. And we see some clusters of cells using projections that have lots of lipids. Now, this is a human developing brain. And so the brain regions and the type of, and the developmental points are very different. I wasn't even very familiar with it. But the lipid heterogeneity is reproducible, robust, and very distinct in time points and cell types within the brain. The problem with this, though, is we have lipid profiles, and our collaborators did transcriptomics on other cells, but we actually didn't know what the cells are. So I went back and said, how do we know what the cells are? So I'm going to show you 3 approaches to get that information. How do you get the cell type? So we're going to use other approaches that are starting to get towards proteomics and others. The first one, again, this is just showing you pictures of isolated cells. And by the way, I will tell you, any good bioinformatics person will tell you 3 points determine a plane. Why do we use so many X's? It's because we all make mistakes. And so, to be honest, if you can figure out the center of your X, I should have put this, and we scribe these in, plus or minus 50 micron, and you have 30 of them, we can actually isolate and locate a spot on our slide plus or minus about 4 microns. And so, I say that because I remember a Bruker engineer very calmly telling me, you don't need to do that. 3 points is adequate. And it's true, except there's errors. And this way it minimizes errors across the whole slide. And so if you ever do this, don't put 3 points on do this. It really does help. But anyway, this is what it really looks like. There's garbage, there's cells. We do our lipids. And then what we do is we use, in this particular case, the commercial Ambergen mass tags. This has been used for mass spec imaging. We'd already been doing antibodies. Now, the first thing that's fun about MALDI is it's non-destructive. Now, obviously, it's not anything that goes into the mass spec is used up. But what we're doing here is sort of weird, is that you put your cells on the target, you put your acidic matrix, dihydroxybenzoic acid, alpha cyano, on it. You do MALDI, and then what you do is you take the slide and drop it in a fixative and cross-link the cells, dissolve the MALDI matrix, and then you do immunohistochemistry. And somehow you've left enough of the proteins that you can do that. And we've done this with infrared spectroscopy, FTIR, as well as Raman. And yes, you can really do this. And so we're doing immunohistochemistry after, but we're using a mass tag instead of fluorescence because you can do more cell types. And what happens is there's a mass reporter. You photocleave it, and then this is different for each cell type. And we didn't come up with this, but it works not only on tissues, it works obviously on single cells. And this is the type of results. This is some work. It says unpublished. It's being reviewed right now. And we use the traditional approach, GFAP for astrocytes, neurofilament for neurons, oligodendrocytes, or myelin basic protein. And by the way, we in 1,000 groups do this. It turns out it's not true. 5% of the oligodendrocytes actually are GFA positive. So they're not as clean as you would like, but we're still sort of doing that anyway. And so we know what the cell types are, we know what the lipids are, and we can assign things. And one of the things we did is we wanted to make this clean. And so if something was positive for GFAP and neurofilament or myelin basic protein or even our microglia, which I'm not showing you, if it had 2 positive stains, then we didn't think we had a clean isolation and we threw it out. We also filtered to make sure in every case that any cells that were within 10 microns or 15 microns of each other, we threw out. So you scatter 35,000 cells and it turns out 5,000 fail our distance criteria. We then had 3,000 or so that failed our one antibody only. And so it doesn't matter because I'm assuming those failure modes are sort of independent of each other. And we end up with 10,000 or more cells. This is what one set of data looks like. Now I can say the neurons and astrocytes have different lipid profiles. You can see those astrocytes versus oligodendrocytes, and you can see actually this is a plot of what some of the lipids are. And neurons, for example, have very much neuron-specific versus astrocyte and oligodendrocyte are a little more complicated. We actually, if you when this is published, you'll see that you can even see neurons that have formed synapses and have active synapses. Different proteins like synaptorevin and synaptophysin actually have different lipids than those that actually don't have active synapses. And so lots of information are available this way. Never to rest on one approach. We've heard about this already. This was published in Nature Biotechnology. This is through the Kelleher lab, but if you actually looked at this, all of the cells came from our group. And so we were on the paper too, and this is now using nanodesi. It's using a cellogram. Every time you take this droplet over a cell, you get proteoforms, and you should read the paper. It's a great study. The Kelleher group did an amazing job. And I also have to say, Pei postdoc is in the transition to becoming a professor in the fall. I guess he's still a postdoc now, but I got to give him credit. Soon it'll be Professor Pei Su at the UC Riverside. But anyway, we actually were able to extract cells that had GFAP or neurofilament and all the others and type the cells. What we're doing now is the Kelleher is throwing their trash back at us. No, they're sending the cells back to us to see if we can now try this differently, because most of the cell you can sort of see behind, and can we actually do MALDI on these cells after they've done this to get the rest of the, and so that will have to stay tuned to the next one. But again, one of the interesting advantages of these probe approaches that you don't get with electrospray is that they're not, it's a disadvantage, right? They don't use all of the material. That's bad. The good news is they don't use all of the material, so you can use follow-up measurements. And so we'll see how that works. Maybe next year I'll talk about how to do both proteoforms and small molecules. If you don't like proteoforms and you don't like antibodies and you still want to be at this conference, there's hope. This is the last approach that we can do this with. Now, you may not know the name Jim Eberwein. Jim Eberwein in 1991 published the first single-cell transcriptomics paper. And what's really funny is he tells me that I was very friendly to him on how to isolate Aplysia neurons in the postdoc at Stanford. He was in another lab. My wife had just had twins. I was sleep deprived. I do not remember meeting Jim. Luckily, he remembers me in a nice way. And so he said, hey, would you like to send us and see if we can do single cell transcriptomics? We've been hearing about transcriptomics and mass spec. In this case, you got to realize it's on the exact same cells. So we did MALDI first. And we did everything wrong. What I mean by that is we take the cells out, we put DHB on it, we do MALDI mass spectrometry on it. This is no RNA ACE. Everything was done wrong. Then we send the cells to Jim, and he says, this is stupid because it won't work. You did everything wrong. But transcriptomics is so amazing that when he gets rid of the DHB, he is actually able to get between 6,000 and 10,000 transcripts from every cell, not as the 20,000 he would have if we hadn't messed up. But we're able to do single-cell transcriptomics on the exact same cells. Now, it turns out this works. Now we're up to about 80% of the time, and 20% of the time we don't get anything. I'm absolutely convinced it's because the FedEx guy somehow, or whoever was in the FedEx, left it in the truck in the summer or something. Because 20% we get nothing, and 80% work really well. And this is just some of the single cells, the lipid profiles, and this is some of the data. It says unpublished. We're just about to deposit this. We have almost everything written, and so I don't mind talking about unpublished data when it's all done. And we actually then can do astrocytes, oligodendrocytes, and neurons, and a lot of other things. I just decided to show you the same data, and that I showed you in the others. And so what I'm trying to say is one advantage of MALDI that's not generally appreciated is the fact that if you like lipids and peptides and metabolites, and I haven't done too many of the metabolites today because I have limited time, you can do that, and then you can do proteoforms or transcripts or others. I don't know how many of these you can gang together. And so it is kind of fun to realize that non-destructive, not using it all up, are either a good or a bad thing, depending on what side of the aisle you're on. Now, I realized I only had about 6 hours to talk. And so I decided to do one last thing, and that's how small you can go. So this is some work a few years ago. These are individual 500 nanometer to 1 micron vesicles. You can sort of see him by optical microscopy. We well separated when he's scattered. This is from my favorite animal, a sea slug. If you can't see him optically, this is using EM. Right? So we're using electron microscopy to find the locations. And once we find the locations, now we know where to hit the laser. And there's plenty of material in these. This is the lipid range. This is the peptide range. And we actually were able to, they've done the Broad Institute, they sequenced the aplesia. We actually were able to find neuropeptides, or actually peptide hormones, in these vesicles that were listed as hypothetical proteins that weren't even, annotated, but we found them expressed, and you said, yeah, they're a prohormone, and we were actually able then to use k-means cluster and say the same cells had 3 different categories of vesicles that had different but overlapping but distinct peptide profiles. And so we can do things that are that big. Now let's really push it. This is some unpublished data to show how far a crazy group can go. This is single mammalian mitochondria, 200 to 400 nanometers. Now we're stuck. We only can do lipids because remember, the last thing you can see is lipids because they're on the surface, so they don't go down as a cube, but as the square. We hope to see cardiolipin and others. The reason this is important is there's a lot of lipid metabolic disorders in mitochondria that result in death or disease. And so there's even evidence that the mitochondria within a single cell in some of these cases are heterogeneous, and you need to see that. Doesn't matter if you have the right peptides, the right proteins, the right transcripts for mitochondria to fizz and fuse, which is required for health. If you have the wrong lipids, they just can't do that. And so this is just some preliminary work. Now we're using a more sensitive instrument. We needed it. We used MALDI-2, which gains us 3 to 5-fold sensitivity. We are really pushing detection limit, not great signals. This is now taking mitochondria. This is only a week old. From liver, heart, brain. In this case, it's just liver and brain, and we see different lipids, and they are very distinct organ to organ. And this is just showing you a little bit of the data that, liver to kidney, or for example, liver to brain, very different peptide profiles. Some of this is expected, some of this is not really known. And it's very distinct individual mitochondria. And so they're organ dependent and across tissues. And I wanted to show you one last little data. This is now a clustering. It turns out most of the mito, at least the way we isolated them in the kidney and the heart, are the same. But in the brain and in the liver, there are very distinct types of mitochondria based on their lipid content. And so this is, again, one of the reasons to push to individual mitochondria to try to see and understand this heterogeneity. Now, what I'd like to do is, and I don't know, we haven't yet done it, it would be nice to say the brain differences are caused because some of them are astrocyte and some are neuron and some are microglia or whatever they are. I have no idea what drives this different yet. This has all been taken in the last week or so, but it really does show you that you can push detection limits to the point where you're at a very small fraction of a cell. And this, I guess was a combination of low-lying fruit in the sense that there are specific diseases now we're looking at to try to understand if mitochondrial heterogeneity underlies some of these. And with that, I'm really done. And I just wanted to end by saying one of the fun things that we've noticed, even when you're looking at lipids and peptides, we find novel molecules in the most well-characterized system. And that works because we're able to find the rare cells. And with that, I would be very happy to answer any questions. And I do have to add, the most important slide is I didn't do any of this work. I get to take credit, but this is the group that did this. And I showed data over about 10 years. And so I kind of showed, this is the most recent picture, but some of the people who did the work. Thank you very much.
Questions?
Great talk. Just to clarify, you mentioned that lipids are organ-specific in those vesicles that you analyzed. And in mitochondria, did you observe the same thing, that lipids are organ-specific or in mitochondria derived from different organs and tissues?
So this data was showing you, volcano plots showing you differences between kidney and liver and others. And so we are seeing differences. You know, for example, the brain mitolipids, these are now mitochondria, are distinct from liver mitochondria. So we did this first. I was really going to concentrate just on the brain, and my student probably correctly said, if this is not going to work, let's try a little bit different. And so we looked at the mitochondria that we confirmed with EM and everything else, and the lipids are indeed different from different organs.
And that's not based on the contamination of lipids from these different organs. That's mitochondria-specific lipids.
When we deposit what we are looking at on the target and we do EM on it, it looks like we have, you know, a pretty clean preparation of mitochondria. We do see the cardiolipins, which are indicating mitochondria, but it would be very hard for me to know if during isolation a few lipids. So, I mean, I don't know if I have just mitochondrial lipids, right? I have mitochondria fractionation that is prepared is to the best of our ability.
Very cool. Thank you.
Yeah. I never like to answer a question. Did you have contamination? Probably. Yeah. 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 That's what I do. Is when somebody asks a hard question, I just hold this up. I won't answer that one. And if it's a good question, I'll hold up the, never mind. Using those to the best of our ability. Go ahead.
So I have a question based on the work, you know, using EM to specifically locate these mitochondria within cells. So it's really precise localization, really exciting there. I wonder if you can talk a little bit about the throughput of an approach like that, how your group thinks about its importance, how you think about increasing throughput, and whether you think maybe as a general strategy, this sort of much more precise localization at lower throughput is a better approach for discovery, or maybe it's dependent on the question you're going after. Just any sort of general thoughts on that you have.
Well, there's two things. The first time you do something, the throughput is atrocious because it took my student a year to get any data, right? You know, because things weren't working. And so in one sense, I exaggerate and say, what's the throughput, not counting any of the development time? I'm going to answer that in two ways. One of the reasons this was exciting is with Jim Eberwein, but also Zhao and others, is the idea of taking individual cells, dissolving them and capturing the mitochondria in bubbles using a microfluidic device with some colleagues at Stanford, and then delivering those onto the target so that we know all the mitochondria from cell 1 are here, cell 2 are here, and all of that is going to be done in a high throughput fashion that I don't have to worry about. And then, since they're going to be deposited where we want, we will then measure the mitochondria, and Jim Eberwein will do the transcriptomics. Will that work? It hasn't yet, but it will, I'm sure, eventually. And so there is some goals that will sort of alleviate some of this. Right now, while we were waiting for that, we just took, you know, fairly defined areas and did a mitochondrial isolation and dumped those on in different regions. And so that prep, in terms of the ultracentrifugation, was the time, you know, so it really depends on what you're trying to do. We've tried all of those. And I'm not yet sure, you know, if you could find the rare cell and look at the mitochondria. What I really want to do is there's some neurodevelopmental diseases where there's evidence that the mitochondria in processes are different than those in the cell soma. And if we can isolate those, you would actually gain some really interesting information. We haven't gotten that far. So that would be next up. I don't know if, you know, we'll see differences and we'll see. So there's a lot of directions this is going to go. The mitochondria sort of really resisted us for a while in terms of just they're sort of borderline in terms of ability to do.
Jonathan, can you talk a little bit about the methods and approaches which you use to minimize technical variability of ionization? You talked about sample preparation being a key part of that, but there is also the variability when you shoot a laser. Sometimes you ionize more molecules, sometimes less. What are good standards and practices there?
So one of the things, it's a great question. So when we do the mitochondria on every slide, we actually have mitochondria from all 4 organs on the same slide to do batch variations. And then we have other slides where we have 3 biological replicates. And then the 4th part of the slide contains a droplet that has the mixed mitochondria from all of them. And so every one of these slides has a bunch of controls on there. We have a bunch of standards that we use, and there's a lot of other protocols. And even with that, the shot-to-shot variability on mitochondria is higher than I would like. We're just too close to the detection limit. And I think what happens is that when you're dissolving, even using a sublimation and things, when you're dissolving matrix, trying to get the right ratio of lipid to matrix, if you have fused mitochondria that are a little bit bigger, probably it's going to take a different optimum than otherwise. And so there is I am not convinced. I think we're introducing bias against certain size mitochondria in our measurements. And one of the reasons we haven't published this yet is I'm still trying to understand what that bias is. I will tell you, which I find very scary, I tend to know a lot about neuropeptide cells in all these different models. And there are some cells that are much more fragile than others. And so when you isolate Purkinje cells or even some of the other big cells in a mammalian brain, you lose entire populations under the conditions that the others come out. And I don't care if you're doing proteomics, metabolomics, or anything else. If you lose a population of cells, then they're gone. And I can't tell you why they're that much more fragile. But that scares me because I sort of assume what's on my target or what we isolated is sort of representative of the tissue it came from. And so whether it's mitochondria or whether it's cell isolation, there are biases that are sort of surprisingly hard to overcome. And we're still trying to understand those. Great questions. I wish I had a universal answer. It really depends.
We have an online question. What is the ultimate resolution you could envision? Could you capture non-membrane bound organelles like stress granules or suborganelle cell constituents like the nucleolus?
So rather than say if they're individual organelles and you can spread them on a target, I don't care about spatial resolution, it's detection limit. And so the question is, can you detect what's in an organelle? And if so, then you just have to have a way of finding them. And so there are reasons for that. I'm desperately trying to do individual extracellular vesicles. And what's interesting about those is a lot of them are small, but there was just a publication in Cell about blebosomes that are 5 microns. So can we see those? Yes. Can we see things that are 100 nanometers? Maybe not. And so we're trying to do those. There, the question is, how do you visualize them? There are lipid dyes to make them visible. And then if there's enough material, we can target them. And we'll see what we can see. One of the things we do is we hit all the objects, and then we move our mask of 150 or 1,000 objects, and we move it over, and we shouldn't have any. Right now, we're not hitting things, and if we get blank, then we did a good job. If there's a universal signal, we contaminate or slide. And so we do a lot of tests. But I think the answer is some of those we can do and some we can't. I mean, the Kelleher group is very smart because it's important, but also single nuclei are great. They're big. So no problem. I mean, I'm not using that as an excuse. There's important questions about it. But single nuclei, yes. Single mitochondria, borderline, but yes. Others, we'll have to see. Great question.
Are there other questions? If not, let's give Jonathan a hand. And I'll also be introducing our next speaker, Julia Sheppy. Julia comes to us from Stanford University, where she's a student in the lab of William Greenleaf. And Julia is working on a very important research area in the context of gene expression. She and her colleagues in the Greenleaf lab have developed a really elegant approach to understand in very intricate molecular details how enhancers and activation domains and transcription factors coordinate together to coordinate gene expression. And this is a really important question. Many of you are probably aware that variation in enhancers, variation in activation domains are a very important link between genetic variation and differences in gene expression, and probably ultimately differences in things like health and disease. And so what I think is exciting about Julia's work is it's really moving us from this sort of binary models on-off to a really quantitative and mechanistic understanding of how these genomic features coordinate gene expression. So please give a warm welcome to Julia.
All right, can you hear me? Great. I'll be okay with that quicker. Thank you so much for such a kind and thoughtful introduction, and thank you for the invitation to come talk. As you could tell from the introduction, not in the single-cell proteomics space, so I've really enjoyed learning more about this field. And in this talk, I'll transition from the last talk of more cell heterogeneity to thinking about molecular heterogeneity, and specifically thinking about how transcription factors interact with DNA to regulate gene expression, and how hopefully you can take away from this some of the methods we use to be able to get really high resolution and quantitative measurements that allow us to actually use really simple physical models to understand what's going on. And the work I'll be talking about today is the first project led with Ben Dowdy and Michaela Hinks through a really fruitful collaboration with the Bintu Lab. And the second project I'll talk about is co-led with Emil Markland, who's a professor at Stockholm University. And so, as many of you know, transcription factors, these proteins, are very important in regulating gene expression. But there's many molecular details that are not yet quantitatively understood in how they interact with DNA and form open chromatin within cells. Historically, we've studied these with bulk genomics approaches like RNA-seq, ATAC-seq, or ChIP-seq. I don't think I need to convince you all why moving to single cell or single molecule is useful and informative. But specifically, we're interested in if we can see underneath one of these ATAC-seq peaks to understand the individual and heterogeneous states that make up one of these peaks. To illustrate this, I'll talk about a very something that's been seen in the field of genomics since the 90s, that as you linearly increase your motif copy numbers, so these little blue boxes denote transcription factor motifs on DNA, as you linearly increase the number of those, you get nonlinear increases in gene expression. And over the years, there's been many hypotheses for why we observe this, whether it comes from cooperativity in binding or cooperativity in activation once you're bound. But because we don't have these molecular measurements, it's been really hard to say which one it is. And so we think what you need is to actually measure the transcription factor occupancy that links your motif number to the ultimate gene expression. So how are we going to do this? We built a very simple synthetic system in which we have a minimal promoter driving expression of the fluorescent reporter citrine. And then upstream, we installed a library of enhancers that have variable motif numbers for the synthetic transcription factor RTED-RVB48, which is DOX-inducible and drives very strong gene expression. We then integrate this at AAVS1 and K562 cells, so they're within the genome. And we can confirm that as you increase this motif number, you do indeed get these nonlinear increases in gene expression as measured by flow cytometry. So in the system, how are we going to measure transcription factor occupancy? To do this, we turn to single molecule footprinting. So this was heavily inspired by the work of Arno Krebs and others. And so how it works is we take our cells, we then incubate them with the GPC methyltransferase, which adds these methylation groups to accessible sites along DNA, giving us a really high resolution and contiguous picture of accessibility. We can then convert this accessibility with an enzymatic version of bisulfite sequencing, and then we just read out these molecules on a standard Illumina sequencer. What's nice about this is that different molecular states will have different and interpretable molecular patterns, such as nucleosomes having larger protection widths or even seeing transcriptional machinery at the promoter. And so to illustrate this point, here are 4 actual molecules from our data set. Up is accessible and down is protected. You might be able to label what's going on by eye, but we built a probabilistic model based on prior assumptions to be able to infer these states. And so for every single molecule, you can count how many transcription factors are bound at the enhancer and whether the promoter is in an active or nucleosomal state. So basically, is the promoter bound by a nucleosome or is it open and accessible? We don't just have 4 molecules. We measure thousands for every single construct that we put in the system. And so here we have 8 motifs in this molecule, and we have each row here is an individual molecule that we've measured, and every column is a site that we get methylation information at, where white is accessible and black is protected. So these stripes of black underneath these motifs represent TF occupancy. And so we can see this now across the library with different motif numbers. And what you might appreciate is there's a really big jump between 4 and 6 motifs, so in that TF binding or those stripes that you see. The reason we're excited about this data is that now for the first time you can actually see what the distribution of TF occupancy is along molecules. So here, if we take molecules that all have 7 motifs, we see that some have all sites bound, some have none, and most have 3 to 4 transcription factors bound. And the reason we're really interested in this is because it gives us an opportunity to test these types of gene regulation models that have been long used by Locker-Bintu and others. And these partition function models are borrowed from statistical mechanics. And so we started with the very simple one that we could come up with, where there's an energy of interaction between the transcription factor and DNA, and an energy of interaction between the nucleosome and DNA. You then list out the microstates, so all of the possible ways your molecule could be, and then assign an energy to each one of those states. That energy then dictates the probability of observing it through the Boltzmann distribution. We then fit this across our data set, and you might be able to appreciate it does a really poor job. So clearly we're missing something. And we thought, okay, it's not just R-TED-R, this DNA binding domain. It also has a separate part to this protein, the activation domain, VP48, that can interact with other cofactors that are known to destabilize nucleosomes, like chromatin remodelers like BAF or acetyltransferases. So we included a third parameter such that when the transcription factor is bound, it's able to recruit cofactors that then decrease the energy of interaction between nucleosomes and DNA. So it's destabilizing nucleosomes when the TF is there. Now with this one additional parameter, we can really nicely fit this bimodal distribution, and not just for molecules with 7 motifs, but across our whole library. So that's great. We tried other models, they don't explain it as well, but we still want to experimentally validate that this makes sense. So the first thing we did was we chopped off the activation domain. And what you can appreciate is that this leads to a big reduction in transcription factor binding. So that tells us that activation domains play a big role in how TFs bind within chromatin, which is really the first time we've been able to see that. And we can now explain it with this really simple competition 2-parameter model. We next wanted to know who are the cofactors that are being recruited. So the first one we inhibited was the catalytic domain of P300, which is an acetyltransferase. And interestingly, we actually, is this one frozen? Oh. I think it's frozen up there. Let me just reshare. Oh. I think I got kicked out of the Zoom. Okay, now I'm back. Okay, beautiful. Pretend that didn't happen. So now you can see the data that when we inhibit this catalytic domain of P300, we actually see a slight increase in TF occupancy. And so P300 is not the cofactor that helps stabilize TF binding in chromatin. Now, when we inhibited the ATPase domain of BAF, which is a known chromatin remodeler, we do see this drastic reduction in TF occupancy, consistent with these activation domains recruiting cofactors that destabilize nucleosomes, and this really drives your TF binding within chromatin. So far, I've really talked about this side of the molecule, the TF binding, and now we can look at how that relates to the other side of the molecule, which are the promoters that we read out along the same molecule. And so we fit this nonlinearity, cooperativity, and TF occupancy quite well. And how does this occupancy then relate to the promoter state? Amazingly, and it didn't have to be this way, what we see is that each TF is basically contributing independently to promoter activity. So there's this really linear relationship in between TF occupancy and the promoter activity. This is a single molecule metric, but it actually has a linear relationship with gene expression for this system. That will not necessarily be true across different promoters, and as you all think a lot about, there is post-regulation that happens past transcription. But in this system, it is a linear relationship between those, and so TF occupancy directly relates to gene expression. And so we've been thinking about this as an additive activation model in which each TF is working independently to drive gene expression. The slope of this line we've been thinking about as potency. So per TF that is actually bound, how much transcription can it activate? And so you then would expect that within this transcription factor, if you tune its dosage, its potency should always stay the same. So the first thing we did was we tuned our transcription factor effective concentration by tuning DOX. So you can appreciate here that TF occupancy decreases. But when we look at its potency, or its ability to activate per TF that's bound, this stays the same. We were also curious if we could actually tune this potency by going back to this cofactor data. So if you remember, BAF inhibition tuned binding, but P300 did not. And so this is quantified on the right. When we look at potency, potency is decreased for both inhibitor conditions. So the ability to activate expression is dependent on both P300 and BAF. And so our models predict that since BAF inhibition hits both binding and potency, it should have a greater impact on gene expression. And indeed, that is what we measure by flow cytometry. So you might be like, this is totally a synthetic system, interesting, but how much of it actually applies to a human transcription factor? The system we turned to was type 1 interferon response, which is your cell's response to viral RNA infection. And so we installed these interferon-stimulated response elements upstream of the same reporter construct. And the way this system is thought to work is that pre-stimulation, some of these inactive subunits, IRF9, are actually bound, but you don't have active gene expression from those subunits. Post-interferon beta stimulation, you get phosphorylation of STAT1 and STAT2, you get the full active trimer that then translocates the nucleus and is able to stimulate gene expression. Interestingly, in our single molecule footprinting data, we can actually see these subunits, these narrow footprints, pre-stimulation, but we don't see active promoter states. And post-stimulation, we can actually see the widening of these footprints on individual molecules, consistent with being able to tell the difference between the inactive subunits and the fully active trimer. And then when we look at these wide footprints and compare them to the promoter activity, this again holds with the independent activation model, and we can see potency being tuned by these cofactor inhibitions, albeit to different degrees than with VP48, since different cofactors are recruited by different activation domains. So to summarize this section, we built this reporter system to be able to connect measurements of TF occupancy and promoter state. We found that TFs compete with nucleosomes independently, but they get a really big boost by activation domains and the cofactors they can recruit. Once bound, they then work pretty independently to activate gene expression. And this cooperativity that we see across motif number is driven completely in this system by the cooperativity in TF binding. And so this is all focused on this one aspect of motif grammar, motif number. There's a lot of other aspects of motif grammar that we're interested in. And the one that I want to switch gears to talk about is how does sequence context outside of motifs actually tune transcription factor occupancy? To do this, we felt we needed to study this in vitro first, where it's just DNA and protein, and then move to the more complicated environment of cells. And so what we turn to is a platform that we use a lot in the lab. It's called HitsFlip. And so the way this works is that we design a library with tens of thousands of DNA sequences. Here we varied the motif, and we also varied the sequence context outside of the motif. You can then sequence this on a MiSeq, and then we actually image the chip while we flow fluorescently labeled protein on it. In this case, we chose KLF1. This is a human transcription factor that plays a really important role in erythropoiesis, and specifically the switch from fetal to adult hemoglobin. And so what this chip actually looks like, each cluster here is a cluster of DNA that we know the identity of. And so we can watch it light up over time, and from this we can extract association and dissociation constants between transcription factors and DNA, and we can also perform equilibrium experiments to extract affinities or the energies of these interactions across sequences. From that, we can construct position weight matrix, and so that is what the KLF1 motif looks like from this data. The way zinc fingers are thought of is that each individual finger, which this one has 3, interacts with 3 bases of DNA, and they tend to be very GC-rich motifs. And so this motif is very consistent with what is previously known for KLF1. What we were really interested in is what happens in these sequences where we're varying sequence outside of the motif. So here, there actually was really substantial effects, and that was pretty surprising to see. The first thing you can appreciate is that these 2 immediately flanking bases to the left of the motif, these 2 Gs, are quite important. It took a long time to think through this and digging into some old literature, but it turns out that this is likely due to direct contacts from an unconventional binding mode of this first zinc finger, meaning that those 2 should actually probably be considered part of the core motif, and the core motif I'm defining as something that is directly contacted when specifically bound. However, if we take those 2 out, there's still all of these more distal effects that can contribute over 2 KT or 10-fold effects on affinity. And so that's pretty substantial, and it really has this preference for more AT-rich sequence in the flanks. And so there's a lot more to dig into there, and I'd be happy to take questions, but a big question is, does any of this matter in cells? To do this, we turn to base resolved deep learning models. So this is BPNet, and these are trained on chromatin immunoprecipitation data. And so from KLF1 data, you can train this to take in any DNA sequence and then predict what your chip profile is going to look like from that. And the reason these are so powerful is you can then do in silico computational experiments where you fix one base, whether that's a base in the flank or in the motif, and run it through thousands of different genomic contexts to see what the average effect is. From that, you can derive position weight matrices that look very comparable. Here, this is for the motif. You can see that it's extremely similar to what we observe in vitro, where it's just TF and DNA, but it's trained only on this KLF1 chip data. We can also do this for the flanks. And I was pretty astounded to see this because these are pretty subtle effects, but it definitely pulls out these proximal flanking Gs and the more AT-rich sequence in the flanks. And so our findings in vitro do seem to be relevant to how these TFs are binding within cells, but it's still a qualitative measurement, and we were curious, how much can these flanking sequences actually tune the fraction of molecules that are bound in cells? And to do that, we then actually went back to SMF, and so we built a library where we have 4 high-affinity motifs. It has different flanks across the library, but along one molecule, each copy has the same motif and flanks. That's just so that we can get adequate signal within chromatin. We then install it at ADS1 and K562s. These are an erythroleukemia line, and so they have very high expression of KLF1. We then predict the energies of each of these sequences using our in vitro derived PWM model and feed it into these partition function models that I showed earlier in the talk. And so KLF1 also recruits BAF, and we can fit these parameters separately on a similar motif copy number style library. And so here we're not training any new parameters to make these predictions. And what we predict is that across the library of different flanking sequences, we should be able to tune occupancy in cells from being completely unbound and nucleosome occupied to completely transcription factor bound. And indeed, that exactly matches what we see in SMF data. And not just by changing those proximal bases, but also we see this effect that as you have more GC content in the distal flanks, you get less occupancy. So in summary, for that second bit, we saw really big effects from sequence context outside of the motif. These in vitro derived preferences are really similar to what actually matters in cells. And we can deploy these thermodynamic models to be able to predict molecular states across not just motif copy number, but other aspects of motif grammars. And so thank you for your attention. And the most important slide is just acknowledging all of the people that contribute to this work, since these are really collaborative projects. So thank you, and I'm happy to take any questions.
Are those like repeated sequence motifs that you're kind of studying where there's kind of nice spacing in there, all motifs for are these synthetic circuits, or are they in regular circuits, or how like frequent is that, and do the motifs ever like change for one transcription factor than another, or like I guess, and how does, if so, how does like having multiple different transcription factors, how do you think about cooperativity in that sense?
Absolutely. That's a really good series of questions. So these are all synthetically designed sequences that we've installed. One caveat that was pretty subtle is that we use a GPC methyltransferase, so it only methylates Cs within a GPC context. And so the reason we get such highly resolved measurements is because we insert them in these synthetic sequences. There's now other approaches that go to deamination where you're not necessarily constrained to a GPC context, but just Cs, and that works probably better genome-wide. Most genomic enhancers are comprised of somewhere between 4 to 6 motifs is kind of the average range. And so it is reasonable in that range, but it tends to be that they're more diverse. So it's not necessarily one transcription factor at a time. It did necessitate like these more in-depth synthetic approaches to understand one transcription really well and understand both how the protein and its activation domains contribute separately to binding and potency. And so if we could characterize that more broadly across transcription factors, you could totally take these same kind of thermodynamic style approaches to be able to model that, but you do need a lot more measurements. Yeah.
So maybe extrapolating a little bit beyond what you've been able to do experimentally, thinking about how far the sequence context matters. Do you have a sense of, is there sort of a continuous decay of that, or is it actually the case that you can extend really far beyond these things and that sequence context still matters for expression?
So you do see that it matters less as you get farther away from the core motif. Something I didn't mention about that data is there's a really interesting periodicity in it. So in vitro, you see a correlation, an autocorrelation in the signal of affinity with the GC content that has a periodicity of 10.5 base pairs. And that's not because you're creating motifs in the flanks. And so the effects from that can go out 30, 40, 50 base pairs in the in vitro data. It becomes more subtle as you get farther out. But the way we've been thinking about it mechanistically is that there's a few models of how flanks can tune TF binding. One is you're creating low affinity sites. We know in our data that we're not doing that. Another is that you're creating overlapping motifs with the core motif. So it's like high affinity and then some lower affinity ones overlapping. Our flanks also aren't driving that mechanism. There's also been studies that you are creating kind of repetitive elements where they're very, very low affinity, but so entropically favorable because there's so many of them. That also doesn't explain our data. And the periodicity in it is really pointing us towards a model where it's tuning TF search. And so part of how TFs explore DNA is through 1D diffusion. And so if that sequence context is tuning hopping parameters, that could explain why we see such distal effects up to 30, 40 base pairs away.
So one of the things that I love about the work that you presented, Julia, is how you're able to make these very simple fundamental models from rather sophisticated data. In some ways, that's a remarkable difference from all of the GPT type models where you take lots of data, come up with something that is fairly complex. Here you take lots of sophisticated measurements and you test those very fundamental, profound concepts with simple models. And I thought that's a wonderful thing for our community to see and think about how we can interpret our single cell protein measurements. And they do have a question, not just a comment. So my question is, when you think of the regulation of transcription and the type of models that you and your colleagues have made, do you think that quantification of the fraction of transcription factors bound to DNA versus not bound to the DNA in the nucleus can be helpful for constraining those models more and asking more questions?
That is an interesting question. So we do, clearly transcription factor concentration plays a really big role in the energetics of when you are bound or not bound. I do think it's an interesting question of how much time does a transcription factor spend unassociated to anything, weakly associated to cofactors in the cell, exploring DNA, or being specifically bound. And that super modulates the fraction of transcription factor that is free. And that is the parameter that matters for determining the thermodynamics of this. And so actually from the paired in vitro and in vivo measurements, where we're able to measure the in-cell effective concentration, which is that free fraction, and the in vitro energies, we can extrapolate how many molecules are free, which seems similar to the order of magnitude of which transcription factors are expressed. But it would be, I think, pretty interesting to know, actually, what percentage of your transcription factor is available for binding, because that is the information that would go into these energetic models.
Thank you.
Yeah, thank you so much.
This is sort of curiosity question. Do you see in your data cells or molecules that do funny stuff that don't really correlate with the linear relationship you saw? I wonder how that would come out in your data, but maybe you do some kind of filter for some noise or something like that, and maybe that correlates with a mutant or something. I'm just curious.
No, that's a great question. So we don't do any filtering. So that was all of the molecules that we sequenced that you saw in those plots. If you do a poor experiment, like if you don't lyse your cells completely and there's some cells that didn't get lysed, then you'll see that the methyltransferase didn't get in and those molecules are totally unmethylated. So occasionally if you didn't do an experiment well, you will see that. We don't throw those out even, we just tend to do a better experiment if that was the case. we have also done these experiments on plasmids, and then you start to see funnier things because plasmids are not nucleosomal the same way that you are when you're integrated within the genome. And so there things start to look a little bit funkier with a lot more spikes up and down and sparse nucleosomes. And so that is maybe more of like weird molecules that we see. But yeah, in general, from the data sets we have, it's pretty not filtered. Yeah.
Any additional questions for Julia? If not, let's give her a hand.
Thank you so much.
Hi, everybody. Our next speaker is Dr. Julia Laskin. She's the William F. and Patty J. Miller Professor of Analytical Chemistry at Purdue University. Previously, she was a chief scientist and fellow at the Pacific Northwest National Laboratory. And her research into bioanalytical chemistry, mass spectrometry imaging, and technology development covers wide-ranging applications, including environmental analysis, biological systems, and material science. So I think we're going to hear some really exciting advances from her today, and let's give her a warm welcome.
Well, thank you very much for this nice introduction and for the invitation to speak here. So I'm probably one of the few people here in the audience who does not do single cell analysis, not deliberately, right? So, but sorry, yeah. But hopefully what I'll tell you today, so this is still in the,
I know, I'm
yeah. So, but the techniques that I'll be telling you today about, we actually do look at single cells on a daily basis, and we look at them in the environment of a tissue. So the technique that I'll be talking about today is mass spectrometry imaging. No, it's still doesn't disappear. So I think we can just go with that. It wasn't like that previously, but that's okay. So, all right, so let me, then you won't see the wonderful faces of my students that will all appear right there in the corner, but I'll point to their contributions. So, okay, so I'll be talking about ambient mass spectrometry imaging techniques today, and I'll tell you some of the developments that bring them to the spatial resolution that is comparable to the size of the cell, or even better than the size of the cell, which allows you now to study cells within the tissue microenvironment and explore their molecular signatures. Okay, this one does not communicate now.
Yes, it's been causing issues. 1 second.
All right, so let me talk through some of that. So the idea is to be able to measure the spatial localization of different biomolecules in biological tissues, including metabolites, lipids, and proteins. And you've heard a little bit about those different aspects in the single cell context yesterday and today. So I'll show you what, when we kind of, abstract ourselves from the goal of doing single cell analysis, and we still we still do it in imaging experiments. I don't think so, but let's see if this works. Okay, this at least works. Okay, so let's see. All right, I still need this. Okay, so let's see. All right. Okay, so that, so here is the idea. We are combining spatially resolved lipidomics, metabolomics, and proteomics to get those comprehensive molecular maps of biological systems. And the advantage of mass spectrometry imaging is that you can do it in a label-free manner. So you have an untargeted, an opportunity for completely untargeted analysis, or you can make it targeted if you have targets. It's selective and and sensitive, and it provides you insights into the cellular communication within the environment of your, say, biological sample or biological tissue. So if you want to do these experiments with the single cell resolution or subcellular resolution, a lot of different technologies are getting there. Some of the most advanced ones are imaging mass cytometry or the beam kind of ion beam kind of analogs of this technique, where you are bombarding your samples with the laser or with the with ion beam, and you have antibodies labeled with lanthanides, and then you measure just lanthanides with your mass spectrometry, so pretty much aerosolize everything out from your sample. And you can do it these days with really, really high spatial resolution. It is a targeted technique, so you have to have your targets. You can image proteins with subcellular resolution, getting very, very beautiful images. From the standpoint of the techniques that I am more interested in, those are molecular imaging techniques where you don't have specific targets, you don't have any labels. And so currently, MALDI and MALDI-2, which is MALDI assisted by an additional laser to enhance ionization, they have reached the resolution down to about 1 micron in some cases. So this is still a very advanced experiment. It's not a routine measurement, but you can get there. And there are also some efforts that are focused on tissue expansion-based mass spectrometry imaging. So if you can't get a better spatial resolution with your system that you have, let's just stretch the tissue and get the spatial resolution using this approach. So the current challenges in this field and where the focus is still on improving sensitivity, throughput to enhance the spatial resolution. There is still a lot of complexity in biological systems. So, and when you don't have any separation, you see all the molecules at once. So we're still focusing on improving the selectivity of the techniques. Quantification is one of the big challenges, and most of the techniques that are available, commercially available these days, they require specific sample pre-treatment, matrix application or such, which may introduce some artifacts which particularly become important when it comes to imaging at a cellular or subcellular resolution. So the technique that we're developing is an ambient ionization technique that you've heard a little bit about. So we'll call it nanospray desorption electrospray ionization or nanodesi. And so it's basically summarized here in a very simple cartoon. So you have 2 glass capillaries or channels in your microfluidic probe, and you run your solvent through one of the channels and you pick it up into another channel and you spray, you generate electrospray here at the end of this capillary. So here is your syringe pump propelling liquid through those 2 capillaries. You make a little liquid bridge here between those 2 capillaries. You extract your analytes into this liquid bridge. You transfer them to a mass spectrometer inlet and analyze by electrospray ionization. And so now all you have to do is to move your sample under this probe. We'll call it nanodesi probe. So this will be our probe. We move the sample around. We acquire mass spectra from different locations. And then we take all the signals we see in our mass spectra, we generate those 2-dimensional maps, and we get those palettes of ion images. So this here in particular, this is mouse brain tissue section, a really old experiment, but just to show that we get hundreds of images in the same experiment. So each of those like little rectangles here is an image of an individual molecule. And what I will be showing you here is the intensity will go from dark to light. So low intensity will be dark and high intensity will be bright. So a very simple concept, by providing this path to the analytes from the sample to a mass spectrometer, we get a very sensitive detection, and that allowed us to reach the spatial resolution down to 10 microns. We've also published something like 5 to 6 microns as well. It's a little bit tricky to determine the spatial resolution in those experiments. We play a lot with the solvent composition, so we could do that for quantification and online derivatization. And we put a lot of effort into the identification using on-tissue tandem mass spectrometry. We currently have this technique coupled to 6 commercial instruments in our lab. We did publish a tutorial on how to modify a commercial instrument to be able to implement those platforms on commercial systems. And of course, there are platforms in the Kelleher lab and other labs worldwide that use this approach. All right, so when we do these experiments with high spatial resolution, we pull the capillaries. So here is my capillaries that I showed you previously, right? So the 2 capillaries that run the solvent, we pull them finely, and they will look like that in reality. So this is our 2 capillaries running the solvent, 1 and 2. And we add a 3rd capillary in between here going vertically. So that's a capillary that senses the distance to the surface. So this is a microprobe technique. So you need to control the distance between your probe and the sample in order for you not to run into some, valleys and hills on your sample and either break the probe or lose contact with the sample. So using this approach, and we also have microfluidic probes that do this type of sampling, which I'm not going to talk about today, but we can do those imaging, we can perform those imaging experiments either with this type of capillary probe or with a microfluidic probe, and we can get a spatial resolution down to 10 microns. And this is just to show you to give you some idea of how the data look like when you are imaging with high spatial resolution. So this is a small mouse uterine section. It's just a little over 1 millimeter across. It has distinct cell types here. So the dark kind of branch type of structure is one type of epithelial cell, so luminal epithelium. You have those kind of dots around them. Those are glandular epithelial cells, and you have stroma surrounding those. And when you look at images of phospholipids here, shown here, some fatty acids and phospholipids, and I give you the kind of key to read those annotations. So we look at phosphatidylcholine, for example, so PC, with 36 carbons in combined acyl tails and 4 double bonds. So this is our nomenclature. And you can see very different localizations of those species, right? I wanted to point your attention to those 3 sphingomyelins. We usually see very few sphingomyelins. But those 3, one is localized to luminal epithelial cells, one is enhanced in glandular epithelial cells, and one is present in both types of epithelial cells. So you can see it all the time. Lipids of the same class, just sometimes differing by the number of double bonds, show you very, very different localization. You can also see something that you would usually attribute to just insufficient spatial resolution of your experiments, but in this case it's real. So those fatty acids, for example, they're not very tightly localized to the epithelial cells. So they're spread out, right? And so we see those kind of shallow gradients in biological systems a lot, which makes it kind of difficult to define exactly the single cell location based just on mass spectrometry imaging data, because biology does not like steep gradients. There are usually no steep gradients in biological systems. All right, so you heard a little bit already from Jared about imaging of proteoforms, and I just wanted to show this early work that was done in collaboration with the Kelleher group, where we were able to take a tissue section, remove lipids from it, and then image it for proteins. We did that with kind of regular probes, not very high spatial resolution probes. We also showed that this works with high spatial resolution probes. What we're able to lean on is that the technique, this nanodesi at the end, ionization, is very similar to electrospray ionization. So you generate multiply charged ions, and then you can use your top-down proteomics approaches to identify them. So we do on-tissue MSMS, and we identify those all of those molecules in imaging experiments. So we have those capabilities, and I just wanted to show you some examples from this very early work that we did, the first demonstration that this experiment works. So this was mouse brain tissue, and we washed it, we analyzed proteins in this tissue, we also did top-down proteomics and identified multiple proteins. We got those different maps of different proteins in mouse brain tissue. Of course, those are quite abundant, so those are some of the most abundant proteins, but nevertheless, we were able to observe 40 different proteoforms, which you cannot do using optical approaches. And so 40 different proteoforms of 19 proteins in this rat brain tissue. And then we also extended this approach to higher spatial resolution, which is still quite challenging, but we can get down to 10 micron resolution in these experiments. So I just wanted to show you what you can learn about different proteoforms by mapping them spatially on tissues. So those are proteoforms of myelin basic protein in brain. So you see, I'm just showing you a subset. There was, I think, 8 or 9 of those that we saw in one experiment. So you can see the localization of those proteoforms in brain. Here are their annotations. And it didn't look very exciting when we saw those data, but then we plotted ratio images, fractional images of those proteoforms. So what we do is we take the signal of this proteoform and divide it by the sum of the signals of all of the proteoforms of myelin basic protein. And when we do that, we see some really interesting things here and some indication of some cellular kind of, of enhancement of those specific proteoforms in cells that are present in a specific environment. So here is an example of an acetylated proteoform that is very abundant here in the cerebellum region, and it's present also in high abundance in midbrain than some other species. Its analog that also has oxidized methionine is mostly predominantly abundant in the midbrain region, and it's actually repaired by the known sulfoxide present in the cerebellum region. So that's probably why you don't have this proteoform in high abundance in the cerebellum region. You have phosphorylated proteoform that doesn't show any specific preference to any specific cell types. And similarly, this acetylated and phosphorylated proteoforms shows a similar flat distribution in this fractional image. And we'll have some unassigned proteoforms. So I'm only showing you to say that, okay, there are some really interesting localizations in some species that we still don't know what they are. But we really hope that as databases are getting more advanced, we'll actually learn and we'll know what it is. But there are some species that we're discovering that are not in databases. All right, so with that, I told you that we can image lipids and metabolites. I showed you that we can image proteins. So it was natural to couple those two experiments and image both, do this multi-omics kind of spatially resolved analysis of tissues in the same experiment. And so the idea here is that you will section the tissue, and the example here I'll be using will be pancreatic tissues that show really beautiful immunofluorescence images if you stain for insulin and glucagon, and that's what we do. So we acquire an optical image of the section, we stain for insulin, and you see those beautiful pancreatic islets here in this section. We stain for glucagon, and we see this very typical for mouse pancreas, the localization of alpha cells that are surrounding beta cells in the islets. And we take a serial section and we image it for lipids and metabolites. Then we can wash the tissue and re-image it for proteins. And we also take the tissue section that was imaged for lipids, stain it for insulin. We can, in principle, take the stained section and re-image proteins, but does not work that well. But we've done that experiment as well. All right, so of course we were, and then we generate, in addition to immunofluorescence images, we generate ion images that we can then correlate to the cells that we mapped using immunofluorescence. So a big question was, do we actually perturb the system after we image the tissue section for lipids and metabolites? So what we did here, we take and stain the tissue after we imaged it for lipids, and we obtained this distribution of insulin in this section. And then we took a serial section that was fresh, that wasn't imaged, and we also stained for insulin. And you can see that there is a pretty good correspondence between those two images. We looked at the overlay. They're a little bit shifted, just as you would expect in serial sections, but it doesn't look like we are delocalizing anything, which means that we can actually do this type of correlative analysis. So then we imaged for lipids, we imaged for proteins, and we got those very detailed maps. So here, this is not a high resolution imaging data, but this is the only one where we imaged for both lipids and proteins. That's what I'm showing you, that I'll show you high resolution images later. So we stopped imaging for proteins just because it wasn't very interesting in this system, because you only see insulin and glucagon predominantly. There's nothing really particularly interesting. You do see also hemoglobin, but again, it's just marking the blood vessel. So it's not very exciting. But nevertheless, so this is what you see here. So this is your optical image of the section. You see 1, 2, 3, 4, 5 islets, pancreatic islets. You see them stained for insulin and glucagon. So you observe those islets in immunofluorescence imaging. And then you start looking at some lipids and metabolites. So we see some of the phosphatidylcholines that are often localized quite tightly to the islets. We see glutathione, so a metabolite. We see some other lipids. We see some things that we still have not identified, likely peptides that are localized to the blood vessel region. And we know that because they correlate with the localization of hemoglobin. So this is our insulin image. This is our glucagon image. And again, we have some other images that look interesting, show some different localizations, interesting localizations, but I'll tell you a little more about the identification. There are big challenges in identifying things in those experiments when you don't have a specific target. All right, so let me show you. So this is just a zoomed in optical image of a section that was imaged with high spatial resolution just for lipids and metabolites. And when you look at the mass spectra of those pancreatic islets, you see very abundant peaks of metabolites. You see lysolipids here, lysophospholipids. You see signals of lipids. So that's all pretty standard. There's nothing surprising here. We look at the images of those lipids, and we see them pretty well tightly localized to the islets. We know that from this immunofluorescence staining of this section. So here are the islets, and you can overlay those kind of outlines onto those ion images that we see. So we see some species that are just very tightly localized to the islets, and some species that show you the inverse localization. So they are in the exocrine tissue. So either in endocrine or exocrine tissues. All right, so that was nothing really surprising here. We see it all the time. There's nothing special about this. But what was really special in this experiment, that in negative ionization mode, we also, when we dwelled into the data, we observed a lot of peaks labeled in red here, that then we identified that those were deprotonated peptides. All right, so we looked at the localizations of those deprotonated peptides, and we found some really interesting ion images. So again, this is to guide the eye where the islets are. And you can see that some of those species, and here I'm only showing you the master charge ratios of those species. I'll show you some identifications of what we could identify here in just a moment. But you can see that some of the species localized again tightly to the islets, just as we saw for lipids and metabolites. But some show very, very diffuse localization, so very shallow gradient that extends from endocrine tissue into exocrine cells. And some show really also some interesting kind of, very few of them do this, but you can see this kind of localization that correlates with the glucagon localization. All right, so a lot of those species that we saw, deprotonated peptides, were identified as C-peptides. And so let me show you a little bit more about that. Again, and I'm not a biologist, so I do my best. So C-peptides are produced supposedly as a byproduct of insulin synthesis, right? So you have your pro-insulin chain, you cleave it here, you get your insulin released, and then the C chains they were considered just byproducts, and they were considered not very interesting systems until later, maybe about a decade ago or so, C-peptides have been recognized as important systems, and in fact, they do play a significant role in type 1 diabetes. So there is a lot of interest to them. And so there are two C-peptides that are usually generated depending on where the chain is cleaved. So there's C-peptide 1 and C-peptide 2. And you can also generate some truncated peptides. So some example is if you cleaved here between the 24th and 25th residue of C-peptide 1, you will generate this DES2529 C-peptide 1, and we see those species in our data. And so we did on-tissue MSMS and a lot of manual analysis. So if anybody knows how to better identify peptides in negative ionization mode, I think I asked everybody I knew would have some experience with that. But we got really not a lot of help with that. So there is really a big challenge there. So we see a lot of deprotonated peptides in our experiments. We don't know what they are. They're very difficult to identify. But in any case, after a lot of manual labor and analysis of tandem mass spectrometry data, we identified several of those peptides, and I wanted to show you their localizations. So here is again, the optical and immunofluorescence images of those tissues, and here are ion images, and just to show you how close or how far away those gradients extend away from the islet, I'll show you the outlines of the islets here. So here are the outlines of the islets. Okay, so you see that a majority of those peptides are not tightly localized to the islets. They are expressed in the islets, and then they're spread out to the acinar cells and exocrine tissues. And so in general, it's known that T cells are recognizing C-peptides, and usually just the original C-peptide 1 and C-peptide 2. And so it's not quite clear what truncated C-peptides are doing there, and maybe they're distracting T cells from killing beta cells, because there's a very fine balance there between the healthy pancreatic islets and islets in type 1 diabetes. So we don't know exactly, again, I'm not a biologist, I don't know exactly what it means biologically, but I think this is a discovery that you can make without having a target, without having any idea of what to expect from those systems. You just come across that in your data, and then that hopefully can help you generate some hypothesis how those cells communicate, right? And how those different species are involved in this crosstalk between different cell types in tissues. All right, so I still have a few minutes left. So let me tell you a little bit more about how do we enhance the selectivity of mass spectrometry imaging. And so here I'm only showing you examples of our work with lipids, but I'm also thinking that there might be something there that we could extend similar, kind of apply similar strategies to enhancing the selectivity of protein imaging. So here is the idea. So we have lipids that differ in just the location of the double bond. And just by mass analysis, we cannot distinguish between them. So how do we differentiate between those different lipids? When we do, when we scan the tissue for just mass to charge ratio, all those isomers that differ by the location of the double bond will be all lumped together. So we'll not know how they are distributed in tissues. So the way we approach this is by adding a photosensitizer to the solvent. So as I said, we can add pretty much anything to the solvent, right? So we add a photosensitizer to the solvent and we run the solvent with this, in this case, the first example, pink photosensitizer through our probe. We pick up molecules from the sample and we then irradiate this volume with the laser pointer. And so when we do that, we generate singlet oxygen. So it's photosensitized generation of singlet oxygen that then attacks double bonds, generates hydroperoxides. And hydroperoxides give very nice fragmentation patterns that are very easy to measure using even very, like unit mass resolution instrument. And so now we can do imaging experiment in tandem mass spectrometry mode and use those diagnostic fragments to map the different isomers of lipids in tissues. And so let me show you one example where this was done with high spatial resolution. So this is this lipid hydroperoxide that we generate. So this is phosphatidylcholine with the 16-0 chain in SN1 position, 18-1 chain in SN2 position. When we react this with singlet oxygen, we generate hydroperoxide at mass to charge 814. We do collision-induced dissociation on this species, and we get diagnostic fragments of two isomers of this lipid, one with a double bond in the location at carbon 9, and another one with a double bond in the location at carbon 11. And so now we can just map those fragments as we go across the tissue, and we can generate high-resolution images of those different lipid isomers. So this is, again, mouse brain tissue. This is a fractional image of delta 11 isomer, and it's kind of inverse delta 9 isomer. So you can see this really interesting differences in the localization of two lipids that only differ by the number of by the location of the double bond. All right, so we further developed a universal photosensitizer. So instead of using the one that I showed you in previous slide, can be only used for positive mode imaging. It generates huge interferences in negative ionization mode. So we found this derivative of a porphyrin. So with the carboxylated porphyrin, that is an excellent photosensitizer that can be used for this to implement this approach in both positive and negative ionization mode. And to our great excitement, we were able to image isomeric fatty acids. So this is fatty acid 18-1, and we can profile its delta 9 and delta 11 isomer based on those fragment ions that they generate. And this is just their abundances here in parentheses. We can also perform experiment where we're imaging lipids in both positive and negative ionization mode. And in this case, we had different classes of phospholipids that we were able to image in the same experiment. So we can now look at this isomeric differentiation of those different phospholipids. And some of the isomers are co-localized and some of them are not co-localized. There are different abundances of different isomers. So this just gives you a lot of additional information and insights into the role of those isomeric lipids in biological systems. All right, so I think I need to wrap up, right? Or do I have 2 minutes? Okay. All right. So just very last kind of bit of, I think, the development that I wanted to show you, and that is the implementation of this platform of nanodesi imaging on a triple quadruple mass spectrometer. So a lot of times you do need a high resolution mass spectrometer to answer your questions. But there are a lot of things that we can do on a much more accessible platforms. And we can lean on the sensitivity, selectivity, and speed of those systems to perform targeted and kind of semi-untargeted or semi-targeted experiments. So the idea here is that we'll generate a list of what is called multiple reaction monitoring MRM transitions, where you select your precursor lipid and you select its characteristic fragment. And that's all. So you will be having those MRM transitions that you will be monitoring in this triple quadrupole experiment. Then you map those diagnostic MRM transitions, again, across the tissue section. We had to develop codes for visualization of those data. So you basically take and you have to spread each of the, have to assign each of the MRM transitions to its pixel. And then you have your pixel grid, and you can then project each of those transitions onto your pixel grid to generate ion images, just as I showed you before. Okay, so here is an example where it becomes really handy. So these are two lipid isobars that are separated by 0.2 millidalton. Okay, so this, to separate those by mass alone, it will require about 4 million resolution. So that's really out of reach for most of the instruments. But luckily, those two isomers give two fragments that can be easily separated using unit mass resolution of a triple quadruple mass spectrometer. And so by doing so, we now can take those isobaric lipids and map them out onto the tissue using their MRM transitions. And we can see there is not a huge difference in localization here, but hopefully you can see that this species, the phosphatidylcholine, is less abundant in the midbrain region of the tissue. So otherwise it would be very difficult to separate those species, but you can do it using a very simple platform. And you can also separate some isomers if you're lucky, if they have distinct fragments. So this is one example of the oxidized fatty acids, where you can map them, those two isomers of an oxidized fatty acid, using the same approach onto the tissue section. All right, so let me wrap up and say that we've made a lot of progress in this field. There is still a lot to be done. I think that if we were able to achieve routinely 1 micron resolution, that would be a game changer for this field, and definitely will bring it much more into the realm of single cell or subcellular kind of characterization of biological systems. Some of the key challenges remain in molecular coverage, identification, and quantification. I showed you that hopefully the future of correlative and multimodal imaging is something that a lot of groups have been doing lately, and you will see more and more of that in this field, where you can get more additional information from other modalities. That, I wanted to thank my group members. A lot of great work, a lot of work that I showed today was done by Manxi Yang, and our collaborators who provided us with some interesting samples, the collaboration with the Northwestern group, Agilent and Bruker teams, our funding agencies, and thank you for your attention. I'll be happy to answer questions.
Awesome. Thank you for the talk. Any questions to start?
Thank you. Two naive questions. How long does it take to scan a brain tissue slides like you showed? Is it a fast process? I understand there are challenges in data analysis, but to acquire data.
Yeah, so it's not a very fast process, of course. It depends on the spatial resolution and on your platform that you have. So I think usually those experiments run for like 2 to 5 hours roughly. I've seen some people running them for 24 hours. We're trying to avoid that. Yeah, I mean, if you talk to biologists, they want to image, a carpet with a micron resolution. And that's of course usually out of reach for us. So we cannot image like a huge section in some reasonable amount of time with very high spatial resolution. But we've done pretty large human tissue sections, like human kidney sections with high resolution. That was probably about a 12-hour experiment when you do it large tissue with high spatial resolution.
Okay. And the second question, when you detected polypeptides in negative mode, so you were not able to find the same peptides in positive mode, is it correct?
No, those peptides do not ionize in positive mode.
Okay. Okay.
Yeah, we see other peptides in positive mode.
Even if you synthesize the same peptide, you
Those
will not.
peptides do not ionize. No, we had standards of C-peptide, just regular C-peptide. It does not ionize. It has a lot of glutamic acid residues. It does not ionize in positive mode.
Thank you. Interesting.
Julia, very nice talk. I have a general question about the tissue, the lipidomics for diagnosis. A lot of people using different kind of methodology to claim that they are able to use the lipid to do cancer diagnosis. And what do you think about this? Is there any specificity on the lipid species?
Oh, for cancer diagnosis, absolutely. Cancer is relatively easy to detect using lipid profiles because a lot of things change in cancer. So yes, absolutely.
Okay, one more question. And when you do the different tissue section across these axes, do you see the different distribution of the lipid, or they are quite homogeneous?
That depends on the tissue, right? So in brain tissue, we struggle a lot to find comparable tissue sections because things change so quickly in brain. But if you were to, like we were imaging, for example, muscle fibers, so you have like this long fiber, you can slice it all you want, right? And not a lot of things will change between sequential sections. With the pancreatic tissues, the size of the islet is about 50 microns. And so, again, you may be slicing, like you may lose your islet in the next section. You may not even see it, right? If you were at the edge of the islet in one of your sections, you may not see that islet in the like adjacent or maybe next to adjacent section. So yeah, it really depends on the system a lot.
I actually have a quick question. I think it's really cool that you can get the kind of resolution for the gradients across these tissue slices. Specifically, when you do the tissue expansion form of this method, do you sometimes see like intracellular gradients? I'm not sure if the resolution is that good, but is that a possibility?
I think it is a possibility. So, and I mean, tissue expansion is a relatively new technique with mass spectrometry imaging. So there's still a lot of validation that needs to be done there. But yeah, once you can see with that resolution, you should be seeing the details of this inside the cell. And there was one experiment where I thought that we saw the cell wall, but you know, it was like once. And we never saw that again. So it's a tough experiment to like really get very to get the signatures that just localize to one specific location, either within the cell or between the cells. Nature likes shallow gradients, you know, so there are no step functions in biological systems usually, right?
Gotcha. And I actually have one more quick question. It's kind of broadly speaking, you discussed that one of the main aims is to develop more and more effective bioinformatic tools for identification. Do you have a particular omics that you're most interested in having those tools developed for? Metabolites, lipids, or proteoforms?
So we're pretty good at identifying lipids. I think overall it's not bad. Well, we usually, the issue, of course, with isomeric lipids, we may not be able to identify which isomer exactly that is. SN1 is in 0 position, double bond locations, all that stuff is kind of challenging, right? But we will at least tell you what lipid that most likely is. With metabolites, well, a lot of times it's very hard. With proteoforms, if you have a database, then you can tell what it is. If you don't have a database, you're doomed, right? And so, and the same with those like C-peptides, for example, negative ionization mode, it's not a lot of tools there for protein identification and peptide identification in negative mode. And so then you really, that becomes really a huge challenge. And we see, like in imaging experiments, a lot more species than we report. And a lot of times we do not report those other species because we have no idea what they are. We see a lot of peptides, we don't know what they are. So that's where the challenge is. There's a lot of information in those data that is not utilized at the moment. They're there.
Thank you.
Really interesting talk. I just had maybe a technical question. In some of the ion images which you showed, to me, and I'm no expert in this, it seems like there are vertical striation patterns. What are those?
Are you saying that there is like a gradient going vertically up? Oh, just lines,
Yeah.
streaks?
Yeah.
So the streaks are usually, if we see the streaks, we usually don't see vertical, we see horizontal streaks, right? So if you see a horizontal streak like that, that means we lost contact with the tissue. So that's pretty standard. 14? Okay, let's see. It's on 14. So I think this is 14. Oh, those are, yeah, this is just visualization. So we run the experiment in lines. So we have line 1, line 2, line 3, line 4. So when now when you're visualizing your data, the software will just fill that entire height of the line. For each pixel. So you detected the signal in the center of that. So you have a spacing between the lines of, say, 10 microns or 20 microns, right? And so the just visualization software will fill in that gap. So you don't have data acquired between, there is a gap between the lines, right? So you acquire data, one line, then another line, another line. There is a gap between the lines that you didn't sample across, right? But software will fill in that information for you. So it will take the signal from that position in that line and spread it over the entire width of the line. So that's why it looks like that. When you have a lot of lines, it doesn't look, if you consider that pixelated. That's what the pixelated image looks like for us because our pixels are rectangles and not squares. So you can imagine a pixelated, image with square pixels. This is a pixelated image with rectangular pixels.
Awesome. Any other questions? Okay, let's give a round of applause for Dr. Laskin. Thank you. So now I think we're going to do another coffee break and enjoy.
We'll resume at 4 o'clock.
So there will be the person to ask about that. Yeah.
See, there's a swap folder that's supposed to be the last time.
Oh, okay, okay.
Moderator is also swapping between.
Oh, actually, the person who was supposed to introduce you has COVID, so he was not able to come to the conference. So yeah, this is kind of an unplanned thing, but I'm pleased to be able to introduce a fellow University of Minnesota person. So yeah. All right, welcome back, everybody. It's my pleasure to introduce Xiaokan Lun. Xiaokan comes to us formerly from Harvard University, but recently he started his own lab at the University of Minnesota in January of this year. And as far as we know, he's still there, which means that he survived his first Minnesota winter, which is an accomplishment in own right. Winter probably ended sometime last week in Minnesota. So we're pleased that Xiaokan can be here to enjoy the warm weather. But more so than surviving a Minnesota winter, Xiaokan is here because he's developed a number of innovative technical approaches that have really pushed the field of single-cell mass cytometry forward. In particular, Xiaokan will tell us today about a novel technique that he developed using DNA barcoding to increase the sensitivity of single-cell mass cytometry. So he's really pushing the field forward with his technical achievements, but I think it's also important to emphasize that he's doing so in service of really important areas of biology. So a major goal of his research is to understand signaling networks and how these interact in cancer and how we can discover new drug targets using these large-scale data sets he's generating. So please welcome Xiaokan.
Thank you so much. Thank you for the really nice and comprehensive introduction. So yeah, I just started my lab at the University of Minnesota a couple of months ago. So I'm gonna present a work that I did when I was still a postdoc here in Dr. Peng In's lab at the Wyss Institute. And so here I was trying to integrate advanced DNA nanotechnology into the platform of mass cytometry to enhance the sensitivity and multiplexity. So yeah, so for anyone who's not 100% familiar with mass cytometry, it is a highly multiplex protein epitope analysis tool that's currently allowed to analyze more than 50, there are more than 50 targets at the same time. Those targets can be protein, PTMs, RNA, and some of the metabolic sites. And it's at single cell resolution, high quantitativeness, and also the throughput can be really high as 500 cells per second. In the measurements, we can reach almost 2 million cells per hour in the analysis. So how do we do that is that simply put, we label the antibody that conventionally you can do with fluorescence agent with lanthanide metals, metal isotopes, and then use these metals to label the protein or protein phosphorylation site inside the cells. And this metal test can be differentiated or to read out by mass spectrometer based on the differences of their mass. So you can result in a really sharp peak over here compared to the spectrum overlap that you often see in the fluorescence-based analysis. And those cells are injected into the instrument. There's a nebulizer can help to nebulize the cell into droplet, and there's cells passing through the plasma, form the ion cloud, and going to the top, and to be analyzed. And the readout will be the abundance of each metals, and this can be translated into the protein or PTM abundance per every single cell. And yeah, so there's a lot of advantages using mass cytometry, but it's also some problem we need to solve. And one of these is the abundance, as Galit mentioned in the morning session, that is also really critical to measure those proteins with low abundance, because a lot of them really play an important role in biology. So currently, there's difficulty to measure the low abundance proteins at the single cell resolution with mass cytometry, especially when these proteins are in the cells in small size, such as human immune cells. And another application, imaging mass cytometry, which apply the laser ablation system, also require a high abundance or require the high sensitivity or the sensitivity to be improved. So the goal of my research path over here is to integrate a method that allow us to amplify the signal from mass cytometry analysis. What I did, or what we did as a team, is that instead of labeling the antibody directly with the metal polymer that can chelate the ions or isotope ions, we label the antibody first with a small DNA oligo. And then we use the antibody to label or to target the protein or protein phosphorylation site inside the cells or on the cell surface. And this antibody, or a lot of antibodies, are actually fixed in the cells post-staining. And after that, we can play around with the cell with a lot of procedures to do the signal amplification. So what we did is that we flush in extender strands, that is A prime, just repeated sequence of the complementary strand of the initiator that you labeled and the antibody. At a low temperature, this extender will fall onto to hybridize to the initiator, and then with the help of the polymerase BST, it's gonna extend the initiator into a double A structure, which become fully complementary to the extender. And then you increase the temperature in the system, allow the extender to fall off from the initiator strand. Now you have a double A sequence compared to a single A before the process. And then what you can do is just to repeat the process for multiple cycles, driving up the temperature down and up for many different cycles, and then you can generate a really long concatemer or extended initiator sequence with hundreds of copies of the barcode A. And then we have the detector strand or detection strand that is conjugated with the metal that can be hybridized to the detection strand binding site, which is just the A sequence, to generate a really high signal or much more increased stoichiometry between the metal and the antibody to allow you to have more abundance detection agents per antibody. And what is also really critical is that we use a modification called CMVK to modify the detection strand, which can be activated by a short UV exposure to get cross-linked into the concatemer or DNA amplification structure. So after that, everything you see in this structure from the metal to the antibody is connected by the covalent bonding. So that can help the structure to survive a harsh introduction environment in the site of analysis. So yeah, basically using the short oligo, it limits non-specific bonding, and using the CMVK-based amplification, the amplification structure can be stabilized and it facilitates the mass cytometry analysis. So we did many of different type of validations, and this is one of these I'd like to talk about. Is we just developed a transient transfection model where the GFP protein is transfected into a group of millions of cells to create an expression gradient. You can have the cell expressing nothing, so expressing low level, high level. Then we use the anti-GFP antibody to target the GFP, and this antibody is conjugated with A's that can be amplified. And the secondary antibody targeting the primary antibody is conjugated with conventional method that we can use that two signal to compare whether this amplified signal still have the good correlation with the conventional signal. So this is what we're supposed to see. Everything should be, the new approach should be nicely correlated with the conventional approach. But if we see something different than that, that means something wrong, that we can keep pushing the experiment or analysis to a better quality. So this is how what we achieved after the final optimization. We found that the secondary antibody by conventional labeling is really correlated with the primary antibody with ACE amplification. And depending on how many cycles you put into the system, you can have a like increased signal, and the correlation is always really good. So overall, we have 13-fold of signal amplification, and in a lot of cases, this is still not enough to read out some really low abundance proteins. Then we developed that we can further apply the branching system to allow us exponentially increased signal by like creating this branching tree structure. With this platform, we can achieve over 5-time increase for every branches over here. That allow us to read out the the proteins in single bacteria cells, such as E. coli. And yeah, another really critical thing for our analysis, because we're doing multiplex analysis, so our method also need to be multiplex. So in the world of DNA technology, the way to do that is by designing orthogonal DNA probe. So we did that, created 50 different probes that can, in theory, to be applied together in the same experiment, and we did it on 33 different probes for experimental validation, and found out actually most of them have really good readout, and only 2 of the 33 strands have more than 10% of spillover or crosstalk, and those ones need to be used with caution. All the others are really good to be used in our system. So yeah, right now is the application. We try to solve this problem one by one. For low abundance proteins, we applied into EMT system. So I'm not going to the detail of the system, but basically this is the cell model that can go in through EMT and be reversed by going by an MET process to epithelial cells, and we harvest the cell at different time course, at different time points over nearly a month of treatment, and trying to analyze the key phosphorylation proteins or surface markers, phenotypic markers, and low abundance transcription factors such as ZEP1, which shows a really nice trend in this then build up pseudo-time trajectory that can nicely reflect all the other markers that are indicating the phenotypical or phenotype of the cells. So yeah, for low abundance transcription factor, this is a really nice approach to use to allow the amplification. And for signaling network analysis in smaller cells, like primary human T cells, we can also use our methods to amplify the signal, increase the phosphorylation from the basal unamplified level by 17-fold, more or less 17-fold, to really allow the tracking of the TCR phosphorylation network in the time course spanning through 1 hour of after TCR stimulation. And you can see the cells are like the phosphorylation target, like CD3 zeta and ZAP70, that are next to the receptor. These are the proximal signaling response at a really sharp peak, and at the 0.5 or 2 minutes, and they start to decrease. And there's distal signaling network like metric ERG, and will gradually growing up, and they reach their peak at 30 minutes or sometimes between 15 to 30 minutes before they start to go down. And an internal control, phospho-SMAD2, which is not relevant to these the network is still stay the same, or like indicating our method to be really specific. And we can use that to build up the network, and we found the distal signal is more sustained compared to proximal signal. And lastly, we can also use that for imaging mass spectrometry, which is an alternative technology that's replaced the sample introduction system with a laser ablation, so you can directly read out from the tissue slice, but it has 1 micrometer square resolution. That means it contains a much less sample material compared to a single cell that you need higher degree of signal amplification on this method for sure. So we modify our approach to make it the cyclic amplification or compatible with tissue slice. And we do that on 20 tissue-specific markers on human kidney tissues, and found out like every single one of the markers just been light up with great signal that allow us to subclassify those cells and put them into different clusters that are blood vessel, glomeruli, and different tubular structures. And within each cluster, there's also a high degree of heterogeneity, which can be analyzed and put back into the original plot to analyze where the heterogeneity is coming from. So yeah, I would like to give a summary, which is that we apply DNA technology with the mass spectrometry to enable the analysis of low abundance marker at the single cell resolution. And yeah, so this is where the lab, Dr. Peng Yin's lab, where I did my postdoc, where this method is developed. And this is my new lab at the University of Minnesota. We're still small, but we're trying to use this method and also develop many varieties of different approaches to help with the single cell proteomics analysis. Thank you.
Questions?
I think Jonathan, great talk, by the way. Jonathan Sweetler made a point during the panel discussion about needing lots of cells, and certainly this is a method for that. So what types of applications do you think having this like much greater number of cells is kind of best for that we can try to leverage and combine with data that other types of methods produce?
Yeah, I think there's a like different, it's really kind of complementary to have different approaches for single cell mass spectrometry. We don't have the coverage as big as proteomics, but we have a good throughput, and the quantitativeness is also like good. And also, yeah, there's a I think our goal, or our also part of the future research line from our lab, is trying to use that as first of all as a screening method, so we can analyze that in like as a screening approach for drugs for different combinations of perturbations. We can use that to profile the human sample, patient sample at a really good speed. We can analyze like millions of cells from one patient times like tens or hundreds of patients in a single days of experiment to get a lot of information, especially when there's like high degree of heterogeneity. And yeah, and I think also although we don't have a really good multiplexity as what you guys have, but there's different ways that we can incorporate DNA markers, RNA, protein, PTM in a single run to generate this kind of multi-omics or multi-parameter analysis. So this is what we trying to do.
Maybe just another question here. So I think the temporal component, being able to analyze these signaling networks, is really exciting. Are there other other types of signaling networks beyond kinases and phosphatases that you're particularly excited to apply this technology to?
I think the main problem is that for any signaling analysis, we need to have good antibodies that can target specifically that particular phosphorylation site on the particular protein, and this is really achievable with phosphorylation. But for a lot of other PTMs, you just don't have the possibility to read out that with this high degree of specificity. But also we are developing other approach, or using DNA probes to try to achieve that by using, for example, two antibodies, one bind to the phosphorylation or a pan-phosphorylation or pan-PTM target, and another antibody bind to the total protein. And then these two proteins can bind to the same target and create a reaction, generate a record when that particular PTM happened on that protein. It starts to record and generate signal. And this is how we try to achieve that and apply that on different kind of PTM.
Great. Well, if there are no further questions, let's give a round of applause to Xiao Khan.
Our next speaker is Samantha O'Connor from the Translational Genomics Institute. Samantha is a recent convert to single-cell proteomics, where she spent previously some time in her PhD at Arizona State analyzing single-cell RNA sequencing data, applying into problems like neurodevelopment and the cell cycle. So I'm excited to hear about her experience in the new field of single-cell proteomics and what she's been up to.
A nectar for you need to use two. Oh yeah. My case for.
Oh. Hi, everyone. Thank you for the introduction. Hopefully I can get these slides up and figure it out. Do this. Okay. Share my slides. Where's the share button? All right. So first, I wanted to thank the Salva Lab for organizing this conference and then giving me an opportunity to talk. I just joined this project in January with no proteomics experience, so keep that in mind for questions later. Oh, where'd that go? All right, so we decided to tackle PBMCs, which we acknowledge really is a system that pushes the limits of single-cell proteomics. It was nice to hear that some other people in the audience share my struggles with PBMC analysis in the proteomics space. But so why PBMCs? Why are we studying PBMCs? They are easily accessible, clinically relevant, and they represent a snapshot of immune status. Single-cell proteomics goes beyond genomics and transcriptomics to reveal immune phenotypes at single-cell resolution. So for example, we can, SCP allows us to characterize T-cell phenotypes, which are really important for understanding immune response in cancer, autoimmunity, infection, things like that. However, as we all know, there are significant challenges associated with single-cell proteomics, and PBMC heterogeneity doesn't exactly make these challenges any easier. Having trouble with the. All right, okay, so one of those challenges is there's so much variability in PBMC cell size, subpopulation proportion, and states. We see that monocytes have around 30 times the volume of T cells, which means that the amount of monocyte material is going to be much larger than the amount of material for T cells. There's this high protein dynamic range that limits the abundance of low abundance proteins, and additionally there's this marker overlap between subsets that really does complicate cell type distinction. So really awareness of these limitations is really essential for accurate analysis and interpretation of this data.
Additionally, single-cell proteomics itself has its own sample prep and acquisition challenges. So here to demonstrate this point, I picked a human PBMC single-cell proteomics dataset with the most significant batch effects here. We see single cells on the y-axis and proteins on the x-axis, and blue means low missingness and white means high missingness. And we can see here in the red dashed lines, there's this substantial protein dropout from carrier channel to single cells. So we aren't seeing any of these proteins at the single cell level. So when you are filtering, it dramatically reduces our data matrix. Additionally, we're seeing these signatures that align for cells that are in the same run order, which we wouldn't necessarily expect to see. So significant batch effects that require correction for meaningful interpretation. Additionally, there is a lack of universal best practices for pre-processing steps, so normalization, imputation, batch correction, and even the order of these steps. So here is an example of sort of the first mouse PBMC single-cell proteomics dataset that we looked at, we acquired. And we can see these are single-cell correlation plots at the peptide level, the protein level, and imputed protein level, and batch corrected level. And you can kind of see that this inherent heterogeneity of these PBMCs are being flattened through these processes. And so we don't exactly want to use this batch corrected matrix as the input into our downstream analysis. So sort of figuring out sample acquisition and sample prep, as well as pre-processing steps for this heterogeneous complex data is really important. So this is sort of the way that we're trying to solve this problem. We're using normal PBMCs, human and mouse PBMCs using NPOPs. Thank you, Andrew, for that technology. And to build this reference single-cell proteomics library for eventual comparison with disease-associated immune profiles. We're leveraging this matched single-cell RNA sequencing data to help and guide with annotation, cell type annotation. Additionally, leveraging these synthetic peptide standards to enhance that accuracy and reproducibility of the cell annotation. And then eventually, we are wanting to validate our annotations with sort of these, these bulk proteomic signatures of subsets of immune cells. And while I don't have this data yet, this is the next step in our pipeline. And so first, I will walk through some of our mouse PBMC data. So we isolated, acquired, and 690 cells and around over 1,400 proteins successfully passed our quality control metrics. We're seeing around 730 proteins per cell here, and some of them are split by being very variable, although it is important to note that we do see very low expressed proteins here. But of some of these highly variable proteins, as a gut check, I just wanted to make sure that they aligned with immune cell biology. So we see CD20 is one of those highly variable proteins. That's a marker for B cell development, differentiation, activation. We see a marker that plays a key role in immune homeostasis, for example, and then S100A9 is highly expressed in monocytes. So that was a nice gut check to see that these variable proteins are sort of cell type specific or have immune related, obviously, because our samples are PBMCs. And then so next we decided to cluster the cells, and I wasn't very optimistic about this just because of our input matrix, but when we clustered, we can obviously see these 6 distinct clusters, and we can tell that they're distinct using with the heat map on the right, where we're seeing these proteomic signatures for each of these clusters in the diagonal. So these proteins are expressed in cluster 0, and they kind of define those proteins or those clusters. So interestingly, there's CD79B, which is really small on the screen, so I've got an arrow, which is a strong marker for B cells. That's a marker for this blue cluster 4, so suggesting maybe that this cluster 4 could be B cells. But because we did have this matched mouse PBMC single-cell RNA sequencing data, we decided to sort of leverage those labels and the gene expression to see if we could map the labels that were for the gene expression data to the proteomics. So I chose 2 different approaches to do this. Seurat has its own function. It's called transfer data, where it basically just takes the reference
cell labels and gene expression profiles and transfers to a query data set. So our query would be single cell proteomics using these anchors. Additionally, we could build different classifiers. So using the PBMC single cell RNA sequencing data as our training set here. Oh, I'm way off. Training set here. And sort of doing this cross-validation analysis, testing and evaluating different classification algorithms to see and predicting with ground truth labels that were already labeled. And so you can compare the predictions with the labels and do a variety of evaluation metrics, which I'll talk about in a few slides. But so first I'll talk about the transfer data predictions. And surprisingly, we see this function. It does yield similar proportions to the matched single-cell RNA sequencing data. So we see the highest percentage of the mouse PBMCs are these B cells. We're seeing the highest percentages in the protein data as well. So this suggests that maybe SCP will highlight the reliability of this approach as a complementary approach to studying PBMCs. Additionally, so this is the classifier evaluation. So I compared each of these classifiers with F1 score, which is a great metric for multi-class classification of imbalanced data sets. It balances both precision and recall. And we can see sort of a wide range of scores. So 0 would mean poor prediction, 1 would be the best prediction. And most interestingly is we're seeing that subpopulations with lower representation. So here we're seeing cell types with 55, or dendritic cells with 55 cells in the training data set. Sorry. And a subset of T cells, they have, on average, lower F1 scores compared to the more highly represented cell types. But there were 2 models, a neural network and an SVM model, that had higher average F1 scores across all of the cell types, suggesting that maybe they are promising for accurate cell type annotation on unseen data, as these predictions were made on a held out set, so they weren't involved in the training. And so when we apply those models to the single cell proteomics data, it's very interesting. We see very different profiles here. SVM, well, it looked pretty promising in the cross-validation analysis. We're saying it's only predicting B cells here, whereas the neural net may be a little bit more representative. If we kind of subset that rarer T cell subpopulation down, we could see sort of a similar match there. But overall, the transfer data function did provide the closest matching cell type proportions to the reference single cell RNA sequencing data set. However, when we overlay those predictions on that clustering UMAP, or the UMAP, those same predicted cell types aren't clustering together. So we thought maybe we could incorporate and spike in these peptide standards, which Ramel had a poster yesterday, and I'm sure he would pull it out for anyone who wants to learn more about it. But essentially, they are peptide standards for peptides that their parent proteins are cell type specific markers. So when we are not spiking in these peptides, we aren't seeing the cell type markers pop up in the single cell proteomics data. So with those markers, we'd hope that we'd see better annotation because they'd be present. So kind of boosting the signal of important markers that we want to see for annotation purposes. And just sort of to emphasize that point, this is single cell RNA sequencing data that we analyzed, clustered, and annotated. Human PBMC data, and just to show that these parent proteins of peptide standards, we see the transcripts are specific markers for subpopulations. So we see CD3G here, that's a marker for a variety of T cell subpopulations, and CD79A is a strong marker for B cells. So we'd hope that we're boosting that signal in the single cell proteomics data and ultimately being hopefully able to annotate it during the clustering as well. So this is the human PBMC single cell proteomics clustering. I did collapse those annotations before prediction because we're not going to reliably be able to identify rare T subset. So I collapsed those annotations. And we can see that although these predictions do look a little bit jumbled, when I do some hypergeometric enrichment analysis, we can see that cells that were predicted B cells are also cells that are in cluster 2, suggesting maybe this cluster 2 could be a B cell cluster or B cells. Additionally, for like monocytes, for example, it has the highest enrichment in cluster 0, suggesting maybe there is some signal through. It's just not the because of the missingness of the single cell proteomics signatures of these specific cell types. We're not seeing all those markers, so it's hard to really cluster them. Additionally, these predictions do align with average frequencies in enriched human PBMCs, so we are seeing that match to biology. And lastly, I just want to show this analysis, which was sort of just for fun, but it's ending up being my last slide. I took the proteins that were identified in our human PBMC single-cell proteomics data and subset my single-cell RNA sequencing data to just those transcripts that were the transcripts matching the proteins, and reclustered just on that subset of single-cell RNA sequencing data to see if those proteins identified would still separate the cell types. And we do see this separation, particularly in the B cells and the monocytes. Obviously, we see T cell subsets cluster together, but it was really interesting to see that these proteins are functional and specific cell type markers. I also wanted to look at the mouse PBMCs because we collected and analyzed those proteins as well, and those proteins still, when matched to the mouse or the human transcripts, clustered with just those mouse features, still indeed did cluster the cell types. So this offers really exciting potential for cross-species and translational immune profiling, and it tells us that these proteins that we're identifying are really important for cell type discovery and annotation. And so in conclusion, I just want to emphasize again that these PBMCs really do offer a readily accessible window into systemic immune function and disease pathophysiology, and awareness of these limitations both on the sample prep, acquisition, and pre-processing is really essential for interpretation of this PBMC data. Classification does produce some cell type proportions consistent with matched single cell RNA sequencing, and the hypergeometric enrichment does suggest that there may be some overlap with clusters and some of those predictions. And then finally, those markers are functionally relevant and do show potential for translational applications. And with that, I just want to acknowledge my team, Patrick Peroty's lab, and Ritton and Christine, who weren't here today. They're instrumental in helping me get started in January. And then our collaborators at the City of Hope, who've donated our samples, and of course, Andrew and Josh at Cell One. And that's it.
I'll start off with a question. So in single cell RNA sequencing, I think like for people who have followed best practices for the analysis, like famously, there's definitely been a lot of debate, like how you do pseudo counts and differences between Seurat and ScanPy, and like these things took a really long time to kind of hash out best practices, essentially involve people berating other people on Twitter. So do you have any thoughts on like the most kind of prompt path to achieving best practices kind of for single cell proteomics analysis in maybe a slightly more efficient manner?
I would like to answer that, but I don't know the answer to it. I think just kind of continued communication with those that are doing PBMC analysis. Like I had a conversation with Luke yesterday about his integration, and so really promising results with his integration. So I think just more open communication about just processing really complex heterogeneous samples in this space is really important.
Yeah, I had a great conversation yesterday about this and really cool work. I processed a lot of PBMCs and they're super fragile, at least in my hands. I'm not a very good biologist that way, but I guess what type of filtering or kind of how do you account for maybe dead cells in the data set, or do you do that type of filtering?
Yeah, so Ramel actually does all the experimental work, but I know that he has used platelet removal kits, and we use Cytox green in our Cell One to remove dead cells. We have sort of tried to go as low as we can on diameter filter. I know Andrew wants me to increase that filter, but with PBMCs being pretty small, we want to try to capture them But yeah.
All right. Thanks so much again, Samantha, for the talk.
Oh my gosh. Yeah, of course.
Is that?
Okay, so while they get set up, so our next speaker is Professor Jesse Meyer. He's currently at Cedars-Sinai. He did his PhD at UCSD in the Commonwealth Lab, co-supervised by Professor Bandiera. He then was at the Buck Institute working with Professor Schilling, and then he also worked with Professor Kuhn for a while as well. Professor Meyer works, I think now, on a lot of computational tools and kind of the mixture or the point in the middle of data science and biology and trying to use kind of predictive methods. He's worked on a lot of things, including trajectory inference, a recent evaluation of ChatGPT. And I think now, given where he is, he also works on a lot of applied biology, so to speak, of course, with good access to samples and so on. So yeah, we're looking forward to your talk and thank you for being here.
Yeah, there you go. Nice. Awesome, thank you. That looks great. I think that's perfect. Oh.
Okay.
Awesome, thank you. I can just do that, that's fine. Okay, thank you. All right. Good afternoon, everyone. What an amazing conference. Thank you to the organizers for giving me this opportunity to speak. I'm really excited to share with you some of our recent work where we're using single muscle fiber proteomics to understand mechanisms of sarcopenia. But before we jump into that, I want to give you a one-slide overview of my lab where we're committed to understanding and driving healthier aging with a focus on preventing sarcopenia and Alzheimer's disease by improving our understanding of metabolism. To achieve this, we generate knowledge from data through visualizations, statistical analyses, and machine learning. And our favorite data to use for this pipeline is proteomics data, while we also explore the synergies of multi-omics and single-cell omics and electronic health records. So today I'll share with you a story that spans all three of these boxes where we're asking the question, what causes sarcopenia? Before we dive into the data, I want to give you a quick overview of what we're covering today. We'll start with some background about muscle aging and why proteomics can give us a unique lens into that process, especially at the single muscle fiber level. Then we'll talk about something we encountered unexpectedly, this protein isoform aging, or isoform confusion with aging. And then we'll conclude by discussing a novel type of muscle fiber. So with this roadmap in mind, let's start with that background. Sarcopenia is the loss of muscle mass and strength with aging. You can see from these plots that starting in our 50s, we all start to lose our strength and muscle mass, and we're left with about half of the strength that we started with in our 80s. Sarcopenia can be slowed by resistance training, but it can't be stopped. It is inevitable, and it'll happen to everyone. And for that reason, we should all care about it. Because this loss of strength leads to a lack of mobility, and that leads to decreased quality of life and higher morbidity rates. So preventing sarcopenia would improve your healthy lifespan. But unfortunately, the molecular mechanisms that lead to sarcopenia are poorly understood, and that impedes our drug development. Your muscles are made up of many tiny fibers that each produce contractile force. You have two main types of muscle fibers. You have fast fibers and slow fibers. Your fast fibers are responsible for high power generation, for example, if you're sprinting or carrying something heavy, and your slow fibers are responsible for low power generation, for example, the posture while you're sitting here right now or running a marathon. This heterogeneity in muscle fibers means that approaches that homogenize whole muscle tissue will lose the picture of complexity that we have in our muscles. And we know that that fiber-specific detail is important because several studies have suggested that sarcopenia is a disease that only happens in your fast fiber type. This is data from my collaborator, Chris Sundberg at Marquette University. And in his lab, they take biopsies from human leg muscle and they measure the power from individual fibers that they dissect out of those biopsies. So you can see from this data that in the fast fibers, we're losing our power in fibers from older individuals. And this happens in both men and women. And you'll notice that there's almost no change in the power in the slow fibers. So because we think that only the fast fibers are affected with aging, we want molecular changes, or we want to understand the molecular changes that are happening in the fast fibers. And in 2017, this landmark paper was published by Marta Murgia and others in the Matthias Mann group, where they reported for the first time single muscle fiber proteomics in human samples. And in total, they measured 152 single fiber proteomes from 4 young and 4 old males. They detected 6 statistically significant protein changes between young and old. And this paper has been highly impactful and it's been cited over 300 times in the last 8 years. So it's a very important paper. But unfortunately, in order to scale up, we need new methods that will allow us to apply single fiber proteomics to population-based studies so that we can improve our statistical power and detect more proteins that might be mechanistically responsible for sarcopenia. That's where my group comes in. So shortly after we moved to Cedars-Sinai, we started applying single-cell sample prep methods to single muscle fiber samples. And in 2023, we published this technical paper describing this workflow applied to 53 single muscle fibers from 1 young and 1 aged female. Our collaborator dissects out those single muscle fibers under a microscope. They put them in single wells of a 384 well plate, and then they ship the plates to us. And when they get to Los Angeles in the mail, we do that 1-pot sample preparation. And this is important because it means that we can prepare samples exponentially faster than those in-stage ship methods that other papers are using. And using the 15 minutes per fiber dual trap single column setup that was developed by Simon Kramer, who's third author on this paper, we're able to collect data from 96 fibers per day, which means we're collecting data almost 10 times faster than that previous study that I mentioned. So one of the first things we checked with this data was whether we could differentiate the fast and slow fiber types. Remember, I told you there's 2 types of fibers. So we reduce our whole proteome profiles into 2 dimensions with PCA followed by UMAP. And on this graph, each point now represents a single proteome from one of those 53 fibers. You can see that we find 2 clusters with the unsupervised Leiden algorithm. And when we overlay the major myosin-heavy isoforms that delineate fiber types, you'll see that this group on the left is correlated with myosin-heavy 7, which is the slow isoform, and this group on the right is correlated with the quantity of myosin-heavy 2 and 1, which are the fast isoforms. So this shows us that we can use these canonical markers of fast and slow fibers to classify our fibers based on the proteomic data alone. So now that we had that information, we designed a much larger study with over 1,000 muscle fibers. And this was the first study in females that we're aware of. And this time we had an average of 100 single fibers from each participant, 5 young and 5 aged. And we did our single cell sample prep in those 384 well plates, and we collected our data with the timsTOF Pro 2 at 15 minutes per fiber using capillary LC actually, because we get so much material from these muscle fibers, we can go to higher flow rate. And the data came out beautifully. We got about 57% of the fibers left were from the young group and 43% were from the aged group. And when we look at the distribution of protein counts, it was higher than we had seen in that original study at around 700 on average, or the median of about 700 per fiber. You'll notice that this number is a bit lower than some of the other single cell studies that we're hearing about. And one reason for this is that we have a big dynamic range issue in muscle fibers where most of the protein is actually the contractile apparatus. So getting 700 proteins in 15 minutes is pretty good, in my opinion. And you'll notice that in the end, we were left with over 1,000 fibers with over 200 proteins that were quantified. So now that you've understood the experimental design, let's dive into what we found, starting with this concept of protein isoform confusion with aging. We can determine the fast and slow fiber types based on the proportion of myosin-heavy 7 and 2 using a simple equation like this. And we call a fiber pure if the isoform fraction is over 80% of any individual heavy isoform. Here's what that looks like for all of the young fibers. Each point along the x-axis here is one fiber, and the y value gives the proportion of each myosin-heavy isoform, 1, 2, or 7. These are the main isoforms. So you can see from this graph that generally we have a high proportion of 7 in purple. These are our slow fibers, or we have a high proportion of 2 in blue. These are our fast fibers. And then we have some in the middle that have mixed proportions. These are hybrid fibers, and those are well known to exist, and those are normal. If we look at the percentages, roughly half of our fibers are slow and 31% are fast, and the rest are hybrids from the young individuals. And remember, this is about 570 fibers that I'm showing you on this graph along the x-axis. Let's look at the same graph for the fibers from older individuals. Now you can see that we have a similar proportion of slow fibers at 49%, but we've decreased the percentage of fast fibers to 20% from 31% in the young cohort. And the balance is made up by these hybrid fibers. So it's well known that from the perspective of the myosin-heavy isoforms, that fast fibers decrease with age and that the hybrid fibers increase. But beyond myosin-heavy chains, several other protein isoform pairs are expected to match fast or slow fibers. For example, circupumps or ATP2A1 and 2. However, it's not known whether these proteins become more hybridized or confused with aging. So this plot shows you a histogram of the proportion of ATP2A1 relative to its isoforms across all of the fibers that we measured. And we have it split by the grouping of old and young. With old in blue old in orange and young in blue. And what I want you to notice is that the young fibers are more likely to have a proportion of either 0 or 1, and the old fibers are more likely to have these intermediate proportions of the isoform. So this perfectly highlights the concept of isoform confusion. When we age, there are more fibers that don't know what they're supposed to be anymore, and they're expressing intermediate levels of each isoform. And again, although this is known for the myosin-heavy and light isoforms, it hasn't been reported for these circupumps or any other proteins that we could find. So we wanted to know whether other protein isoform groups are becoming confused with age. And to test this, we devised this unbiased pipeline looking for protein isoform group proportion differences that might be different between the old and young fibers. And this is complicated, so let me walk you through it. So we start with 509 protein isoforms in 142 groups. We calculate the isoform ratios, we group them by old and young, we sort from low to high, and then we use a KS test to determine whether those two distributions are statistically different. And we use stringent filtering of those corrected p-values to decide whether an isoform group has become confused with aging. And the rest of this is all just putting it into groups, so you don't have to worry too much about that. But this pipeline discovered many confused isoforms among the proteins that are known to distinguish fast versus slow fibers. That's what I'm showing you here in these graphs. So we did find this circupump was statistically confused with age. And these plots are a little different than the way I showed it to you last time. So you're looking at the ratio of the isoform ordered by increasing purity of that isoform across all of the fibers that we measured. And you'll notice there's two lines again for young and old. And you can see that the shapes of the orange and blue lines are statistically different as determined by that KS test. And you'll notice that generally there's less pronounced transition for the fibers from old people. And you'll notice a number of other contractile proteins that were also dysregulated. To our surprise, beyond the canonical fast and slow isoforms, we found 23 additional isoform groups that were dysregulated with age. And I'm showing you two examples here, ECI2, which is involved in lipid metabolism, and RTN4, which is an important protein in many different cellular processes, and it's found in the endoplasmic reticulum. So among the other dysregulated isoforms that are listed below, you might notice that a lot of these are actually mitochondrial or metabolic proteins. So to summarize this first section, these data reveal that this pervasive age-related isoform misregulation might be a potential novel mechanism undermining muscle power as we get older. The fast fibers are losing their identity, and they have these confused isoform proportions, and that changes their contractile mechanics and their metabolism and reduces our maximal power. In this section, we will talk about a novel muscle fiber subtype that may explain those differences that I just showed you. So for context, while we've been working on trying to publish this paper, another study was published that measured the proteomes of over 1,000 single muscle fibers. And in this study, they found evidence for only 2 muscle fiber types. And those types were clearly correlated with either myosin-heavy 2 or 7. And there's a little bit of myosin heavy 1, but it's always in the fibers that have myosin heavy 2. It's essential to note here that this study only used fibers from healthy young male volunteers. Because in disagreement with their paper, when we look at our proteomic data, we find 3 fiber types with unsupervised clustering. And when we look at the myosin heavy isoform markers that we've seen before, we see that this one cluster on the top left is our slow cluster. This cluster on the bottom is our fast cluster. And we have this third cluster that we're calling dysregulated, and it has a mixture of the myosin heavy 2 and 7. Notice that this dysregulated cluster is the only one that's more prevalent in the fibers from old individuals. Remember, our baseline rate is 57%, so this is a substantial deviation from that. And also remember that the study I showed you on the last slide only included data from healthy young male volunteers, so it's possible that this third fiber type is actually specific to females or specific to older individuals. So what is this third fiber type? In this analysis, we use Wilcoxon rank sum tests to look at the markers of each cluster. And I've overlaid here the main 8 markers of this dysregulated cluster. These 2 in the middle, PRDX3 and LGAL-S1, may signal inflammation or oxidative stress or compensatory regeneration. The 4 proteins on the edges are actually metabolic proteins, and the increase in all of them together is actually contradictory. CKM and enolase are supposed to mark glycolytic fast twitch fibers, and carbonic anhydrase and myoglobin should mark slow twitch fibers. So this is further evidence suggesting that this cluster is metabolically confused. It's known that the fibers can switch their types. There's a number of things that can cause that, for example, space flight or changes in activity. So we wanted to model the underlying process of type switching, and we can use trajectory inference to do that. And that's a metric of omic similarity among our muscle fibers. And it enables us to predict how the fibers might change from one state to another. So here we use this PAGA trajectory inference to determine the relationships between our 3 clusters. And in this analysis, I'm showing you the data when we put in all of the fibers. And you'll notice that the line between fast and slow is maybe slightly stronger than the other 2, but overall the relationships are quite similar. If we run PAGA on only the young fibers, you'll notice that now this dysregulated cluster is the odd one, and there's a stronger connection between fast and slow. And remarkably, if we run it on only the old fibers, we see that PAGA determines a shift in this relationship so that now we have roughly equal paths from fast to slow and fast to dysregulated. So this suggests that this path from fast to dysregulated might be becoming stronger with age. We can further use these relationships from PAGA to infer a pseudotime, which you can think of as a guess of how these fibers might be transitioning along the process of interest. And if we pick the fast fibers on the bottom here as the root node, we can plot gene quantities along that inferred pseudotime. So this plot from fast to slow along this left path shows you that our fast contractile proteins here are decreasing along pseudotime with an increase of the slow contractile proteins. So they're becoming slow fibers. And at the same time, our glycolysis proteins are going away while the TCA proteins are coming up. If we look at this, the path from fast to dysregulated, you'll notice that it's generally the same trend, except these glycolysis proteins are now flipped. So they're becoming slow fibers, but they're increasing their glycolysis proteins. Again, further evidence supporting that these might be metabolically confused fibers. And remember that the slow fibers are supposed to generally be oxidative, so this is contradictory. Tying this back to the first section, it appears that these 2 findings might be related. So here I'm showing you blue dots are the single fibers from that dysregulated third cluster. The green dots are the fast fibers, and the orange dots are the slow fibers. There are 2 lines again. You probably can't see that there's triangles and circles for whether they're old or young. And you'll notice that most of the fibers from this dysregulated group are those with these intermediate levels of the circa isoform fraction. And we think that this is an interesting and important observation because it's consistent with this broader information theory of aging that was set forth by David Sinclair's group at Harvard University here in Boston. And the idea is that during development, our bodies produce these cells with well-defined identities, and then those cells lose their identities as we age, and that causes tissue and organ dysfunction. So in their original work, they showed this in the context of epigenetics, and our work might be providing some protein level evidence for this at the isoform switching level. To summarize all of these unpublished results that I just shared, we found this story about isoform confusion, where the old fibers have more intermediate levels of these various protein isoforms. I also showed you that this transition from fast to dysregulated is stronger in the fibers from old people, and the path from fast to dysregulated includes a shift to slow contractile proteins with an increase in glycolysis proteins. So these three results together support a story related to fiber identity loss with aging, which is consistent with this information theory of aging. I didn't have time to get into the statistical analysis that we originally set out to do with this data set, but we find one protein that by itself can classify young versus old fibers, this MYBPH, and actually not much is known about the function of this protein other than the fact that it binds to myosin heavy chains. So we're looking into the function of that right now, trying to make virus and knock it out in mice. We're also investigating the possibility of whether this third fiber type may be considered a form of post-mitotic senescence, because some of the proteins we found are markers of senescence, especially LGAL-S1. But overall, I hope that I've convinced you that single fiber proteomics can give us a unique lens into the biology of our muscles, and it can give us hope that we might be able to find a mechanism that we can target to prevent muscle loss with aging. One unrelated slide before we conclude. This is a platform that we built for conducting single-cell omic data analysis. And if you want to check it out, you can scan this QR code. I'll also have the URL on the next slide. But this platform is unique because it allows you to do your data analysis with a drag and drop JavaScript interface. So you can design analysis pipelines and run them quickly. And what's really cool is once you decide on a final analysis, you can share your data, the analysis pipeline, and the results that you produced all together with a short URL that you can put in the methods of your paper. So it's a great way to increase the reproducibility of our scientific studies and also lower the barrier to doing interpretive analysis. And I forgot to mention that it includes a node that will allow you to generate your own personal instance of cell by gene, so you can interact with your data and overlay whatever metadata you want onto your clusters. Now I'd like to thank everyone who's been involved in these projects. Cameron has been leading the data analysis for the single muscle fiber proteomic data. He's been with us for less than a year and he's doing an incredible job. Simon Kramer collected that data and Yu Ming helped with the sample preparation of those fibers. Amanda led the original technical study that we published in JASMUS that I mentioned, and Lex is leading the development of Pisces, which is the URL is down here on the bottom. This has been a close collaboration with Chris Sundberg's group at Marquette, including multiple people on his team, Isabel, Laura, and Carlos. We're grateful for funding from the NIH and the ASMS Research Award, and we're also grateful to work with so many people at Cedars-Sinai on the single-cell proteomics team, especially Jenny Van Eyck and Sarah Parker and all of these other people. I included my contact information here in case you want to chat about anything. And finally, I'd like to thank all of you for your time and attention. Now I'd be happy to take any questions that you have.
Hi, lovely talk. A couple of questions. So first, I guess you're looking at multinucleated cells, right? So I'm curious if every nucleus does slightly different things. So if you could do a single nucleus analysis and you'll see a bifurcation rather than a third population popping up. And then I have another related question, which is myocytes are known to switch metabolism as they differentiate during development. And so I wonder if what you're seeing is a sort of de-differentiation, especially with this glycolysis TCA cycle switch that you see. And then third question, I guess I really loved your data, is about if you have in the old population data on strength, and if you can correlate what you see with that as well.
Yeah, those are all great questions. So I'll start. I think the first question was about the fact that they're multinucleated and whether there's potentially heterogeneity in the nuclei. Is that right?
Yeah, and let's say you see that third population, right? But maybe it's driven by one nucleus doing one thing, and then you have a mix.
Yeah, it's a really good idea. I have limited knowledge about muscle physiology, but these are questions that I also ask my collaborator, Chris. We've been talking about trying to do single nuclei proteomics and even trying to explore the heterogeneity along the fiber. Because there's multiple nuclei, it seems like there could be localized pockets of different proteomic signatures, and so that'd be a really cool thing to explore.
Then maybe the strength, if you like.
Oh yeah, so he does, yeah, they measure whole limb function for all of these people. And our plan is that we wanna do combined measurements of power and whole limb function and morphology and proteomics. That's where we wanna get to. So we're working towards that.
I guess the third was about de-differentiation with the glycolysis to oxphos switching.
Yeah, it's a good hypothesis. How would we test that? We can talk later.
Thank you.
Thank you.
Nice talk. Sorry, I'm curious whether you've looked at dystrophic muscle. And the reason I ask is generally the way dystrophic muscle is analyzed is you essentially inject, membrane impermeable dyes into muscle or into animals, and then look for uptake of this dye to look at markers of damage. But it's really not a great assay, to be honest. And I don't know, at least a decade ago when I studied it, there wasn't much known about the proteome or really anything related to the biology of muscle that lacked, a critical factor like dystrophin, but wasn't yet fully damaged and apoptotic, let's say. So I'm curious that whether you've done anything like that. And then as a side, have you looked at cardiac muscle at all? I'm curious to hear whether that's, you find similar trends. And there isn't really fiber type in the way, you see it in skeletal muscle, but it might be interesting.
Yeah, I'll start with the cardiac muscle. So that's something that actually Jenny and her team are looking at. They isolate single cardiomyocytes from human hearts, and they just published their first paper on that in MCP. So I direct you to check that out. It's a really cool paper. We have not looked at muscle from people, I think you're saying people with muscular dystrophy. Yeah, we have not, but now that we have this technology to do high throughput single fiber proteomics, we could look at any muscle. And actually, the data I showed you is from the timsTOF Pro 2. We're now running these fibers on the Astral, and we're getting like 2,500 proteins. So I'm excited about applying this to lots of areas in the future. Thank you for the question.
Any more questions?
A microphone here. Hey, Jesse. Very interesting talk. I have a couple of questions. The first one is, you focus substantially on the ratio between isoforms, and this is part of how these fibers are being classified, so it makes sense. But I wonder if there are significant abundance changes in many proteins that are not just restricted to isoform switching, but general change in protein abundance that might be helpful to bolster your conclusion of how these muscle fibers are changing. And my other question relates to the sampling of different fibers from these samples, especially when it relates to having a shift in the distribution over different muscle fibers. Then having unbiased similar sampling of fibers across different samples, different ages, becomes important to be able to make that inference robustly. Can you comment on how you're doing this sampling and what are good practices to ensure uniform sampling?
Yeah, that's a great question. So they do the muscle fiber isolation in Milwaukee, and I've talked to Chris about this, and he says that there is a wide range of sizes and even integrity of the fibers. Apparently it's well known among people who isolate muscle fibers that there are a lot of really sort of weak fibers that are they'll just fall apart, and it'd be very difficult to measure the power that they can generate. So we might be undersampling some fiber types. But I believe as far as their dissection, they're putting anything they can in the plate. But that's a great point. To your first question about differential abundance, I didn't have time to get into that, but we did run statistical analysis using linear mixed effects models. And consistent with the idea or the intro that I shared about the idea that the disease is primarily in the fast fibers, we see almost no changes in the slow fiber type, almost no changes in the dysregulated fiber type, and I think 55 changes in the fast fiber type. So there's some remodeling that we're detecting in the fast fiber type. I think we're still a bit underpowered, and we're not quite detecting everything that we can and should. So we're hoping to scale this up to 30 or more participants and try to get more power.
And so if I understand correctly, your strongest signal is at the level of isoform switching because these other changes are relatively small compared to the sample sizes that you have?
I would say that's generally true. I think there's a signal in the statistical changes, but I thought this isoform story was a lot more interesting, so that's why I wanted to share that today. The changes that I found statistically weren't immediately coherent to me in a way that I thought would be cohesive for presentation. So,
Thank you.
thank you.
I think if there's no other questions, we can move on. Okay. While they get set up, I'll next introduce Hannah, who's going to be talking to us about statistical methods for single-cell proteomics. Hannah is in the pain lab at BYU. She's briefly been a neighbor of ours in the past too. I don't know if virtually or actually here, but she's worked with Professor Vitek as well for an internship. Hannah has explored fragmentation spectra for single-cell data. She's also looked at, sample sizes for pseudotemporal ordering. So yeah, Hannah, we're looking forward to your talk. And let me know if you run into the dreaded technical issues.
Okay. Just share my screen, I guess. Oh, sorry. Stop sharing. You should be able to share now. Okay. Okay. All right, so yeah, my name is Hannah Bokwig. I'm in Sam Payne's lab at BYU, and I've been collaborating with Olga Vitek to come up with statistical methods designed for single-cell data that will more accurately detect differentially abundant proteins. So this is the outline of the talk. First, I'm gonna go over the problem statement, what people are currently doing, gonna present the data sets that we use, then I'll get into the results and the models, and then I'll leave you with our recommendations. So why do we even need a statistical method just for single-cell data? So a very common method that is used in bulk proteomics is group comparison, where you average all the proteins, all the healthy subjects, and compare them to the average of all the disease subjects. And you go protein by protein looking for proteins that will be differentially expressed between the two groups. Single-cell data gives us an additional layer of replication. So with bulk data, we just had replicates of people, but with single-cell data, we have replicates of cell for each person. And with this information comes a lot of questions that we are posed with now. For instance, each cell is like biologically unique. So how do you actually incorporate that cellular heterogeneity into a method that typically just averages all samples together? Also, you have cell type information now. So here I'm showing green cell type and blue cell type. How do you incorporate that into it? Also, you have to have a way of annotating these cell types. Here they're colored, but they don't come that way. We have to do that ourselves. Another question is, what happens if you're not able to sample as many cells from some subjects? Is that going to affect the results? How do we handle that? Can we just ignore it? So these are the kind of questions that we need to answer if we're going to really take advantage of all the information available to us with single-cell data. So this is the data matrix kind of showing all the features of single-cell data and what we need to incorporate. So we have condition, subject, cell type, and then the cell level information. And we also have the between subject variability, so differences between people, and then the cell variability, which is differences between cell. And this is very important that we incorporate these correctly into the model to improve the model sensitivity and accurately describe the data that we have. So if we're able to do this and incorporate all the features of single-cell data, we can ask really interesting questions that bulk experiments are unable to ask. And here are some examples. How are smooth muscle cells changing between healthy patients and patients with a heart aneurysm? How are tumor cells reacting to a small molecule inhibitor compared to somatic cells? And what are differences between T cell subtypes? So this is what people are currently doing. They're currently trying pseudo-replication, where you treat each cell as a biological replicate, and they're trying pseudo-bulk, where you're averaging all cell types together. Also, for annotating the cell type, there are two methods being used. One, you can use an external biomarker, or you can use k-means clustering. What we need is a method that separates subject variability and cell variability, incorporates the cell type, and allows us to test for the various hypotheses I just mentioned. So incorporating cell type is important for detecting proteins whose expression relies on cell type. So that's what this graph is showing. It's showing a differential protein that is going up in the green cell type and down in the blue cell type. So it is differentially expressed, but only when you'll only detect it accounting for cell type. As soon as you average these cell types together, you obscure the difference. And I'll refer to this as the X pattern throughout this presentation. All right, so the data sets we used two. I simulated a data set, so we had known positives and known negatives, so we could track what the true positive and false positive rates were doing. And in this data set, I generated it to have two conditions, 10 healthy subjects, 20 diseased, 2 cell types, and 1,000 proteins per cell. So 980 of these proteins are our true negatives, so they're not changing between cell type. 10 proteins follow the X pattern, and 10 proteins are increasing in both cell types. And to show that the results we see with the simulated data remain true for a real data set, we use this biological data set from Sarah Parker's lab at Mount Cedars-Sinai that has two conditions, wild-type mice and mice with Marfan mouse aneurysm. We have 3 female mice and 3 male, but here I'll just be showing results for the male mice. 5,000 cells total and 5 cell types, but in this we only annotate the fibroblasts and the smooth muscle cells. All right, let's get into the results. And this first section, I'm just gonna focus on how to actually incorporate this additional level of replication. So this is the main work that we did, is formulating these group comparison models. So on the right-hand side are models that capture what people are currently doing, and on the left-hand side is gonna be our recommendations. So we characterized each model, we summarized the assumptions that each model relies on, and then we formulated the hypotheses. So these first 3 ignore the cell type. These next 3, they subset cell type to kind of account for, well, I know that if I average all cell types together, I'm going to be losing information. So it selects for one cell type. But then you're losing power because you're getting rid of, obviously, a lot of your data. And then our recommended model, it incorporates the cell type, but it does have to meet like additional assumptions that your cell types will have equal variance. All right, so for the sake of time, I'm just going to go through 3 models, models 1, 2, and 7. So model 1 is when we use cells as biological replicates. So this is the data frame I showed earlier that shows all the features we need to incorporate. And this is what it looks like when we treat cells as biological replicates. So notice here we have 4 subjects and 16 cells. Here we're saying, okay, actually I have 16 subjects, and basically each subject is giving me 1 cell. So this not only gets rid of cell type information, but it also means that we're telling the model we have far more data than we actually do, which overfits the model and will inflate our false positive rate. So we can clearly see this in the simulated data. So on the right-hand side, I'm showing the true positives, with this pink line showing the proteins that are increasing in both cell types, and this blue line showing the X pattern proteins. And if you just look at this, you might think, actually, this is a great model because I'm detecting a lot of my true positive proteins. But if you look over on the other side, this is our false positive detection rate. And you can see out of the 980 true negatives that we have, 250 of them are falsely being detected as changing. And I also generated data sets with increasing variability along this x-axis just to see what variability does to our results. And you can see that even when we increase it a little bit, we're at 50% false positive rate. So it doesn't matter that I detected 10 true positives. They're just kind of lost in the overwhelming amount of false positives that we found. So we see this we ran this on the mouse data set too, and we're finding more proteins than we expected to find, although we obviously don't know what here is a true positive and what is a false positive, just given what we know about the model overfitting the data in the simulations. We suspect that a good chunk of these are false positives. All right, so the next model is pseudobulk. And you can see right away that we're treating subjects as the biological replicate, which is good, so it won't have that overfitting problem. But it is still averaging all the samples within a subject, so you're not going to get your cell type information incorporated, and you are combining your subject variability with your cell variability. So you can see right away, because we're not incorporating cell type, we're not detecting any of our X pattern proteins, and our sensitivity for the proteins that are increasing in both cell types is pretty low. And this is really seen in the mouse data set, where we're not able to detect any differential proteins. And we do expect, we know there are differences between the disease mice and the wild type mice. We expect to see something. All right, so finally, I will present our recommended model, which incorporates all the features of single-cell data. So it uses subjects as the biological replicate. It separates between subject variation and cell variation correctly. It incorporates the cell type, and it can support multiple hypotheses. So I talked about examples of these hypotheses earlier, but I'll just generalize the 3 types we can test. And the first one is we can compare conditions 1 and 2 for each cell type. So how is the green cell type changing and how is the blue cell type changing? We can take it a step further and compare the difference of differences. So how much more is the green cell type changing in comparison to the blue cell type? And finally, we can compare the differences between cell types. So how is green cell type different than blue cell type? These are the results of comparing conditions within each cell type using our recommended model. And you can see that now finally we are detecting these X pattern proteins, and that also our sensitivity for the increasing proteins is much higher. And these are the results on the mouse data set. So we did it for the smooth muscle cells and for the fibroblasts. And you can see in the smooth muscle cells, we didn't really find any differentially expressed proteins, whereas in the fibroblasts, we did. And this is a distinction that's impossible to make with bulk data. All right, so now that we have the model that we want to use, we're going to go on to talking about how to appropriately annotate your cell type. So there are 2 methods that people are using. One is you can use an external biomarker, which is where you have a protein that is known to be expressed in the cell type you're looking at. And so you use that marker, that protein, to mark the cell, and then you remove it from your data before going on to group comparison. And then the other way is you use all your proteins to cluster using k-means or something, and then you kind of use that marker to say, okay, I think this is my smooth muscle cells, and I think this is my fibroblasts. And then you take those same proteins you use to cluster to go to group comparison. And this leads you to a problem known as double dipping, where you are creating clusters and then using those same proteins you use to cluster to compare. So the analogy Olga likes to make is you tell tall people to go to one side of the room and short people to go to the other side of the room, and you say, are there differences between these 2 groups? And of course there are, because you design them to be different. So because you are designing these clusters to be different and then testing for differences, you are inflating your false positive rate. So again, we see this in the simulations. This top row is when we use a biomarker protein to annotate our cell type, and we get 100% accuracy. And this bottom row is when we use the double dipping problem. And even though we're finding some true positives, again, our false positive rate is so high that our true positives would just be lost in the overwhelming amount of false positives. So finally, we did this for the mouse data set. We did both methods of cellular annotation. And this side is showing when we use the external markers and then remove them. And then this is showing the k-means and then using all those again to cluster. And you can see that there's very little overlap between these 2 groups. And also, given what we know like the problems that come with double dipping and that we know it leads to an inflated false positive rate, we're fairly suspicious of the proteins that are found just by the k-means clustering method. All right, so I'll leave you with our recommendations. We recommend using a linear mix model that distinguishes cell types using external markers and not just averaging all your cell types together. We recommend you have a well-defined question because as you can see, there are a lot of questions and a lot of different models you can use. And so if you just go and use all of them, you'll get into p-hacking territory where you just go until you find an answer that you like. Also, it's important to check your assumptions, such as equal variance. And you can just leverage any standard implementation of this, such as MS-STATS, which is what we did all of this work in. All right, I'd like to acknowledge everyone that was involved in this work, Olga Vitek and her lab, and everyone in Sam Payne's lab, and finally Sarah Parker and her lab for generating the data, and of course our sources of funding, the NIH and BYU Simmons Center for Cancer Research. I will take questions.
I would love to hear your perspective afterwards on metabolomics, but maybe more general. So how do you think data structure affects what you just talked about, missing data, technical noise? Are there additional considerations?
So for technical noise will be, because I talk about your subject variability and your cell variability, and in the model, your technical noise is like wrapped up into your cell variability. We don't have a way of distinguishing those two. And then all of our simulations were done just with like 100% data completeness because I didn't want it to be like, oh, this is a missing data problem. This is a problem you would have and things to consider even if like you had 100% complete data. But yeah, I'm sure that missing data will give you trouble, so.
Would you make these same recommendations for single cell RNA sequencing community, which seems to use the...
No, yes, this is not specific to proteomics. All of this is whatever single cell thing you're doing, this is applicable to it. It doesn't matter what omics you're in.
Okay, if there's no more questions, I think we can move on. Thanks. Thanks, Anna.
Thank you.
Good evening, everybody. It's my pleasure to introduce our next speaker for the evening, Alexander Ivanov. Dr. Ivanov is an associate professor in the Department of Chemistry and Chemical Biology and a faculty fellow at the Barnett Institute of Chemical and Biological Analysis here at Northeastern University. His research focuses on ultra-high sensitivity proteomic profiling of limited samples, the characterization of proteoforms, protein complexes, and protein interactions, as well as the exploration of extracellular vesicles for diagnostics and therapeutic development. Please join me in welcoming Dr. Ivanov.
Oh, great. Thank you for your kind introduction. Thank you for sticking around. And Nikolai, thanks for your invitation. So as you may have noticed from the program, so I have the longest title in the whole conference, and I suggest that Nikolai introduces prizes for shortest and longest titles of the conference. And since I'll be introducing hyphenated technologies, so I use 5 hyphens in this title, as you may see. So this is difficult to beat. So definitely worth the prize. Okay, so I've been told that I need to declare a conflict of interest. I spun out a company or a small startup to be eligible for STTR-SBIR grants. We are applying now, and hopefully we'll explore commercialization potential for our technologies in the future. So I'll be talking about 2 directions, and Michal is here. Michal is the first one. So 2 directions in our technology developments. developments on porous layer open tubular columns. And yesterday, this term came up during the discussion after Ryan Kelly's talk. So here I'll be talking mostly about porous layer open tubular columns. And either we are using them in pressure-driven technologies for separation or in electric field-driven separations, capillary electrophoresis. I need to mention Michal, Yun-Fan, and Liz. And in the previous years, Soma contributed to data analysis, and Liz, Michal, Yun-Fan, they generated the data. So we discussed all these possible needs for single cell analysis and subcellular characterization, possible neonatal samples, small animals, animal samples, and so on. We discussed that proteomic characterization is more important or maybe more informative than transcriptomic characterization in many cases. And definitely there are challenges in characterizing proteins, many proteoforms and many peptides and proteins in small samples. So, and as Alexei mentioned yesterday, in the analysis of small sample amounts, single cell proteomics, there are multiple challenges. So at each and every step, and each and every step had to be brought to a perfection or improved, and any improvement would matter. In this talk, short talk, I'll be only discussing our improvements in separations, either LC or CE. So the point I'm trying to make, the main point, probably the main conceptual point that I'm making here is that when we decrease the flow rate from relatively low flow rate here, 64 nanoliters per minute, to 12 nanoliters per minute, we increase the signal to noise quite dramatically. Of course, it brings benefits and some also disadvantages or limitations, which I'll talk about in a little bit. But the main gains in signal to noise and sensitivity are defined by those reasons listed in the bullet points on the right. So, and in my lab, we are exploring mostly, and today I'll be talking mostly about porous layer open tubular columns, but we are using all modalities of separation columns shown here. Monolithic columns, which are polymeric sponges, polymerize inside the column, porous layer open tubular, which I'll show in more detail, and bead-packed columns, as well as capillary electrophoresis, and we are using shiftless electrospray. So the main differences between the bead-packed columns that we all are using, and the field is using bead-packed columns all the time, and the porous layer open tubular columns. So if we take a look at these TM images, and these are capillary columns, the same 360 micron OD outer diameter, the same polyimide coating. And the difference is in the inner diameter, where we are using 75 or 100 micron ID columns in the bead pack, and 5 or 10 micron ID down to 2 micron inner diameter in the plot columns. And as you see, if we zoom in into these perfectly spherical beads, some of those are not that perfect. And here we are generating a covalently attached porous layer attached to the inner wall of the capillary. So we need to operate those columns at substantially lower flow rate to benefit from the gains in their performance and to increase the sensitivity. And there are quite a few limitations currently in the field. The pumps delivering ultra low flow at about 10 nanoliters per minute or 20 nanoliters per minute are not that readily available. What we are doing, we are using regular pumps. We are operating them at about 300 nanoliters per minute or 500 nanoliters per minute, and we split the flow. To make the setup functional. Yesterday, Ryan discussed the benefits or potential disadvantages of using trapping columns. So possible losses in a flow through and possible band spreading, so the separation performance can be compromised somewhat. However, to operate our ultra-low flow columns, we cannot load sample at realistic sample volumes of 1 microliter, 2 microliter volume in a reasonable amount of time without using trapping columns. So to load our sample in the volumes I just mentioned, 1 microliter, 2 microliters, we have to use the trapping column, either bead-packed or monolithic columns. That's what we are exploring in the field in my lab. So we load them quickly, and it takes a minute or 2 minutes, depends on the sample volume. We can load up to 5 microliters, 1 microliter, whatever we want. And then we switch the configuration and we stop splitting the flow here. And we split the flow in the first T, and then we generate 20 nanoliters per minute going through the analytical load column. So that's how we operate regular conventional non-LC systems with the split flow and the valves. So, and we are making our own tips. We pull them. We have tricks here and there. So, and that's the general setup that I'll be discussing pretty much in the rest of the talk. What is new here? We published 15 years ago pretty impressive results in MCP, low zeptamol detection limits. 5 zeptamols, actually we had results down to 1 zeptamol, didn't show it in the paper, but we looked at the cells even now for cells isolated from whole blood. The numbers are pretty impressive using the equipment that we had at that time. So, but unfortunately, these columns never picked up. So they were never used broadly used in many labs. And there are challenges. So not that easy to operate ultra-low flow. So splitting is needed, then experience is needed. And also what we figured out, that you see those bumps, and if we zoom in, maybe on the next slide. So the structure, these little balls, they can be relatively humongous. We are using free radical base polymerization. And those structures, they can be detached from the column. They clog the tip, they clog the column. Sometimes we just polymerize the column and it's clogged already. It was not usable. So what we did with Michal during the last couple of years, we optimized the polymerization procedure. We optimized the morphology of the plot column. So now it's pretty smooth, very thin layer of the porous structure. And now the lifetime was expanded, the reproducibility was expanded, and usability was substantially improved. So these are the main advantages. So that's where we started. These ball-like structures, they were clogging the column. And actually, and that's the new morphology, new design of the porous layer that we are using right now. And actually, the thickness is shown here, about 150 nanometers. And what we found out, we polymerized, we used the original method to polymerize columns, and we just cut the column in several places. And we saw those structures again and again in multiple places. Now, when we do the same thing, we polymerize column, cut it in several sections, very reproducible morphology, very thin layer, no this bulky stuff inside the capillary. We do it next day, the following day, and so on. So it's the procedure is dead on, optimized, and that leads to the improved reproducibility and lifetime. So if we compare multiple runs, HeLa from peers, injecting nanogram, sub-nanogram, a few nanograms. The runs look alike for different columns, different days, different runs. If you look at the reproducibility between multiple columns, multiple runs, either number of IDs, peak shape, peak width, so the reproducibility is there. And no matter on which day, no matter when we polymerize, so that's the major improvement. And then if we compare the performance of the plot column-based setup with the trapping column up front, we use a short monolithic column here with bit-packed columns. So either commercially available or packed in-house, so in the lab. And we go from low sub-nanogram amount up to 50 nanogram level amount, proteins on the left side, peptides on the right side. And we see about 50% more or so peptides when we use the plot column for exactly the same sample, HeLa digest. And when we get to the 50 nanogram level, the gains are leveling out. We are not getting any more gains because the capacity of those columns are relatively low. So we benefit more in sub 50 nanogram levels of sample load. So that's the message. And all these tests and benchmarking of columns was done on bottom-up samples, digested HeLa, standard from peers. And then we are using the same.
What's your data?
In this case, DDA only, Lumos Fusion, 3-generation older instrument than you would use. But at least we had a good control, the same instrument, the same method, the same data acquisition. Now in timsTOF DDA, I didn't include the slides. We had 5,000, 6,000. So it has to be also benchmarked in a good way. Anyway, then if you use the same setup, we change the trapping column, and now we are looking at top-down intact proteins. We just sonicate, open the cells and spin down and inject aliquots of 200, equivalent to 200 cells. Good reproducibility, again, between the runs. In this case, we use FAMES. It doesn't matter. It can help. You may not use it. And I just have a couple of slides. Very nice peaks, sharp peaks for proteins, which is rare for intact proteins. Again, reproducible, covering quite a broad dynamic range for intact proteins, which is important. And then we are getting quite, for 200 cells, cell equivalent, in one run, we are getting about 330 proteins, 700 to 800 proteoforms, which is, again, using older instrumentation, even comparing to the leaders in the field, quite impressive. There is definitely room for improvement. And then I looked for Michal earlier today. He's working on cell-in-one and injecting single cells to the columns, but this is an older slide. He injected 6 cells at some point. We got some coverage, mostly in this case, higher abundant proteins, and I will not go into details, multiple isoforms, which are not like histones isoforms, which are difficult to detect using bottom-up approaches. Now switching gears and a few slides on using the same concept of porous layer open tubular column, but as a trapping column in C. And we were quite excited by the results we published during the last couple of years, where we used CMS to show single-cell glycomic analysis, actually surface glycomic analysis, and single-cell top-down proteomics analysis. First papers in both fields and in both directions. And Yunfan and Annelies worked on that. Also, the same method was used to characterize blood isolates. So, but now we implemented the plot column and applied it, polymerized it in the inlet of the CE capillary electrophoresis. So about 5 centimeters or so 0.57 we played with lengths. And we inject the sample, keep it there for less than a minute. It gets absorbed to the stationary phase. And then we elute in several runs. In the flow through, for example, we can look at metabolites, glycans, and then we can elute, let's say, for top-down protein intact proteins, or if you digest peptides, and then lipids. So the concept is to use that technology for multi-omic analysis of small samples. So, and the initial results, so the columns look a little bit different. This is a 30-micron internal diameter column. The morphology is somewhat similar, but different from the plot columns that I showed for LC. Unifound polymerized them. Again, no bulky structures. They're not easy to clog. And initially, we tested the columns for bottom-up proteomics, HeLa, digest, eluting in several fractions. So the flow through here, replicates for in different runs, then 10% acetylnitrile, lower hydrophobicity peptides, then higher hydrophobicity eluted at 20% acetylnitrile, 40%, and so on. Here, 4 fractions for each sample, and analyzed in 4 different runs. In the next iteration, we analyze everything in 1 run. So we inject the sample, it gets absorbed here, then the flow through is pushed a little bit from that plot section, separated by a plug of BGE. Then we elute with, let's say, 10% acetylnitrile, again, a plug of background electrolyte, another elution with, let's say, 20 or 30% acetylnitrile, and so on. So 4 fractions within the same run. And if we previously, and we published just CEMS, we had to stretch the separation to about 45 minutes to get more or less good data. Here, 1,600 proteins, intact proteins. But we had to slow down the separation to get those proteins detected. Now, when we combine those, in this case, 3 fractions, flow through, we combine with 5% elution, 11%, 40%. And these are replicate analysis of the same HeLa digest. Within the same run, about 16, 17, 18 minutes, we basically cut down the time of the analysis 3-fold. Got here an increase of 25% in IDs. Fresh data, probably 2 weeks old or so. So that's fresh from the press, not from the press, but from the lab. We are working in this direction right now. And then the idea here to move on, we kind of worked on different aspects. Look at top-down intact proteins, and we published on that. And actually, at the end, we typically see peaks corresponding to lipids. And a bunch of lipids are shown here. We didn't work yet on separating them better to increase the depth. And so I'll not go into more details. On the right side, in the flow-through and negative mode ESI, we are looking at glycans and metabolites. And so I just included 1 slide on that. We have quite a few initial data points there. So that's the concept, that's work in development. And with that, I just give you 2 choices. Either continue exploring bit-packed columns, or consider in the future plot columns as a red pill versus the blue pill. Thank you for your attention. Thank you to the funding institutions, and thank you to the lab and collaborators. Thanks a lot. That's the talk.
So with the multi-step elution, where you just have discrete concentrations of acetylnitrile, can you comment on the quality of separation without using a gradient?
You mean on a CE part, right? You're talking about elution on the CE.
Oh, I was under the impression that this was also plot columns, the last portion that you presented, that was CE?
Yes, the last
Oh, I
portion
see.
is all
I see.
that's
So you use
AC.
different concentrations just to dislodge some
Yes.
material, and then you use electrophoresis to separate. Okay, that
So
makes sense.
that part is responsible for CE separation. High voltage is applied 20 kV, 25 kV across the column after we elute.
Okay, yeah, I understand. That's very helpful. So another quick question with the plot columns, you're getting the superior performance for very small samples. And do you have a sense of how much of the benefit is coming from improved separation versus the lower flow rate and improved ionization? Combination of both, perhaps, but what do you think is most important?
I think right now, mostly from ultra low flow, definitely a combination of things potentially, but as I mentioned, trap column may distort the separation a little bit. So if you do the same experiments without considering any time delays, the separation is much better. With the trap column, we are using a compromise, but we are working on practicality. Potentially, we need to introduce some refocusing, reshaping of the band, which we played before, but we need to do that. So if we have refocusing, we can continue to benefit from the improved performance as well as the ultra low flow rate, which is translated into the better ionization efficiency, ion transfer, and utilization.
Okay, maybe another very quick question. So with the electrophoresis, where you have the plot column loading, is it something that you think you can automate? The sample loading?
Oh, it's automated. It's from autosampler. We just inject and then inject the BG from another vial, inject the elution buffer from another vial. It's all automated.
Cool, thank you.
Sorry for the confusion. I went through this fast. All right.
There are no other questions, then please join me in thanking.
Thank you.
Okay.
Yes.
Hey,
It's a good time.
one more talk for the afternoon. I hope you guys can all join me in welcoming Joel McDade. So Joel is trained as a cell biologist and with a degree from Michigan in molecular physiology. Since that time, he's actually spent most of his time here in Massachusetts, and it's currently coming to us as the technical sales director from Quantum Sci. So let's please welcome Joel McDade.
Thank you. Do a little test here. Oh, I don't want to succumb to the technical issues. All right, that's okay. I think maybe, did he have it or, let me see. That does it. All right, cool. Yeah, so first of all, thank you. It's great to be a biologist here with all of you wonderful proteomics folks. It's very humbling, but I appreciate the time that you guys are sparing to listen to my talk. And I also recognize that I'm standing between you and the bar, so I'll try to keep this short. Yeah, so my name is Joel McDade. I'm the Director of Technical Sales with Quantum SI. You know, I'm here to talk to you today about what we feel is an orthogonal technology for protein analysis. And I'll tell you a couple case studies. I'll introduce the technology for those of you that perhaps haven't heard of it before. And then I'll go into a few case studies that highlight the orthogonal value of this technology, one of which you might not necessarily think, but it's in the mRNA therapeutics space. And I'll talk a bit about that, and then I'll talk about a few papers that are probably closer to what you guys think about on a daily basis, which is how our technology couples with mass spectrometry. And then we'll hopefully have some time for Q&A. All right, so this is going to be obvious to everybody in this room. It's not going here. Hold But the proteome is complex, and this complexity brings a variety of opportunities, but also challenges, right? So on the first front, sort of proteins are complicated. There's a lot of proteoforms. In order to get through all of these proteoforms and understand their biology, you obviously have to sift through a lot of proteoforms that aren't represented at the genomic level. In the case of engineering proteins that you want for a particular function, you have a swath to choose from, but of course then you have to screen through all of these proteins through some manner, and that's cumbersome. And so, obviously, if you talk to synthetic biologists, which I'm from that background, you talk to folks about this design, build, test cycle. I'm going to design a thing, I'm going to build the thing, and then I'm going to test the thing. That's a pretty painful process, and so tools that accelerate that are of significant benefit to not only drug developers but also basic biologists. And at Quantum SI, we feel novel tools are needed to address these challenges, and we hope you directionally agree with that. And in order to really expand the toolkit that that proteomics protein analysis folks have, we launched what we call the Platinum Pro. So this is the second generation of a system that was launched 2 years ago. We position it as really an easy to use option for doing basically bottom up proteomics. So we could really teach someone new to protein work how to use the technology. So it's a great orthogonal technology. The basic foundation of how it works is shown here. So the scientists would generally start with their protein or proteins of interest. You generally enrich for those proteins, and then you digest them using a series of off-the-shelf kits that we have provided. You add a linker to the peptide library that's generated, and then you flow those peptides onto a chip. Okay. The individual peptides then sit down with their C-terminus through a biotin-strobivatin linkage at the bottom of the well, and so you have these individual peptides in an individual well. In Branford, Connecticut, we do directed evolution to come up with these what we call recognizers, which are essentially fluorescently labeled proteins that are designed to recognize a series of amino acids, and they're specific for that. And the signal that they produce when they bind is actually unique depending on the amino acid, and it's influenced by downstream amino acids. So we have a whole algorithm. I hate throwing AI into the mix. I think it's a little bit kitschy, but we generate a lot of data using these recognizers. We model off that data. The software knows what it's supposed to see, or what it's actually looking at when it sees it. And so the basic premise is you have this peptide library, you flow it onto a chip, individual wells get individual peptides, you get a series of binding events with these recognizers. You generally get tens, if not hundreds, of binding events per amino acid. So it's a very data-rich technology. So let's take our NGS hat off, right? We don't need that many reads with this technology to be confident. We'll get into more of that later. But each of these signals will vary depending on its, which amino acid it is. Here you can appreciate the intensity differences between the different amino acids. There's other differences I can get into, like how long these actually stay bound. We call that pulse duration, and also the decay curve, right? So all of these things are captured by the software. This particular thing is squished in time, so you don't get to appreciate all the kinetics, but they're there, trust me. And then this is actually taken and generated what we call a kinetic signature plot. All of this is automated, and then that kinetic signature plot is actually what's used by the software to go in and say, okay, let's look across the proteome. What does this peptide look like? Okay, and we do that all automated, right? So essentially, the workflow-wise, you set up a run virtually through your laptop, and then you load the system. The run takes about 10 hours. The data transfer is automated, and so is the analysis. So you run a primary analysis. This is really calling that kinetic signature plot, and then you run a secondary analysis, and that can be either, hey, I know what I'm looking for, please align against this protein, or hey, I have no idea what I'm looking for, please infer what that protein is against the proteome. So that's the basics in a couple minutes there. The data, what does the data look like? So we provide insights at really the protein level. So for protein level, peptide level, and amino acid level. So here we're showing PDL1 sequenced on our technology. At the time, we got about 8,000 reads that are attributed uniquely to PDL1. You see each of the individual peptides that have their own number of reads per peptide. You also get an FDR score, which is modeled off of what you folks generally would do. Again, not a bioinformatics person. I can't give you the exact details, but that's at least what I'm told. And then down all the way to the amino acid level, you actually get data on each amino acid that is recognized by the technology. And by data, I mean pulse durations, how long does that binder stay on, and you get a histogram of that. That can be impacted by things like PTMs. And so we're working through some of that biology now, but we're very excited about the value that this sort of analysis can provide, and some other stuff that I won't get into in the interest of time. But all of this is produced at a glance, at a completely automated, so you don't really have to do anything for that. All right, so that's the core technology. What are we doing with it? There's a variety of things that we're doing with it on the application front. The first story I'll tell you is about protein barcoding. And this is the idea that we can generate a highly sequenceable protein barcode that we know to be sequenceable and engineer it on proteins and perform things like pooled screens. And this is of value because sometimes those pooled screens are performed with the same exact protein or highly similar variants that are very difficult to resolve on traditional technologies. And the use case that I'll show you is mRNA. You can confirm proteins, basic QC, right? This is not necessarily earth-shattering, but to some, it beats a Western blot. You don't need an antibody for it. Things like protein-protein interactions. So we're not necessarily going to get the same amount of proteins that you guys get, full disclosure, on a mass spec run, right? But as a biologist, someone looking at protein-protein interactions, it's a pretty simple solution for that use case, and we're building out workflows for that. And then finally, we're getting really creative with it. So for instance, with our recognizers, you can actually take your favorite protein of interest, turn it into a recognizer with our chemistry, and perform binding assays on chip. So one of the things we did is we made a new binding assay using a BCL2 recognizer and some peptides that were thought to interact with BCL2 and potentially inhibit BCL2, and we're starting to perform binding assays there. You can do that on SPR, but the challenge is that with SPR you have to know what you're looking for. With this, you can perform binding and then sequence to get the identity. So that's an application that we're working on. I don't have data to share, but hopefully we will soon. And then we're applying this to, across cell and gene therapies, vaccines, antibody biologics, all the things that you'd probably expect. So in order to kind of talk you through the barcoding application, I need to give you a bit of a background on mRNA therapeutics if you don't already have it, and some of the challenges that they face. So I assume all of you have heard of mRNA LNP-based therapeutics and all the things that we're very excited about as it relates to immunogenicity, ability to deliver outside the liver, all the different possible benefits of this technology. The scientists start with a large number of potential candidates. So you can vary the lipids, you can vary the buffers, you can vary the mRNA chemistry, you can vary the cap, 3' UTR, et cetera, et cetera, et cetera, right? And the goal is really to get down to, selective reveal here, a small number of candidates that are ideally suited, right? And ideally suited meaning they get to the tissue, they translate, they produce protein, right? And they have a variety of tools to do this. So they have cell-based assays, they have in vivo mouse studies, they have in vivo non-human primate studies, right? Pretty obvious. The problem is that for these particular candidates, generally speaking, every single one of these codes for the exact same protein. So although optimizing mRNA delivery and translation is essentially the essential assay for knowing which one of these candidates wins, you can't multiplex them, right? If you do multiplex them, you're generally looking at using NGS, which paper after paper coming out showing that, hey, look, delivery doesn't equal translation. You can have a lot of mRNA there, and if it has a lot of ribosome on it, actually, hey, it gets degraded, so the total protein output's lower. So that's part of the problem with this is now you're left with this costly arrayed screen where every one of these guys is, obviously there's ethical concerns, but they're like 25 grand a piece, right? So this is not a friendly situation to be in, although you need to do it, right? So where do we come into the picture? So we had this technology that we developed in our lab, which essentially what you do is you append to your gene of interest, your mRNA of interest, a highly sequenceable protein barcode. So it's your gene of interest encoded as RNA, an affinity tag, a linker, a lyse cleavage site, and then a highly sequenceable barcode, Okay, we've gone through in this publication and talked about design and complexity and limited detection and all these sorts of things that I won't bore you with it today. But essentially the point is that we can detect these barcodes and sequence these barcodes, and each one of these barcodes is going to be unique for a given candidate. So you can pool all of these candidates together and perform an in vivo or an in vitro pooled screen with a direct protein level readout. And this is very hard to do with traditional technologies, right? And so we've published this paper. It's in the process of being formally published. It's in bioRxiv, where essentially we describe the workflow where you would take your protein of interest. One can envision other applications for this: antibody biodistribution, uptake, secretion, variety of things. But in this case, we've just defined the technology, and we have all the different components, broken out essentially. And the workflow takes about 3 hours from the time you have a sample to the time you're sequencing. And so it's very, very fast to get this data, and it's quantifiable because the output is reads, right? So it's a very nifty technology to be able to apply to some of these questions. And so we've published the actual biology work in a technical note that I'd encourage you guys to check out if you're interested. But it describes essentially the process of putting these mRNAs into an LNP, the variety of LNP candidates, mixing those together, injecting them to an animal. The barcoded mRNA, the winners, so to speak, get translated. Transcribed shouldn't be there. Sorry about that. You then extract the tissues, and then you purify out the barcodes and sequence them on Platinum, and essentially what you get is a rank order of your candidates. And so you're able to uniquely attribute whatever those conditions are with the barcode. And so you get, a pretty elegant solution. And obviously the costs on this, if you drop the number of monkeys you need by a factor of 8, you're saving a lot of loot. So it's pretty exciting. Okay, take a step back. Now we're talking about just, protein characterization. So we published a paper recently in bioRxiv. This is the Neil Kelleher paper, where we're looking at, the complementarity of our technology with something like top-down proteomics. And so we're looking at this case at IL-6, and the idea is that, we're taking a top-down approach as well as the bottom-up approach with single molecule sequencing. And so what we saw is pretty neat. We generally picked up hydrophobic peptides on our technology, and versus some of the hydrophilic peptides tended to show up on top down. Overall, the coverage was complementary. It was complementary, so we got about 52% coverage total. So again, it's just speaking to this idea that that we could be complementary with this technology. And then the final case study I'll share is with Gloria Shankman at UVM, University of Virginia, and she's interested in tropomyosin, which is actually a gene I studied, ironically. But this has a lot of heterogeneity between these tropomyosin isoforms, but they're highly similar. And so some of them will actually cause disease, even though they're just point mutations. And some of the point mutations are things like L to I substitutions, which are not necessarily ideal for mass spectrometry based, though you guys, I'm sure, will argue with me about that. But similar masses equal, similar readout. And we're able to discern some of those on Platinum because we don't care about mass necessarily, right? And so we're also starting to look at things like phosphorylation on the technology that's shown in the paper. And so I'd highly recommend you guys take a look at that as well. This is for those of you that have been following us. This is kind of like just a snapshot of where we've been and where we're going. So, we launched when we published this paper in Science back in 2022, sort of came to market in December 2022. I joined right around, I think it was like somewhere in 2023. And then we very quickly since then have launched, we've doubled the number of recognizers. We've quadrupled the amount of occupancy on the chip. We've launched several new kits improving the chemistry. We've launched a new system. We've launched new software. The technology is really, really, really moving quickly. I haven't been at a company where the technology moves as quickly as this does. And so we're very excited about where we are today, but of course where we're going. And we would love to have you guys on board as if you ever want to test the system, anything like that, just let me know. With that, I'll take any questions, and hopefully I'm under time as promised.
Thank you very much. We already have.
I have a couple of quick questions.
Sure.
How many amino acids do you have recognizers at the moment?
Yep.
And can you give us some rough order of magnitude estimates for the specificity of these recognizers? You mentioned they're specific, but everything has finite specificity. So what is the range?
So I can tell you we have direct recognition of 13 amino acids at the moment. Like I said, was 3 2 years ago, and now six recognizers recognizing 13 amino acids. I can't speak truthfully, like I can't speak to how specific they are. And how much real space we have there. I can tell you, we have intensity to work with, we have pulse duration, and we have decay. So there's 3 variables that give us.
Oh, yeah. So there are two levels of specificity. One is detecting the optical signal and being able to associate it comes from this recognizer. The other is a particular recognizer being specific for its cognate amino acid, because it might have finite affinity to another amino acid. So there is both the biochemical level specificity and then the optical detection specificity.
So I don't have a good answer for that, truthfully. Sorry. I can follow up with you if you'd like, though.
Two other very quick questions. Do you have commas? Essentially, when you have a dark amino acid without a recognizer, can you be confident that there is a spacer in that particular position?
So we do have measurable and reproducible effects of downstream amino acids, the sequence context on a particular recognizer. So we can start to infer what the invisible amino acids are. I can tell you.
But can you say how many of them there are? Let's say.
So it's stochastic, right? So all this is happening in a single pot, right? And we don't have necessarily, because it's stochastic. You may clip off an amino acid at point number 5 at 5 minutes. You may clip off an amino acid at 10 minutes, and all throughout that time, you're going to essentially look at a blank. So it becomes a bioinformatics question of really like, it's essentially a rolling frame, right? And so we can't confidently tell you because we don't have microfluidics, right? So it's just kind of the way the system's designed.
Okay.
We don't know exactly when the cut happens.
Final quick question. So if you digest representative human proteome with lyc and you look at peptides within the length range that is optimal for fingerprinting, quantum psi, what fraction of those peptides might have sequences that are very difficult to fingerprint regardless of their abundance?
I can tell you the typical talking points that we have are generally like, the sweet spot of the technology is somewhere between 10 and 20 amino acids. The lower limit of that is driven by the fact that the shorter it gets, you'll still see it on the technology.
But let's say you're in your ideal spot.
I
can tell you that, for example, in mass spectrometry, we can be in the ideal length spot. There might be peptides that ionize very poorly, so our sensitivity would be low. They fragment very poorly. It is difficult to identify them. Similarly, I can imagine their sequences that are just much harder to find quantum size. So that's what I'm trying to
that's where I'm getting. So at the lower end, I think what you have to deal with is the shorter the peptide gets, the less likely you have a unique peptide, right? So what we'll see is we'll generally sequence them quite well. So anywhere from 1 amino acid up, you will see those species on chip without a doubt. The challenge is then bioinformatically, how do you can't confidently say that's attributed to any protein in particular, right? We generally say you need 4 recognition segments. So that's 4 constant stretches of where you're confidently saying a recognizer is binding, and 3 unique recognizers in order to bioinformatically say, yes, this is something that we can align to, right? On the other end of the spectrum, you have potential secondary structures because proteins like to fold. I'm not aware of a hard upper limit that we've identified. I can tell you anecdotally what I've seen, and we can sequence quite long peptides, but I don't have a robust sort of map of what we can and can't see. All of it's digested by lyc, right? And so you're sort of at the whim of lyc, which you probably know better than I do, in terms of what you produce on the back end.
Thank you.
Yeah, thank you. Yes.
Was intrigued by the tropomyosin data that you presented. So first question is, in that approach, are you specifically enriching for tropomyosin?
It is enriched. It's recombinant. Yeah.
It's recombinant.
Some of them are synthetic peptides, and we've worked with recombinant proteins.
Okay,
yeah.
so thinking about instances where we have splice forms of proteins, do you have a sense of your prospects for pulling those out of complex mixtures, and then also the ratio between, let's say, spliced and unspliced that you need to have good, calls on that sort of thing?
We've recently done that gymnastics in a quite detailed manner, and I think the point of it is it's an emerging application for the technology, but in truth, I think we really need more coverage and we need more sequencing depth to be able to confidently pull it out of. It's going to be protein dependent, right? Is the reality when you're dealing with the type of coverage that we have. So we've seen cases, for instance, with pharma clients where they have a very high value variant that I won't get into detail, but like very high value variant that they need to map and they need to be able to confidently call. And in those cases, there are instances where our technology does quite well with that. And we can a priori, we have an in silico prediction tool that we can run all these proteins through and find the variants that work. But I would not position this today, truthfully speaking, as a broadly useful variant detection technology. I think there's some dependencies with where we're at this particular moment. Yeah. Yeah, of course.
I have one last question.
Yeah, sure.
For you. So the time scale of these can, depending on the application, be many hours. 10 hours was the one you mentioned for a full lysate. Is that right?
It's for a standard sequencing run. Yep.
Standard sequencing. And that's driven by the kinetics of each of the individual recognizers. Is there any way to accelerate that?
Yeah, you can run it for less time, but the reality is you may miss some of those longer peptides in the acquisition. So for instance, so it's stochastic. We know that the immunopeptidases will cut every 10 or so minutes. So you need to assume that across the proteome, lyse cuts and generates a peptide of a certain length. You need a runtime of a certain time that allows you to confidently capture all of those amino acids. But you can set it to as short as you want. So for instance, with the barcoding application, because they're so uniform and they're so short, you can most likely run it for less time and still get the data acquisition out. So it's a trade-off, right? Yeah.
Thank you very much. Let's thank Joel one more time.
Thank you, guys.
All right. Well, as our last bit of business before we adjourned for dinner, I and the poster evaluation committee had the extremely difficult task of choosing the winners of our best poster competition. And before I announce those winners, I do really want to emphasize that it was a very difficult task. There was a great breadth of really interesting science that I thought was very well presented by all the presenters. So I think it would be nice to give all of the presenters a hand because I think they did an excellent job. So with that being said, we have two awards for best poster, and you can come up and receive your award. And then Rachel, who just left the room, she's waving goodbye there. You can see her at the desk outside to claim your prize. So our first poster award winner is Ron Zhang. And our second poster award winner is Ramel Gletton. Congratulations. And I'll turn it over to Nikolai for some concluding remarks.
So this was a lot of fun for me. I love the diversity of topics covered. I love how interactive and friendly this community is. I certainly feel that's a very safe environment to ask sometimes not all that enlightened questions, but learn from what has been presented and brainstorm for new ideas. And for me, over the last 8 years, this conference has served as a yardstick for the progress of our community. And when I think of the early days when we started in 2018 and the progress along the way, I see it's been really remarkably fast and it's not slowing down. If anything, it's accelerating. Now we have technologies that are producing much more quantitative data at larger scale. So there is ever increasing focus on the biology that we can do with the technology. And I think that's very exciting. It's ever increasing scale of both the depth of the proteome, the throughput, and I can easily imagine next year, speaking of throughputs, that are going to make our current high throughput studies appear very modest and rather slow. And I trust that our community and our friends around the world are going to contribute towards making this a reality. So it's wonderful, wonderful for me to be part of this journey. And I want to thank all of you for enabling it, for coming here and sharing your results, discussing openly, contributing to our collective developments, making this field so vibrant, because I do believe deeply that the proteome remains unexplored in very many ways in terms of the depth, sequences, modifications, interactions, temporal dynamics, all of that. There is a big frontier out there that together we can start making progress towards. So thank you very much for all of that. And I look forward to seeing you certainly next year, but also in between in various other occasions and interactions, and look forward to helping you adopt technologies that have been shared here and building our community. Together, we are stronger. Thank you. So the next thing on the menu is going to the bar. We are going to go to 5 Horses, which is a short walking distance from here going through the main campus. We can assemble outside and walk all together. You're welcome to join the main group and come with us, or if you want to stop by quickly your room and then meet us in 5 Horses, that's another option. So I look forward to another fun dinner with more discussions.